Well, first of all, thank you very much for the organizers, Dr. Afroz, and the chairs who are sessioning my talk. Thank you for the kind invite. This is my second visit to Srinagar. 11 years back, I came here with my family. And this time also, I'm coming with my 2 kids. And it's been always pleasure to come here. It's a beautiful city and really means, those who are living here, I feel that they live in paradise. Nothing can be better than this for the living. So, and at the same time, the great science is happening here. I think a lot of good research happening in University of Kashmir, in SKIMS, and many other places. We always read the papers, publications coming from this place. So I'm really feeling very honored to give this talk here. And I know this is an Immunocon conference, and most of the speakers are immunologists, but I'm going to speak a little bit a different topic, which is called cancer cachexia. But one thing which I can ensure you that all the molecules, the cytokines, and the pathways which you study in immunology, they're also very relevant. So it's a different aspect of immunology, that same kind of molecules, what they can do to your body, and how important they are, the important players they are in muscle biology and this cancer field. So the talk I'm going to talk today is about the ER stress, and I have several introductory slides because I know that many of you may not know this field. So I'm going to give you 5-7 slides to the good introduction about what is the problem, and then I will present some of our recent findings in this area. So the cachexia, many times I give a talk and I ask the audience that, have you heard this term before? What is cachexia? And many of them, they say that, oh no, we never heard this. And it's not a surprise, cachexia, because this is a term which is not so popular, but it's getting popular because of its importance, and especially for the cancer patient. So just to tell you that what is cachexia? Cachexia is a Greek word. It is coming from the 2 different words, kako. Kako means bad, and hexia. Hexia means physical state. So it's a bad physical state. So what is cachexia? It's basically a multifactorial, life-threatening metabolic syndrome that mostly involves the loss of muscle mass and sometimes fat mass as well. And I don't know if you have noticed or not, many cancer patients, when they progress in the cancer, you see that they lose their body weight. They become very thin, and most of this loss in body mass or the body weight is happening because of the loss of muscle mass and sometimes fat also. So 50 to 80% of all cancer patients, they undergo cachexia. And cachexia itself is responsible for 20 to 40% of the cancer patient's death. So this is really a very important problem, and that's why in US and in European countries, and now it started happening in India also, that people are paying a lot of attention to this disorder, because this determines the success of your chemotherapy, radiotherapy, and all kind of immunotherapy, even as you know that most of the time the medicine or the chemotherapy is given based on the body mass. If somebody cannot tolerate that chemotherapy, that means the person is going to not be treated for the cancer and the person dies. So maintaining body mass during the progression of cancer is very, very important, and we study those mechanisms. So as you can see from this slide here, that many cancer like lung cancer, liver cancer, pancreatic cancer, colorectal cancer, 50 to 80% of these patients, they undergo this cachexia. So the cachexia has different stages. So initially the person is normal, and after that, they start losing unintentional weight loss. And when the weight loss is around 5% and they just stop feeling hungry, the anorexia happen. So that is called pre-cachexia stage. After that, when the weight loss is more than 10% of the initial lot, we call it cachexia. And again, the anorexia, which is loss of appetite, it is associated with cachexia. And after some time, this becomes refractory cachexia. That means there is a severe body wasting, and you can do anything. Generally, the patient is going to die in few weeks to months after that stage. But when somebody reaches to refractory cachexia, you cannot do much, but there is potential to intervene at pre-cachexia and cachexia stage. So as I said, the preoperative cachexia is associated with poor survival of patients with many types of cancer. And because that, as I mentioned, that all the therapies are given based on the body weight, and if you don't have body weight, you cannot do this. And if that much of dose is not given, you cannot stop the cancer to grow. So the most important thing which we have to recognize that while we understand that this is a very important problem and it has to be treated, but there is no drug at this time. There is one drug which was recently tested. Its name is Ponsigram, and this is like antibody against GTF-15. This has shown very good results in the phase 1 and phase 2 clinical trials, and is progressing with the phase 3 clinical trials in Pfizer. So cancer cachexia, I want to give you some introduction about it. So what happens when the tumor grows in the body? The tumor produces various factors, and many of them are pro-inflammatory cytokines like interleukin 1, TNF, TWEAK, interleukin 8, and all these molecules. So these cytokines, although they are immune origin or from the tumor origin, but muscle has receptor for this. These cytokines, they bind to the muscle and they activate some signaling pathway that leads to the activation of various proteolytic pathways that degrade the muscle within the muscle fibers, and that leads to the loss of muscle mass. So the same thing, tumor also, produce some other factors like parathyroid hormone and TNF-alpha. They work on the adipose tissue. They change like from the white adipose to the brown adipose, and brown adipose are like more energy consuming kind of tissue. They causes like basically energy consumption much faster than the white adipose. And also there is a lipolysis where the fatty acids, they are hydrolyzed and these are taken up by the cells. And then, I don't know if there is this pointer. Yeah, oh yes, it can help. So these free fatty acids, when they are taken up by the muscle cells, that also leads to muscle wasting or muscle atrophy. Same time when the tumor is grown in the body, our immune system become active. It tries to basically stop the tumor growth, but the activated tumor cell itself can produce factors like TNF or TWEAK, and they can basically aggravate the muscle wasting. Same thing, there is inflammation in the gut, and there is a altered production of ghrelin. Ghrelin is a very important hormone which is like important for the appetite. So if the brain develops ghrelin resistance, and that causes anorexia. Same time, these pro-inflammatory cytokines, they can also do cardiac muscle wasting through the activation of this pro-inflammatory transcription factor called NF-kappa B. So my lab is traditionally interested in understanding this. Just a minute. Yeah, so our lab is focused on this aspect of muscle wasting. We have like this cytokine called TWEAK. This was identified in our lab, and we showed that it is a very important muscle wasting cytokine. And we also study these other mechanisms like the proteolysis and denervation and all that. What are the underlying mechanisms and how they cause muscle wasting? So I just want to give you a little bit more idea. We all know what is our muscle. We all have a lot of, the muscle in the body. But, what happens is that in many conditions like, as cancer growth is one, and during aging, people lose their muscle mass. You can see in this picture that the right side person does not have that much of muscle compared to the left. It has happened because of the muscle wasting. But one thing which is important to recognize is that during these conditions, there is no loss in muscle cell. The number of cells remain the same. It's only the mass, which is the proteins which is filled in the muscle cell, that becomes degraded and the muscle becomes thin. When the muscle becomes thin, it has like reduced power, strength. That means the ability to perform the work is reduced. So in muscle field, we know it very well that once the muscle is like the person is grown up, like in the adolescent stage, the number of muscle cells are fixed. So it's just the increase in the muscle mass or decrease. If there is an increase in muscle mass, we call it muscle hypertrophy. And if there is a decrease in muscle mass, we call it muscle atrophy. But if you look at like the normal muscle, we basically, as we have these muscles, we make a section, and you can, look at the cross-sectional area or the diameter of the muscle cell. You can see here in the center, there is the normal muscle, and in the left side, there is muscle wasting or called atrophy. You can see the number of cells are same, but the surrounding areas around the muscle is filled by the fibrosis and collagens and like this. But when there is a muscle growth, which is called hypertrophy, again, the number of cells are same, but your diameter or the size of the muscle fibers is increased. And I want to tell you that the muscle mass, as I said, is maintained by the balance in the protein. Like, that there is always a rate of protein synthesis happening, but at the same time, there is protein degradation is happening. So in normal person, this is basically a delicate balance is there. But in many conditions, like when you do exercise and eat good food, the protein synthesis is increased, and this leads to muscle hypertrophy. Whereas in conditions like aging, cancer, the rate of protein degradation is significantly higher than the rate of protein synthesis, and these cytokines, all they basically work, and then that's how you lose the muscle mass, and you lead to the condition called muscle atrophy. So in my slides, I'm going to show you many times these kind of sections with the muscle. So basically, what you would see is that compared to the normal, when we see a decrease in the cross-sectional area or the diameter, there is a muscle wasting or muscle atrophy. That's what we mean. So I want to introduce another term, which is called endoplasmic reticulum and ER stress. So as we know that endoplasmic reticulum is a basically membranous structure which is around the nucleus, and this we have rough endoplasmic reticulum and smooth endoplasmic reticulum. The main function of ER is the protein synthesis, protein folding, modifications, and in smooth endoplasmic reticulum, it is involved in the lipid synthesis. Again, the ER is very important place for calcium storage and intracellular transport. In most of the case, this ER functions are maintained, but there are many conditions. For example, when you have a mutated protein that is not able to fold, and we call it like the misfolded proteins, when they accumulate in the ER, or there is a metabolic or nutrition stress, or there is disruption in calcium homeostasis, oxidative stress, and all those, even chemotherapy and radiation therapy, they can disrupt this homeostasis. When this homeostasis is disrupted, the ER develops stress, and with the condition which we call ER stress, the ER stress, it leads to the activation of a very highly coordinated signaling network, which we call unfolded protein response. And the main function of this unfolded protein response is an adaptive thing, is that it produces molecules or the chaperone proteins which can improve the folding or which can basically restore the homeostasis in the ER. But when there is a continuous activation of ER stress or unfolded protein response, the same pathway can be maladaptive, and that can lead to the disease state. So we have been studying this, the role of this unfolded protein response in our lab for past 10, 12 years in various aspects. Today I'm going to talk about cancer-induced muscle wasting, but we have also studied other aspects of ER stress and unfolded protein response in muscle biology. So just to give you an idea that this is what is the normal case, like the top thing here, you can see there are three proteins, ATF6, IRE, and PERK1. These are the transmembrane protein. They are present on the endoplasmic reticulum. So when there is no stress, there is a protein called BIP. This protein binds to these sensors, and they are inactive. Nothing is happening. The protein folding, everything is going on perfectly fine. But when there is an accumulation of misfolded protein, you can see here in this cartoon, this BIP protein, basically they go detach from these sensors, and they go bind to the misfolded proteins here. So when these sensors are not bound to this BIP protein, they becomes activated. So like ATF6 is one of the sensors which which goes to the Golgi, where it undergoes like N-terminal cleavage and becomes an active transcription factor. Then IRE1 alpha is also undergoes autophosphorylation, and it has two kinds of activity. One is the endonuclease activity, where it basically causes alternative splicing of a mRNA called XBP1, and when this alternative splicing happens, it basically leads to the production of mRNA, which translates into protein, which is a very active and potent transcription factor. At the same time, the IRE1 has a kinase activity, and this kinase activity can lead to the activation of JNK, and which can induce autophagy and apoptosis. So there is a third pathway which is driven by this protein called PERK. So PERK is also activated in this similar fashion as the IRE1. It undergoes autophosphorylation. This main function is to phosphorylate this protein EIF2 alpha, which basically inhibits the protein synthesis. So basically not many new proteins will be produced, so that there will be less stress on the ER to do the folding and other kind of things. But at the same time, this PERK arm of this thing leads to the translation of a transcription factor which is called ATF4, and ATF4 basically produce like various proteins involved in the chaperones, which are important for maintaining the homeostasis of the ER. So we have studied the role of the PERK arm of the UPR, and we have also studied the IRE alpha and XBP signaling in skeletal muscle. So I'm going to show you our data, about this. So, this is the first thing which we did long back. We had like a model of cancer cachexia. This is called Lewis lung carcinoma. This study was published in 2016. That means it's almost 10 years old now. So we found that in the skeletal muscle of this LLC tumor-bearing mice, if you compare with the control, there was an increase in the expression of many molecules which are involved in the ER stress or UPR. So this was not limited to Lewis lung carcinoma. We had a model of colorectal cancer as well, and we found the same kind of phenomena that this expression of these molecules is increased here. And that gives us a hint that the ER stress or unfolded protein response are activated during the cancer-induced muscle wasting or cancer cachexia. So first thing what we did is that we used a molecule which is called 4-phenylbutyric acid. This is a chaperone. It's a chemical chaperone. It binds to like the misfolded proteins, and it is very important, potent inhibitor of the ER stress and unfolded protein response. So what we did is that we grew the tumor in the mice and treated the mice with this 4-PBA every day for 18 days, and then we collected the muscle and analyzed them. So surprisingly, what we found that when we treated the mice with 4-PBA, there was a decrease in the body weight by PBA treatment alone. And Lewis lung carcinoma decreased the body weight as well, but when there was a combination, there was even a more decrease in the body weight. And same thing is the strength. The muscle strength was reduced with the 4-PBA as well as with the cancer and cancer plus 4-PBA. So we basically, this is again a slide. I told you that this is how we measure the diameter. This is the gold standard for studying the muscle wasting. And you can see here that when you treat it with the 4-PBA, we find a very good decrease in the cross-sectional area. Cancer also reduced it, but when there was a combination, this is basically more reduction in the cross-sectional area. That means more muscle wasting when you inhibited the ER stress or UPR pathway using this PAN inhibitor, which is called 4-PBA. We did some mechanisms, and we found that the protein synthesis was inhibited through this AKT-mTOR pathway. So, this data showed us that total inhibition or just, pharmacological inhibition of ER stress UPR is not a good approach. It's not going to work. So, but we know that there are 3 pathways, and they may be important in different aspects of it. So then we started teasing out the role of the individual pathway. So first we studied the role of this PERK, which is one of the pathway which is important, for the protein synthesis inhibition in response to ER stress. We got a mice which is homologous for PERK. We made a conditional knockout where we can delete PERK only in the skeletal muscle, and then we treated this is like inducible knockout mice. We treat the mice with tamoxifen, and then we perform our experiment. So what we found was, again, the similar thing what we found with the PAN inhibitor, that when we deleted this, the PERK in the muscle cells, there was a reduction in the muscle strength, which we measure by the grip strength. And there was also a reduction in the force production, suggesting that there is muscle wasting happening just by deletion of the PERK. We did experiment where we basically grew the tumor LLC Lewis lung carcinoma in the mice, and we found almost the same thing that, the deletion of PERK basically causes muscle wasting, and in presence of tumor, it even further aggravates the muscle wasting. So that means the PERK arm of the UPR is not a target for cancer cachexia, and it is important to maintain the muscle mass in normal condition. So after the PERK, this is the PERK arm which we showed, but the another important arm is this IRE XBP signaling pathway. And, we did not have IRE1 flox mice at that time, but we had XBP1 flox mice. So we did the same thing. We basically generated mice in which we can delete the XBP transcription factor specifically in the skeletal muscle. And we performed similar kind of experiment. We put the LLC tumor in these mice, and at like a certain stage, like 20 days, 21 days after, we take out the muscles and then made the cross-sectional areas to see what is happening. As you can see here, this is very clear that this is the normal mice, but when you put cancer, the Lewis lung carcinoma here, there is a reduction in the cross-sectional area, but this reduction is much less or almost negligible when you have the muscle-specific XBP knockout mice. And this is the quantification. So this gave us an idea that this IRE XBP arm of the UPR is basically causing muscle wasting, not the PERK arm of the UPR. So we went ahead and recently we performed a study with the pancreatic cancer, model. This KPC, which is a PDAC cancer model, this causes very strong muscle wasting in the mice, and it is being studied very well. And a lot of labs use this model because the results are very reproducible. And so what we did is basically we took the mice first, and then we injected the KPC in the pancreas of this mice. It is an orthotopic model. And after like 21 days, we collect the muscle and we analyze them. So what we found is that, this is the RNA sequencing data here. You can see the normal PBS and KPC, many molecules which are related to the IRE XBP arm of the UPR, they were highly upregulated in the skeletal muscle of this KPC tumor-bearing mice. And we did this, experiments by independent experiments, qPCR and all that. You can see that ERN, which is IRE, XBP1, and some of the downstream target like the DNA, JB, and all them were highly upregulated. We also did Western blot, which further confirmed that the IRE1 and the splice XBP1, their level is increased in the skeletal muscle of KPC tumor-bearing mice. And this ubiquitin conjugated protein, LC3B, these are basically the proteins which are involved in muscle degradation. So this confirms that the muscle wasting is happening here, and at the same time, the components of this IRE XBP pathways were highly upregulated in these muscles, So we did the same experiments. Again, we made a muscle-specific XBP1 knockout mice, and we did like our standard experiments. We grow this KPC cells in these mice, and then compare the grip strength. So you can see here that this is like the flox mice, this is the knockout mice. There is no difference in naive condition, but when you put the tumor there is a decrease in the grip strength. And when you have this knockout mice, there is a not that significant decrease from here. And if you compare these, the knockouts, the muscle strength is preserved there. And this is the body weight, like there was much less decrease in the body weight in the knockout. This is like individual muscle, like tibial anterior, GA, soleus. We do this kind of experiments all the time. This is the percentage decrease in the wet weight of the muscle. And you can see here that there is much less decrease in the wet weight in response to tumor growth in the knockout mice. And there was no difference in the tumor weight, that this, whatever protection is happening is not because of the tumor growth, it's just because of the signaling within the muscle. And this is, again, I mentioned to you that, this with KPC, you can see a very nice decrease in the cross-sectional area here. But if you look at the knockout mice, that KPC-induced muscle wasting or loss of cross-sectional area is more or less preserved. And this is reflected here in this quantification. And if you look at the percentage decrease in the cross-sectional area, you can see the knockout mice are more or less protected. There is hardly any decrease in the knockout mice. So once we've confirmed, and we did a lot of cell culture experiments where we use conditioned medium, treat the cells, and we found the same thing, that if you inhibit this pathway, we can protect the muscles from undergoing degradation or the protein degradation. So then we started looking at the mechanism, and there are many signaling pathways which are implicated in muscle wasting in response to cancer. So some of them are like TNF and TWEAK. They are pro-inflammatory cytokines. They basically activate NF-kappa B, which then induces the transcription of some of the molecules of ubiquitin proteasome pathway and autophagy. Same thing is interleukin 6 pathway is very important. It functions through the STAT3, and then the myostatin, activinin, and TGF-beta signaling pathways. This leads to the activation of this SMAD signaling, and this SMAD signaling leads to, again, these are transcription factors. They all go in the nucleus, bind to the promoter region of various genes, and most of them are E3 ubiquitin ligases and autophagy genes, which are important for muscle protein degradation. So what we did is that we first investigated what is happening to these molecules, and what we did is that we performed the quantitative PCR, analysis of the control and knockout mice, skeletal muscle from there, and in response to KPC condition, KPC tumor growth. You can see here that in the KPC case, there are the many molecules or pathways which are involved in the catabolic processes. This was reduced, whereas those involved in the negative regulation of protein ubiquitination, their expression was increased. And these are some of the muscle-specific E3 ubiquitin ligases. We found that by deletion of XBP in muscle, their expression is reduced. And the top two molecules are the components of ubiquitin proteasome system, and the bottom two are the autophagy system. This is Western blot further confirming that whatever we see with the real-time PCR, that there is an increase in the ubiquitin conjugated protein in the control mice in response to tumor growth, but that the same kind of increase is not happening in the knockout mice. And this is just the quantification of this Western blot. So another thing which we found, and it is well known, that in the case of the muscle wasting, there is an increase in the activity of interleukin-6 JAK-STAT signaling pathway, and we found with this real-time PCR analysis, the RNA-seq analysis, that there is an increase in the activation of this interleukin-6 JAK-STAT pathway. But we wanted to know whether if the deletion of XBP1 does anything. And you can see here that most of these molecules, which are components of this pathway, are increased in the normal mice, but in the skeletal muscle of the KPC tumor-bearing mice of the control mice, but in the knockout, the expression of these molecules was reduced. And we did some Western blots. STAT3 is the most important molecule in muscle wasting from this family, and its phosphorylation at this Y705 is very important for its activity. And you can see here from this Western blot that in normal mice, there is an increase in the STAT3 in response to tumor growth, but this was almost completely inhibited in the knockout mice. So in the field, this is also known that tumor cells, they produce some molecules, and especially the cytokines, and these cytokines, they go in the circulation, and then they work on the muscle, and what they do is that they increase the rate of fatty acid oxidation. And when fatty acid oxidation is increased, that leads to the production of oxidative stress from the mitochondria. That itself is toxic for the muscle. It can cause muscle wasting, but at the same time, it can activate some signaling pathways such as P38 MAP kinase that can cause muscle wasting. So we wanted to know, so one of the pathways which we found was that this fatty acid oxidation pathway was activated in the tumors of this KPC, in the KPC tumor-bearing mice, but when we deleted this XBP1, there was a drastic inhibition in the expression of many of these molecules. You can see here the last 3 lanes and these 3 lanes. This was inhibited. It's not only that this pathway we looked at across the what is known in the field. We looked at many pathways, but this was another thing which we found was significantly inhibited in these mice. This is the real-time PCR which confirmed that yes, if you delete the XBP1 in muscle, there is a reduction in the expression of many of the molecules involved with fatty acid oxidation. This is the enzymatic measurement of the fatty acid oxidation activity, and this is a assay which measures the carboxylated protein or irreversibly oxidized proteins. You can see in the flox mice there is an increase in this thing with the tumor growth, and this was quite significantly inhibited in the knockout mice. So we found this thing, XBP1, is a transcription factor. It can bind to the promoter region. It can, manipulate, or it can increase or decrease the expression of some genes. So we analyzed the promoter of many genes which are involved in the protein degradation during cancer cachexia and muscle wasting condition. And, these are the site where XBP can bind in the promoter on the DNA sequences. So what we found that LC3B, which is an important component of autophagy, and its own promoter XBP1 can bind it. And this is another site where some of the E3 ubiquitin ligases and like interleukin-6 and PDK4, these molecules are involved in muscle wasting, and we found that XBP1 has consensus binding site in the promoter of some of these molecules. We did some chromatin immunoprecipitation assays, and we confirmed that yes, the XBP1 binds to this box 32 LC3B, ATG, and XBP itself. This is in vitro data, and here is the in vivo data. Even interleukin-6 is regulated by XBP1. So anyway, we did a lot of other experiments to prove that XBP1 is directly, basically inducing the expression of these molecules during cancer cachexia. So finally, we did an experiment in which we wanted to find out that, okay, well, if you delete the XBP1 or inhibit IRE1, there is a protection in muscle wasting in response to KPC tumor growth, but can we do it pharmacologically as well? So we use this molecule which is called 4-mu-8C. This specifically inhibits the endonuclease activity of IRE1. That means the XBP1 production, splice XBP production is inhibited. So we injected mice with the normal mice with this KPC tumor, and then after 10 days, we started treating them with this inhibitor every day. And around 18 or 19 days, we collected the muscle and analyzed. And you can see here that the grip strength, there was a decrease with the KPC, but when you treated with the mice with 4-mu-8C, there was a significant improvement in the grip strength. And same thing with the wet weight of the muscle mass, the percentage decrease was much less when it was treated with the 4-mu-8C. And there was no difference in the tumor weight. And this is our standard experiment, the gold standard for measuring muscle wasting. You can see that there is a significant decrease in the cross-sectional area with the KPC tumor growth, but when the mice were treated with 4-mu-8C, that was not happening in this case. And this is the quantification, again showing that there was a protection in muscle wasting, and that the decrease in cross-sectional area was much, much less here. The biochemistry showed almost the same results which we got with the genetic mouse model. There was increase in ubiquitin conjugate proteins and other components of ubiquitin proteasome and autophagy, as well as the STAT3 phosphorylation that was increased, but treating with this molecule 4-mu-8C blocked that activation. So I think that's it. Here is the conclusions we drew from this study, that IRE-XBP signaling is activated in skeletal muscle during cancer cachexia. Genetic and pharmacological inhibition prevents or attenuates the loss of muscle mass in tumor-bearing mice. And the main mechanism through which IRE-XBP1 works is the JAK-STAT signaling pathway, fatty acid oxidation, and that XBP1 is capable of directly binding to various molecules involved in protein degradation in the skeletal muscle during cancer cachexia. If you look in the cartoon form, this is the tumor cells, this cachectic environment is there. It basically increases the activation of this IRE-XBP1. It can directly induce the components of this proteolytic system, or it can activate, induce interleukin-6 production that can lead to the activation of JAK-STAT pathways and other things which causes the muscle wasting. So with this is my lab at University of Houston. We are a small lab. I want to thank, and most of this work was done by Aniket Joshi. He was my PhD student. He moved to a postdoc now. And I also want to thank the funding agencies which helped this project. But before I end, I want to advertise my conference as well. So we are doing a conference in Houston in February next year. This is a cachexia conference, and it's an international conference where you will meet the people from 10, 12 countries. We did it last year, first inaugural conference, which was very successful. But we are repeating this conference, and if somebody has interest in cancer cachexia field, please, reach out to me. We'll try to help you to bring to Houston, and give us the chance to do your hospitality as well. Thank you very much.
Too. And I enjoyed it tremendously, being basically a biochemist. So linking the biochemical events, so biochemical events linking with the cachexia and muscle loss. So I enjoyed it like anything. So you want something?
Yeah. First, I would like to open the forum for the question. Please, 1 or 2 questions.
Yes. Yeah, very good question. So we find, you're right, that we saw increase in the CHOP level as well, which is regulated by the PERC arm of the UPR. So many times CHOP and XBP1, they work together, and CHOP can also be activated through other mechanisms. So when we did the Western blots for phosphorylated PERK in the same experiment, we did not see any increase there. So CHOP is increasing, but I don't know if that is contributing to muscle wasting or not, but there are like different kind of reports. Some says that CHOP is there to protect the muscle. So it might be a compensatory mechanisms to help prevent it. And the second question was that how the XBP-IRE1 is activated. That's a very important question. As you know that this is ER stress is developed because of there is a perturbation in the things in the ER. And you know the cancer itself is a very active process. You know the muscle wasting itself is a very active process. You have to make various proteins. In fact, even the proteins which are involved in protein degradation, you have to synthesize them. So there may be a very high demand for protein synthesis that leads to the activation of this thing. And this demand is not ending, you know, it's continuous because the cancer is there continuously. So it is possible that this is the reason for its activation. But we published a paper before that the tumor-derived factors, they can also activate it independent of ER stress. So the toll-like receptors, we studied it. Many toll-like receptors goes up in this thing, and we deleted MyD88 as a downstream of this thing, and we found that it protects muscle wasting. And one thing it does is that it inhibits the activation of spliced XBP1 as well. So both of the mechanisms are possible.
Yeah, a beautiful talk. I was wondering, in fact, it's a naive question. Does this axis also work in this age-induced muscle wasting? I mean, if you restrain this XBP-IRK axis, do you think that if you restrain it, then it will restrain the inhibited, then it will restrain this age-induced muscle wasting as well?
So you are saying that there is any similarity in age-associated muscle wasting
Yes.
and this thing. So, you know, that's a very normal question. It's a good question because the mechanisms are more or less, it's not always the same mechanisms, but protein degradation also happens. But the age-associated, the different mechanisms are involved there. So IRE XBP1, we looked in the aged mice and there is not much change there. So aging-associated muscle wasting mainly happens because of the inhibition in protein synthesis, whereas the cancer-induced muscle wasting happening because of the increase in the protein degradation. And at the same time, various different molecules are involved. In cancer, you have tumor-derived factors, pro-inflammatory cytokines, which play very important role. But I want to admit that the intracellular mechanism, let's say ubiquitin proteasome pathway, autophagy, and some of these pathways are common, and you know the trigger may be different for each condition, but underlying mechanisms, there may be some similarity in them.
Thanks.
And I will take another one question.
Thank you very much. It was a very interesting talk. Thank you very much. And we are working on the same pathway in neurodegeneration, but using Drosophila as a model organism. And what we find is when we downregulate the XBP1, we see its effect on the immune pathway, the TOL and the AMD pathway.
Yeah.
And insects, as you know, they only have innate immunity. First question. So what do you see here? Did you look into the immune response? Do they affect the immune pathway somehow?
So,
And second question is, XBP1, there it has a spliced and unspliced form.
Yes, yes.
So if you're knocking out XBP1, you're removing both the forms.
Yes, yes.
So which one do you think is more effective in muscle wasting?
Yeah, so both the questions are great. So first thing is that its role in the immune component, so that, you know, maybe that's the next experiment we want to do, is that delete the XBP1 in the immune component and see if it, the immune cells are very important for muscle wasting, you know, especially in cancer cachexia, they are there. And definitely the IRE XBP is involved in the regulation of immune system. In fact, the XBP we knocked down mice, the flox mice we got from Laurie Glimcher from Harvard, you know, she's a big immunologist. So I don't know right now that what is the XBP1 doing in the immune component, but definitely this is worth you know, trying to find out. We will knock out in the some of the immune cells and see if we can have some effect. Other thing is that unspliced XBP1. So unspliced XBP1 is not doing anything. It's just the spliced XBP1 which is important. So it deletes both of them, I know, but all our experiments suggest that IRE1 mediated increase in the alternative splicing of XBP1, that is important, and it's the spliced XBP1 transcription factor, which basically directly binds to the promoter region of many of these catabolic molecules which is involved there. And I want to tell you that in the normal mice, we don't have any phenotype. When you just delete unsplice this one, they look as good as the control mice. It's only when the tumor is growing, then this is activated, and then the effect is happening.
This last question.
So I have a general question, like you see human body or animal body has tendency to survive irrespective of whatever it faces, maybe a cancer or anything. So what do you think is the normal function? I mean, is there any advantage that the animal body or human body induces this ataxia in response to cancer? I mean, does it have any advantage?
Of what?
For survival of cancer patients? I mean, losing muscle proteins, not the muscle cells. I mean, does it offer any advantage to the body? Is it some sort of, body trying to save itself?
Yes, absolutely. That maintaining the body mass is very, very important for, in the cancer patients. This was not recognized before because initially people thought it's just the cancer. You are not treating, if you treat the cancer, everything will be fine. But even for treatment of cancer, maintaining the body mass is very, very important. And, you said that we are not losing the muscle cells, but we are losing the muscle mass, the cell. So maintaining the protein content is important because all the activity, physical activity, otherwise you will have morbidity if you don't have it. And at the same time, muscle is a very important metabolic organ as well. A lot of glucose and all the metabolic activity happens there. So maintaining certain level of muscle mass is, I think, very important for, yes. Yeah, because the cancer is also a metabolically very active thing happening in the body. So cancer needs nutrient, and muscle is a very good source for the nutrient. It can degrade the proteins that become like amino acids, and all those are released, and they are taken by the cancer cells to grow themselves. Yes.
So one last question from Dr. Jyoti.
Yeah. So what I wanted to, I have two questions and one comment. The comment is I'm from ACTRAC, and there are two scientists working on similar pancreatic cancer and cachexia. If you are there in Mumbai, we can arrange your talk. Second thing, because immunometabolism is now, kind of taking leap. So have you looked at, because fatty acid metabolism is connected to, say, M1, M2 regulation, involvement of immune checkpoints also. So have you looked at that? And second question, madam also mentioned innate immunity, and you also mentioned TLR. So have you looked at associated innate immune inflammasomes pathway? So these are the two questions.
See that we did not study innate immune response pathways, but it has a role in cancer cachexia. And my colleague Dennis Guttridge at University of South Carolina, he studies and he looks at macrophages and other cell types, the signaling within them and how they are impacting the muscle wasting during this thing. But you guys are working on cancer cachexia, so definitely my next visit, I'll come to Mumbai and speak there.
Yeah, great. Okay, thank you.
Okay. Thanks to everyone, and we'll have to.
There's one more question, Dr. Taneja. Oh, no, okay, that's it. Okay.
Thank you, Professor Kumar, for that illuminating lecture. May I now request Professor Nibriti Das to kindly felicitate our esteemed speaker, Professor Ashok Kumar. May I request Dr. Pradeep Sen to kindly felicitate Professor Nibriti Das. May I now request Professor Nibriti Das to felicitate Dr. Pradeep Sen. Professor Nibriti and Dr. Pradeep Sen. We now move on to a keynote from an exceptional scientist whose work at the intersection of epigenetics, muscle biology, and cancer has earned her recognition across the globe. To preside over this session, may I respectfully invite Professor Shripad Patil, National Institute of Mental Health and Neurosciences, Bangalore, and Professor Abid Hamid from Central University of Kashmir. Please kindly take your seats on the dais. Professor Reshma Taneja is a professor and head of the Department of Physiology at the National University of Singapore and a leading researcher in epigenetics, skeletal muscle biology, and cancer. Her work has significantly advanced our understanding of chromatin regulation, muscle regeneration, and rhabdomyosarcoma. Professor Taneja is a recipient of several prestigious honors, including the Venus International Women's Award, Faculty Research Excellence Award, Graduate Mentor of the Year Award, Leukemia and Lymphoma Society Scholar Award from the United States, and the Basil O'Connor Research Award. She will speak on interrogating drug-tolerant states in embryonal rhabdomyosarcoma. Professor Taneja, we are delighted to welcome you to Immunocon 2026.
Good morning, everyone. Can you hear me? Okay. Okay. So I just wanted to start by thanking Dr. Dil Afroz and the organizing committee for the invitation to be here. It's really a privilege and honor to come to this conference and meet new friends, as well as meet and connect with old friends that I'd lost touch with for a long time. So what I'm going to do today is actually talk to you about something that is somewhat aligned with what Ashok started off, and in fact, thanks to Ashok for setting the stage on muscle biology. I'm going to deviate a little bit, still talking about muscle cancer, but slightly different pathways. And again, it's not necessarily an immunology talk, but you'll see connections to the immune system and to molecules that you may be familiar with. So my lab has been interested in understanding this childhood cancer, a pediatric cancer known as rhabdomyosarcoma. This cancer occurs in very, very young children. It could occur from anywhere from 1 year after birth to about 10 years or so. And it's a highly aggressive cancer. So rhabdomyosarcoma, this pointer doesn't seem to work. Maybe I can use this, okay. Right, so what you see here are images of children with this particular cancer. And it's a relatively rare cancer, although it is the most prevalent soft tissue sarcoma that's known in children. One of the bigger problems of this cancer has been that, as I mentioned, it's highly aggressive. But more often than not, children are diagnosed with this cancer when it's really spread. So it's when it's high risk, metastasized in different organs, and therefore becomes extremely difficult to treat. The children that have high risk cancer basically have less than 30% chance of surviving for 5 years. And if one looks at the incidence of this particular cancer type in different geographical regions, you can see that the incidence varies depending on the location. But on an average, let's say in Asia as well as in the West, it accounts about 4% of childhood cancers. So what we know now is that this disease seems to arise, or this particular cancer seems to arise because of a failure of making skeletal muscle. and Ashok talked about how skeletal muscle is made. What you see here is that typically we have muscle stem cells that divide, give rise to cells that are known as myoblasts that proliferate, and these then eventually differentiate to become myofibers. And this is what Ashok was talking about in terms of looking at cross-section areas of the fibers. So there's a differentiation program going from muscle stem cells all the way to differentiated fibers. And in rhabdomyosarcoma, this ability of cells to transition from proliferating cells to making muscle fibers is blocked. So we know that RMS or rhabdomyosarcoma is a pathology of blocked myogenic differentiation. And in the field, a lot of people have been trying to ask the question, how do we actually get these cells to differentiate again? Because that's the primary problem. These proliferating cells that are blocked in their ability to differentiate continue to proliferate and become neoplastic. So one of the ideas in the field is to somehow find mediators that arrange or organize this differentiation and see whether those can be used as potential targets to induce differentiation of rhabdomyosarcoma cells. Now, rhabdomyosarcoma, there are many subtypes, but the two main subtypes are the embryonal subtype that account for about 70% of cases, and the alveolar subtype that accounts for about 20% of the cases. Embryonal rhabdomyosarcoma has a slightly better prognosis, whereas alveolar tends to have worse prognosis because it's highly aggressive. So given this backdrop that it's a childhood cancer and we don't fully understand how it arises, several groups have been trying to understand the genomic landscape. So what are the mutations that occur in these cancers? What is it that drives these cancers? And in a very nice study that was published about a decade back, a very comprehensive analysis was done. And what you see here on top is the alveolar subtype in red, and in blue are the embryonal subtypes. And as you would see, majority of the known tumor suppressors or oncogenes are actually not disrupted in these cancers. So classical mutations that you would normally have are not seen in these cancers. And in the embryonal subtype, there are some mutations in RAS signaling, but again, it's not very prevalent, it's not very high. There are some loss and gains of chromosomes, but again, nothing that is really striking. So given that there's a dearth of somatic mutations, the question really in the field has been, so how does these cancers arise? What is it that's driving them? And that's where a lot of focus in recent years has been in terms of trying to understand the epigenetic changes and also understanding cancer stem cells. So I'm sure all of you are aware that cancer stem cells play a prominent role in cancers in oncogenesis. They're important for initiation, for resistance to chemotherapy, as well as for tumor relapse. And remember, these cancers I'm talking about are very aggressive in nature. What is quite intriguing is that despite the rich knowledge of cancer stem cells in many other adult cancers, these stem cells are very, very poorly understood and characterized. We don't understand their molecular signature, we don't understand the metabolic properties, and so this is another area that is actually very important to look into. So with this backdrop of trying to figure what is it that drives rhabdomyosarcoma oncogenesis, my lab has focused on three primary areas. The first is trying to understand the epigenetic landscape, because we know that there are not too many mutations in tumor suppressors and oncogenes. We don't understand fully the tumor initiating or cancer stem cells, so what are the properties of these cells? And what are the mechanisms that really drive aggressiveness and relapse that actually causes the fatality in the children? So what I'm going to do today for the most part is share with you completely unpublished data relating to the third question. But I'll just quickly summarize what we've done over the past decade or so in terms of the first two questions that have been the focus in my lab. So we started many years back looking at epigenetic changes, trying to understand what is it that's altered in the landscape. And we had done this epigenetic screen looking at alveolar as well as the embryonal subtypes. And what we did was simply throw in drugs that target different chromatin modifiers that are listed here. And what we found was that some drugs such as UNC 642, 38, etc., that target these particular chromatin modifiers, G9A and EHMT1, were highly effective in reducing viability. And we've done a number of studies to understand how these factors function. And by far, one of the most prominent hit was BRD4, which again we've looked at recently and tried to understand its functions. And I'm going to summarize almost 6 or 8 years of work with this one slide to tell you that what we found is that G9A actually is important for this differentiation block that I was talking about. And it seems to function in the two subtypes, alveolar and embryonal, through distinct pathways. We think that in the alveolar subtype, it represses P10, which affects the PI3-AKT pathway. And in the embryonal subtype, it goes through Wnt signaling. So this particular axis is important for the block of myogenic differentiation. Whereas GLP and EHMT1, which has a similar function in terms of their enzymatic activity, has a distinct function, and it's important for maintenance of stemness properties in this cancer. And BRD4 is highly oncogenic. There are two isoforms, I won't go into it, but the long form is oncogenic, whereas the short form is actually a gatekeeper and prevents metastasis. So a slightly complex biology in BRD4, whereas these are fairly straightforward in differentiation and stem cell maintenance. Now, one of the clinical problems that I think, so we work with a pediatric oncologist, and he always tells me that one of the challenges he faces when he treats these kids is essentially the chemotherapy regimen. Now, the children with rhabdomyosarcoma are treated with a standard of care regimen, which is vincristine, actinomycin D, and cyclophosphamide. So this is the standard chemotherapy that's given. And you can imagine that if you were to give the same chemotherapy to a child that is given to an adult, it can obviously have very, very strong devastating effects. It affects the development of the children, but more importantly, chemotherapy itself, that damage actually induces, with a 16-fold higher risk in these children, the possibility of having a second primary tumor. So these children become predisposed to having more cancers. And so a clinical trial was done many years back where they tried to reduce the dose of cyclophosphamide, which is one of the more toxic reagents in this regimen. And surprisingly, when you reduce the level of cyclophosphamide and just look at local failure recurrence, you can see that if we reduce the dose, it actually becomes worse. It's not getting better. And this is a problem, this is a clinical problem in the field that a lot of oncologists want to try and solve. So in this particular clinical trial, basically the dose was reduced down. You see the dose that was initially given and then the reduced dose, and this resulted in more local failure compared to without the reduction. So the question is, why does that happen? What is happening with reduction and why is recurrence happening? And so we tried to model this clinical problem in mice by using PDX models, so patient-derived xenografts. And I'm going to share with you one example of what we've been trying to do with this particular approach. So what we did was to take rhabdomyosarcoma cells in a PDX model. We inject the mice, let the mice be till the primary tumors form. We then give them the same dosing that was used in the clinical trial, which is the reduced cyclophosphamide dose. And you can see how it was extrapolated in terms of the allometric targets for mouse compared to what was used in humans. So this is the dosing that we had used. And with treatment, of course, the tumors go away. It's quite effective. But then once you remove the chemotherapy, within a month, the mice develop tumors again. So there's clear recurrence happening here. And so what we did was to take the primary diagnostic tumor that was injected, the one that was treated with chemotherapy, so the regressing tumor, and the one that came back, the relapsed tumor. And we did single cell analysis to try and find out what is exactly going on, what's causing the recurrence to happen. And I'm going to share with you data of the single cell analysis as an example of what we think is going on with possibilities of which cells are important for recurrence. So after normalization for batch as well as for samples, we did Leiden clustering that you see here, and we annotated the cell types based on expression of different marker genes. And we found that the rhabdomyosarcoma cells actually are a heterogeneous population along the mesodermal myogenic axis. So what we find is about 9 clusters that we can identify based on marker genes. Cells which are in the G2M phase, the ground phase, which is more like the satellite cells or the muscle stem cells, cells that have hypoxic signatures, mesenchymal cells, myogenic cells, as well as proliferating cells. And this is an overlay of the diagnostic tumor, the regressing tumor, so RG refers to regressing, which is treated, and the relapsed tumors. And the dot plot here shows markers that are expressed in these different cell states. So you can see that the myogenic cells are marked by expression of myogenic markers such as MyoD, troponin, myosin heavy chain. The ground state is marked by PAC7 as well as MIF5. And the mesenchymal cells are marked by CD44 and MEOX2. And I'll just sort of summarize. I don't want to go into details of all the findings, but I just want to summarize key points that we took away from this study. So one of the main things that stood out was that in response to treatment, so here's the diagnostic, the initial tumor, the regressing tumor, and the relapsed tumor. And you can see that these cells in orange, which are the mesenchymal cells, increase in response to chemotherapy and then start going down again once the tumor relapses. So what this started telling us that in response to chemotherapy, there's an emergence of this type or increase in the mesenchymal population that seems to go back. And of course, when cells are treated with chemotherapy, you have increased a decrease in the number of S phase cells because the cells start to die out, so that's expected. And then they come back again. So I just want you to keep that in mind that the mesenchymal cell population is increasing. So we started looking, we took subtypes of each cell and started looking at the different populations. And what you see here is that if we start looking at the proliferating cells, so in brown is the cells in G2M, in S is the S phase cells. We start looking at these cells, you can see that there are hallmarks of apoptosis that are enriched in these cells. And you can see the apoptotic scores being enriched in response to chemotherapy. So this is the diagnostic tumor. The increased signal shows there's increased apoptosis, and then it goes back again. And if you look at standard markers of apoptosis, such as BAX, GAD45, FAS, these all increase in the chemotherapeutic treated tumors compared to the regressed or the initial tumors. And that's not surprising. If we now look at differentiation, so another phenomenon that's known to happen in response to chemotherapy is cytodifferentiation. So cells start to differentiate. And here, again, if we take the subset of myogenic cells, you can see that this is the myogenic population. The hallmarks of myogenesis are induced. And again, here's the diagnostic, the initial tumor, the tumor treated with chemotherapy, and this is the one that is regressed. And again, you can start seeing increase in markers such as desmin, myosin heavy chain, troponin T, and these stay on until a little bit later. What was also important to note is that while we see increase in markers of myogenesis, we do not see markers of any other cell type. So it's not that the cells are diverting and becoming something else. So they stay within the myogenic lineage. But what was actually most exciting and interesting to us was the fact that in response to chemotherapy, we saw this increased expression of mesenchymal-like cells. So this was very prominent, and you can see that this population of cells shows increased EMT markers. And again, you can see mesenchymal scores or EMT scores in the diagnostic, it's quite low, goes up quite prominently in the treated tumors, and goes away upon recurrence. And again, I mentioned CD44. You can see that it's highly increased. Collagens is increased, MGP is increased. So EMT-like markers go up in response to chemotherapy. And there's this emergence of this cluster. So from this, our takeaway was that when the tumors are treated with chemotherapy, some cells die, the proliferating cells die. Some cells start differentiating, the myogenic cells. And what is interesting is that there's an increase in the population of mesenchymal cells. So we thought that given that chemotherapy is sort of perhaps allowing for emergence of this population, this may be important for the recurrence that happens later. So that was the model we had in our mind. What was also very important is that in addition to these EMT markers, what was very apparent is in terms of the GSEA analysis, hallmarks of TNF signaling, NF-kappa B signaling, EMT that I mentioned. So clearly, hallmarks of immune signaling, NF-kappa B signaling that seem to be important for the mesenchymal cluster. We also started looking at the regulons, the transcription factors that are associated with each of these activities. And what you see here is the myogenic cells have MyoD that is expected. The S phase cells are characterized by E2F1, which again is expected. And you can again see in mesenchymal cells the expression of factors such as JUN-B, JUN-D, JUN-C that are increased in this mesenchymal population. And you can see that the mesenchymal population has increased in JUN activity. You can see that here in the plots, as well as JUN expression. So JUN expression also goes up in the mesenchymal cluster. So among all of these changes, CD44 was very interesting to us because CD44 is known to be involved in many different cancers. It's associated with chemoresistance, with relapse, with stemness. So we thought it's worth pursuing CD44 analysis. And so the first thing we did was to take tumor sections and look at CD44. And again, what you see here is the diagnostic tumors. This is the regressing tumors which were treated with chemotherapy, and you can see CD44 expression, the protein expression go up strongly at this stage, and then it goes down again in the relapsed tumors. We also took advantage of available data sets, publicly available data sets of single cell analysis. And again, if you look at CD44 expression in single nuclear RNA-seq analysis, you can see that the mesenchymal cells express high levels of CD44. So again, everything was pointing towards the fact that in response to chemotherapy, mesenchymal cells are increasing, seemingly marked by CD44, which may be important for the relapse that occurs later. So just for those who might not have thought much about CD44, it's a transmembrane protein. It interacts with its ligand, hyaluronan, and there are many other ligands, osteopontin, etc., that interact with CD44. But HA is one of the classic ligands of CD44. And CD44 has been known to be altered and upregulated in many different cancers. This is a slide taken from hepatocellular carcinoma. And through different pathways, CD44 is important for drug resistance as well as for HCC progression. So it's important for proliferation, survival, metabolism, etc. And because it's actually increased or elevated in expression in many different cancers, there's been a lot of attempt to address how we could target CD44. So there are many different approaches that are being considered using blocking CD44, targeting HA synthesis itself. And many of these are currently being tested in different cancers. However, currently there's no FDA-approved drug to target CD44. And of course, targeting CD44 could also lead to complications because CD44 is expressed in many different cell types. So it may not necessarily be the best drug target directly. Given that there are no FDA-approved compounds, we thought we should start looking at natural compounds that affect CD44 signaling. And we came across this compound known as verbascoside, which is a plant derivative that is synthesized from different amino acids. You see tyrosine and phenylalanine here. And this results through different pathways in the synthesis of verbascoside. And verbascoside has been tested in many different settings in mouse models as well as in cell culture. And it seems to impact NF-kappa B signaling, cytokine production, and reduces inflammation. So this seemed like a good possibility to try and target CD44 in our setting. So what we did then was to take verbascoside and do colony formation assays with patient-derived cell lines, RD18 and JR1. And you can see the number of colonies with DMSO. And you can see when we treat cells with verbascoside, it goes down dramatically. This is the chemotherapy that is normally used, and you can see that it's fairly effective. But if we use both verbascoside and chemotherapy, it almost completely wipes out all the colonies. So very strong synergism between verbascoside and the drug. This is in a second cell line. Again, verbascoside decreases, chemotherapy reduces, but together they almost completely wipe out colony formation. We also did some MTT assays where we looked at synergy between verbascoside and the chemotherapeutic agents. So here's the chemotherapy alone, and you can see that this results in a drop in viability, which is expected. But together with verbascoside, it's almost completely ablated. And this is with 3 different patient-derived cell lines. We see pretty much the same effect in all cell lines. So this now seemed to be a very promising possibility to try and target CD44 through a plant derivative. The question then is, how is verbascoside functioning? And what we did was to look at CD44 expression. You can see that with verbascoside alone, nothing happens. This is with chemotherapy, CD44 goes up, which is what I showed you earlier. And the combined treatment does not affect CD44 expression. So remember, verbascoside is thought to block CD44 signaling, not expression. And we don't see any change in expression here either. So that was reassuring. But at the same time, it affects signaling because we can see genes that are important for EMT, such as collagen A1, serpin, start to drop in response to the combined treatment. So it starts having an effect, the combined treatment has an effect on EMT and genes that are important for it, but not necessarily CD44 expression. So now the bigger question is, how is verbascoside actually even functioning? What do we understand in terms of mechanism? And so this is the work of a very talented student. So she actually did a competition experiment where she was looking at the molecular dynamics between CD44 HA interaction with and without verbascoside. And so what we're looking here as is the stability of hyaluronan bound to CD44 in orange, in the absence of verbascoside and in the presence of verbascoside. And this shows that in the presence of verbascoside, this complex becomes unstable. So what verbascoside is doing is destabilizing the CD44 HA complex. She then actually looked at the interaction energy between these complexes. And so what you're doing is simply looking at the interaction energy over a period of time in this molecular dynamics simulation study. And what you see here again is that verbascoside significantly alters this interaction energy. And then what we did was simply to take a snapshot of the molecular dynamics simulation trajectory. And what you're looking at here is the CD44 HA complex with verbascoside at the beginning of the experiment, so where verbascoside has not yet functioned. Verbascoside is in green, so over here, and HA is in orange, so the natural ligand is in orange. And so this is in the beginning, at the start of the experiment, and at the end of the simulation, you can see that HA is completely displaced, and verbascoside has taken the pocket of the ligand binding. So essentially what verbascoside is doing is displacing HA as a ligand from binding to CD44 and therefore affecting CD44 signaling. So we think that this is actually a very promising approach where we can actually displace through interaction energy changes the ability of CD44 signaling to occur. So I want to summarize what I've shown you today. What we've looked at is high-risk rhabdomyosarcoma samples which are treated with the standard chemotherapy, vincristine, actomycin, and cyclophosphamide. And we think that chemotherapy actually induces apoptosis as well as differentiation of these cells. However, when the drug is removed, you actually have the emergence of this population, a mesenchymal population, that then allows relapse to occur. And this particular population is marked by CD44. It's marked by EMT signatures, NF-kappa B, junk signaling, as well as inflammation. And we think that by blocking this particular axis by verbascoside, CD44 signaling, we can actually bring down the mesenchymal gene expression and remove this population. So conclusions are that chemotherapy selectively enriches for mesenchymal-like cells. It induces apoptosis in differentiating and proliferating myogenic cells, as well as upregulates junk and inflammation markers in the mesenchymal cells of the regressing tumor. Through molecular dynamic simulation, we find that verbascoside is a competitive inhibitor of CD44 transmembrane protein, and treatment of RMS cells with chemotherapy as well as verbascoside. So remember, chemotherapies at lower doses results in increased cell death as well as clonogenic ability. So we think that this could be a potentially good target to look at relapse mechanisms. And with that, I'll stop here. I'll just conclude by thanking people in my lab. The project I talked about today was mostly the work of a very talented graduate student, Jay. And some of the work was done by Amok, another PhD student. They've both graduated since. A big thank you to our clinical collaborator, a pediatric oncologist who's provided us with all the PDX samples, and our funding agencies that have supported my research all along. Thank you very much for your attention.
Thank you, Dr. Reshma Taneja, for a very detailed talk on the during treatment and post-treatment changes in the rhabdomyosarcoma in the pediatric population with a nice animal model. The paper is open for discussion.
Yeah.
2 questions.
Thank you very much. It's very nice study. I have quick 2 questions. In your WAC treatment, the procedure where you have the tumor transfer experiment, do you see change in the immune profile in those tumor where recurrence is happening?
Of course, yes.
So what kind of changes you observed?
So we haven't fully characterized. In fact, this entire project is still a works in progress. We haven't completed it. We haven't even finished the animal experiments with the combination therapy. We do see changes. It's a bit too early to fully tell you about it, but hopefully I'll be able to tell you in another 6 months' time.
Second question with your CD44 signaling molecules. Do you see again, because CD44 is there on a lot of cells and including immune cells,
yeah.
When you alter, do you see the immune phenotype alteration by altering the CD44 signaling other than what you see in the tumor cell?
So absolutely, you're spot on. I think those experiments that we still need to complete in terms of profiling all the immune cells. So that's something that we're still doing at this point. But
Yes,
good
Dr.
question.
Ashok.
Yeah.
Hello. Here.
I have a question regarding this in the.
Okay. So what I wanted to talk about that, this Verbaxco site, it also known to be a NLRP3 inflammasome inhibitor.
Yes.
So have you looked at it, because it will really control the inflammation maybe in a better format?
Absolutely. So there's a lot that we still need to understand in terms of mechanism and the cell types that are critical for this phenotype. Because when we do the experiments, colony assays or even MTT assays, we're doing it in a 2D culture, right? So basically you're not complementing it with immune cells. But the fact that the tumor cells themselves are responding suggests that immune cells, of course, would be important, but the tumor cells themselves are actually responding to CD44 inhibition too. But definitely a good question and something we need to look at.
Yes, Dr. Ashok.
My question is
Yes.
regarding this. In the expression profiling, you have shown the genes which are overexpressed in such type of pancreatic cancer, and I didn't see overexpression of GLP or G9A in that slide. But in the subsequent slide, you said that these methyl transferases are upregulated. So I couldn't get the connection there.
Yeah, so I didn't show all the data. I didn't show the epigenetic markers that are altered with the profiling that we've done here. What I mentioned is studies that we've done in the past where we found that GNIA and GLP are actually inhibitors of myogenic differentiation. In this analysis, we do see a lot of chromatin modifiers. We haven't fully characterized them because our focus was actually not so much on differentiation per se. We're more interested in looking at what is it that's causing relapse. And differentiation is not the problem there. The problem is that there's this emergence or survival of these mesenchymal-like cells that are causing the tumor to relapse. So these are separate problems in some way, controlling cell death in response to chemotherapy, controlling relapse mechanisms, controlling differentiation. So I didn't go into the epigenetic changes. We did see G9A GLP come up, but we didn't look into it because that was not the focus of this particular study.
Yeah.
Deepankar.
Yeah, thank you. Following the same line,
Yeah.
I mean, you have been studying the epigenetics, because epigenetics in this context, the unpublished study, do you think that the epigenetics plays role here in kind of increasing these mesenchymal cells? Have you studied that?
Absolutely, absolutely. So there has to be a response that is regulating the expression of all the mesenchymal genes. And we think that some of the epigenetic changes could be upstream of the changes that then lead to their increased expression.
Sorry, one more question. You showed that there was a change in the transcriptional program, but there was not a specific mechanism. Do you think how does this chemotherapy induces the transcriptional regulatory networks that induces these increase in these mesenchymal cells?
So the question is, how does mesenchymal gene expression go up?
Yeah, I mean, is there any particular pathway or particular transcription network programs that are that leads to the induction of these mesenchymal cells upon this chemotherapy?
So as I mentioned, what we did see coming up a lot and very prominently is junk signaling, NF-kappa B signaling. And we haven't explored whether NF-kappa B is actually important for the emergence of these, so in terms of transcription factor activity, whether that's actually critical. But Jun activity seemed to correspond very strongly with the mesenchymal population. So we think that somehow junk signaling is important, although whether it's for expression of the genes, for survival, we haven't looked at that yet.
Dr. Deepan.
Yeah.
Yes.
Good talk, Reshma. I was wondering, can you shed some light on your use or approach of using a plant compound against CD44 as opposed to using an antibody against CD44?
Yeah, so actually, in the field, there's been a lot of effort to use CD44 antibodies, target the ligand. At this point, nothing was commercially available, so we were actually a bit handicapped in terms of resource as well. None of the antibodies are commercially available. We know that work is going on, but there's nothing that's currently available that could be used in mice. So that was one limitation. One of the problems with CD44 is, of course, that like all other antibodies or drugs, resistance develops, and or it could be toxic because CD44 is not exclusive to these cells. It could be expressed in many other cell types. And so in general, using a blunt approach that targets a molecule that's expressed in many different cell types is generally not the best. So we were trying to see whether we can still tamper with signaling without actually having the toxicity effects. So the cells are vulnerable, the drug is tolerable, it's a plant derivative, so in toxicity issues, there's no problem. So that's the line of thinking, but in reality, many of the reagents are not commercially available too.
Last question from a co-chair.
Thanks for the wonderful talk, Professor Reshma. I was wondering regarding, we started with the mutations have less role in your case, in your model system, and epigenetic basis is getting more attention. I was wondering vis-a-vis, we started with the N is equal 10 and recurrence was in N4. It is 40% of your sample size once they were treated with chemotherapy.
Sorry, say that again.
So we had N10
Yeah.
and recurrence was in 4. So it's a 40%. Can you specifically highlight what could be the attributing factors? Because we largely attributed the epigenetic basis over here in your case in this rhabdomyosarcoma. How to highlight this high level of this recurrence rate in your case?
So as I mentioned, in terms of chemotherapeutic standard regimen, the tumors relapse. That's actually the main problem with the children because they tend to develop another cancer, and the same tumor relapses as well. So this recurrence is one of the bigger issues, and that's essentially what we were trying to model in this particular. So we use the lower doses of cyclophosphamide with the idea that we want to see whether in mice we can see or replicate the same phenomenon that was seen in the clinical trial. The proportion of relapse is not surprising. It's very similar to the human data.
Using antibiotic alongside it in the chemotherapy cocktail.
Using
Have
what?
you have tried to, some antibiotic in the chemotherapy cocktail?
Yes, yes.
Any specific reason for that?
No, so these are the standard of care drugs that are used for patients. So vincristine, actinomycin D, and cyclophosphamide is basically what is given to patients.
Thank you, Dr. Reshma, for the talk. In this National University of Singapore, let me add some point. This was in the bracket of top 10 universities globally. So Reshma has given a good talk from that. So thank you.
Thank you so much, Professor Taneja. I now request Professor Shripad Patil to felicitate our worthy speaker, Professor Reshma Taneja. I now request Dr. Abid Hamid to felicitate Professor Shripad Patil. I request Professor Shripad Patil to felicitate Dr. Abid Hamid. Thank you, sir. We now have a technical presentation by Agilent Technologies. May I request Mr. Ajay Modi to make the presentation. The time allotted is 10 minutes.
Good morning, everyone. Hello. Yeah. Good morning, everyone. So I'm going to give you a brief glimpse of our cell analysis portfolio, which is from Agilent. So we have got a wide range of complementary products which cater to the cell analysis. We have got the imaging and microscopy system. So we have got automatic cell imager come multimode readers. We have got the confocal imaging readers. Then we have got the real-time cell analyzer, which is based on cellular impedance. So this can be used for potency assays, cell killing assays, apoptosis assays. Then we have got the seahorse analyzer, which actually measures the cellular metabolism in real time, and it's a label-free. Then we have the flow cytometer, the Novocyte, where we got a wide range of flow cytometers. Then we have the detection systems, especially the microplate readers, multimode readers. In liquid handling system, we have got the microplate washers, dispensers, and of course the automation, where we have the stacker, automated CO2 incubators, and the analysis software. So we have got the wide range of Novocyte flow cytometers, which are used for various applications like activation, proliferation, immunophenotyping, cell counting. Then we have got the Agilent Biotech imaging and microplate readers, again used for various applications like cytotoxicity, cytokine detections. The Excelligence real-time cell analyzer, as I mentioned, you can be used for proliferation or potency assays, especially the immune cell killing assays. And then the Seahorse analyzer for metabolic profiling, fitness of the cells, exhaustion, and various other applications. So I would like to introduce the Citation 9, which is recently launched. It's basically a multimodal system which has got the multi-mode plate reading capabilities, where one can perform the UV visible absorbance. UV visible absorbance, fluorescence intensity, luminescence. One can also perform flash luminescence, flash fluorescence, filter-based luminescence, especially the bright assays, nano-bright assays, time-resolved fluorescence like HTRF or TF-red assays, fluorescence polarization, and alpha screen. So it covers a wide range of multimode reader capabilities. In addition to the multimode reading, it also has got the imaging and microscopy feature, where you can do fluorescence imaging, you can do up to 4-color imaging. It has got a bright field, high contrast bright field, especially very useful for your label-free cell counting or scratch assays. Then we have the phase contrast feature, the color bright field, and upright. Upright is really a very, very useful feature, especially if you're doing ELISpot imaging. So it covers a wide range of capabilities, everything packed into one system. Yeah. Sorry. Oops. Sorry about that. Okay. So we do have the optics. So this is the optics. What you see, it's for the multimode. So it's got a quadruple grating monochromator, which allows you to perform UV visible absorbance, fluorescence, and luminescence. We have the filter-based optics, which allows you to perform sensitive fluorescence assays like the TF-red or fluorescence polarization. And the bottom one is the optics for the imaging. So if you see, all the optics are independent, so no compromise. And this system provides you the quantitative univariate data and multivariate data, especially if you are doing some phenotyping assays. So this is the arrangement, what is being provided. It's got a quadruple grating monochromator, so which allows you to select any excitation, any emission wavelengths for the fluorescence. You can use any wavelength from 200 to 999 nanometers for absorbance as well as luminescence. For imaging, we have got the LED, so you can do up to 4-color imaging, and we have the camera, 16-bit camera. The system can hold up to 6 objectives, right from 1.25x up to 60x. So you can do multicolor imaging and overlay the imaging. So we have got objectives right from 1.25x up to 60x. So we use Olympus objectives over here. So you get a wide range of magnification with the system. So this system provides you the feature for your bright field imaging, standard bright field. You can do high contrast bright field, especially very useful for your label free. Then we have the phase contrast. We also have the color bright field, especially very useful for your histology samples like H&E stains. Then we have the upright microscope, which can be used for your transmitted light imaging or reflected light. The impacted applications are, again, you can use for ELISpot, colony counting, and various other applications. So you can use it for ELISpot imaging as well. Oops. This system is also modular and upgradable. It also offers you the feature for your CO2 and O2 control, especially very useful for your live cell imaging. For your flash luminescence or flash fluorescence, we have the injector. So this system can be also used for your calcium imaging. And for automation, we have got various other products like the stacker, where you can stack up to 50 microplates. So you can have a walk-away system. We got an automated CO2 O2 incubator, where you can have up to 8 labwares. It can be a microplates or slides, petri dish, and the joystick controller. So these are the various options available for upgradation of the system. Then I would like to change the gear now. We'll move on to the real-time cell analyzer, which is Xcelligence. So we have got a wide range of systems available. So these are real-time cell analyzer, which measures impedance. And we had a recent one launched in 2019, which measures impedance and imaging both simultaneously. So basically, this includes a 96-well plate, which consists of a gold biosensor, so which enables you to measure impedance. So this can be used for your imaging as well as measuring impedance. So basically, it measures the cell index. So the cells are seated onto the plates, and as the cell proliferates, you will see an increase in impedance. So this enables you to exactly pinpoint when the killing takes place. So once you add the apoptosis inducer, you will exactly see when the cell killing takes place. So it's a very good platform for your cell killing assays or cytotoxicity proliferation. Then I would like to give you a brief about the CHOS technology. So we have got a wide range of instruments, right from the 8-well format to 96-well format. So this is a very good platform for measuring the cellular metabolism. So what it measures is the glycolysis, that is the extracellular acidification rate, or ECAR, and the OCAR, which is the oxygen consumption rate, which is actually the mitochondrial respiration. So we have got different products available. We have got the system for the 96-well format, 24-well format, and 8-well format. And I would like to highlight the recent model which is launched, which is called the Flex, XF Flex. So this is a new system which is compatible for your 3D models as well. So this can be used for the organoids, organ chip, or 3D printed tissue organoids, even tissue slices, 3D printed tissues, or zebrafish embryo, or islet cell. Up till now, all the systems were suitable only for the 2D, but now we got the 3D model as well. And I would like to just touch upon the flow cytometers. What we have got, we have a wide range of flow cytometers, right from standard 1 to 3 lasers, 4 lasers, and we have got the 5 laser system, Pantheon, where you can have up to 30 fluorescent channels. And the recent one, what we have launched is the Opteon, where you can have up to 5 lasers and 70 detectors. And these are the various applications which are impacted by this product, where it can be used for various applications like cancer biology, cardiovascular research, cell biology, immunology, immuno-oncology, and so on. And I just wanted to announce we are having a webinar, a cytometry webinar on 3rd and 31st of July. So where we will be talking more about the spectral flow cytometer. Please do take time to maybe attend this. Thank you.
Thank you. So we now have the privilege of an invited talk from within the Government of India's scientific establishment, a perspective on how India is building its genomics future from the ground up. To preside over the session, may I respectfully invite Professor Madhu Tapadia from Banaras Hindu University, Dr. Sunil Raghav from Institute of Life Sciences, Bhubaneswar, and Dr. Ulfat Ara from Sher-e Kashmir Institute of Medical Sciences to kindly take their chairs. It's our distinct privilege to welcome Dr. Richi Mahajan, Scientist D at the Department of Biotechnology, Ministry of Science and Technology, Government of India. Dr. Mahajan is an expert at the forefront of high-throughput genomics, bioinformatics, and national health data infrastructure. His critical contributions include an impressive portfolio of approximately 16 high-impact research publications in leading genomic journals, alongside a stellar record of managing national databases and published intellectual property patents. Today, he brings us an invaluable, high-level perspective on transforming data into scalable healthcare solutions and sovereign scientific capability. Dr. Mahajan will speak on From Information to Innovation, Building India's Genomics Ecosystem. Dr. Mahajan, we warmly invite you to the podium.
presentation अभी पे चला है. ये स्लाइड चाहिए. अच्छा स्लाइड ये तो इसमें कर सकता हूँ ना.
Thank you, organizers, for having me here. Thank you for the kind introduction as well. Today I'll be taking you through the journey of what Department of Biotechnology has done, a moonshot project, which is actually revolutionizing the genomics ecosystem. Sorry? Okay. Do I have to wear something else or have to be closer to the mic? Okay. Thank you. I'll try to be as closer as much because I'm anyways loud. So, okay, so to start with, I will just like to mention what I mentioned yesterday, that we are 160 billion by economy and we are marching towards 300 billion. And there are several advantages why India is actually moving towards 300 billion by economy. We are early starters. We have ease of doing business, which has been recently launched, ease of doing science. And we have huge HR available for doing all this world-class research. And we're second highest, and Indian bioeconomy right now is the third largest destination in Asia Pacific regions. And these are the key growth drivers with the skilled biotech workforce, 75 incubation centers, what was being mentioned yesterday in Industry Academy talk, and government enabling government policies, as well as now we have focused on biomanufacturing. And that is what I was again mentioning yesterday, that right now we are currently based at 4.25% of the GDP, but we aim at around 10% of the GDP. Honorable Prime Ministers and the Union Cabinet has recently, or probably I must say in 2024, has launched Bio E3 policy, another step of a stronghold step towards 300 billion bioeconomy, and the commitment to advance future that is more sustainable, innovative, and responsive for global challenges. And under this ambit, there are 6 verticals which have been defined. This is readily available. But today I'm going to talk about the omics revolution in science and technology. And India is strategically leveraging the power of these omic technologies for actually comprehending the productivity to feed its 1.4 billion people, as well as the health, mitigating the health challenges as well. So talking about precision medicine, it is about one size doesn't fit all. Everybody responds differently to different medicines and different molecules. Change in one base pair can actually lead to change in the whole expression as is evident from this cartoon. So what happens today, I mean, is that we typically say are diagnosed by we go to a doctor, we are given diagnosis, we run through some tests, and based on that, we are given a symptomatic treatment. But this is only managing of the disease and not predicting of the disease progression. And we need more holistic view towards this. So what will be the future? Future will be actually shifting from symptoms to systems. We need to have AI twins or multi-omic twins, and we need to have a lot of data for that. And those data being generated, or I must say real-time data being generated, may be applied to the pathway simulations where a personalized health plans could be figured out for all the patients, and even that would be complementing the doctor's advice. Okay, this is from a movie actually, Apollo 13. Houston, we have a problem. Jacques Figert said this around 200 miles away from Earth, right? And there was a problem in oxygen tank, and it was looking like end of the road for them. And then actually Apollo, the team back in NASA, they actually made a first twin of the space shuttle, and they actually helped them out to figure the problem, and it was relayed to them real time, and the engineers in space actually could figure out the problem and save the spaceship, and they could come back. That was the first twin. That was the birth of first digital, the first model twin, I must say, or a digital twin. And from there, the technology has really revolutionized. The market has been really growing, and there have been a lot of global players. I'm just showing these slides just to show that there has been a boom in the biotech industry as well in the field of digital twin after 2015. It was the term was coined somewhere in 2002. Then it was mostly applicable in aeronautical sciences, and now this field is booming in biological sciences as well. So it has a lot of clinical applications in cardiovascular diseases, surgeries, pharmacy, orthopedics, and diabetic management. Even diabetic management is available in India as well. So this is some of the technologies which are available globally as well. They are in very nascent stages. They are in very predictive stages, but still available, where you can have the predictive treatment and clinical pathways to cater to the needs of the patients. And many companies are also venturing into it. This is a very a story of Liam Helliker and Thomas Helliker, where actually Thomas is a immunologist, and his son was diagnosed of SKID, and doctors had given up on him in 2021. So he started making a digital twin, digital twin for his son, and by the grace of God, the son is still healthy, and he's 13 now. But all this requires a lot of data, right? And as I mentioned in my first slide, data is key for carrying out all this kind of work. But if you see the Indian scenario, India is an ethical museum with more than 4,600 populations, 532 tribes, and we have a huge genetic diversity, as well as we have a unique genetic diversity due to congenital marriages, as well as high endogamy being which is being followed in many tribal groups all over the India. And the genomics data is also very, like the Indian representation in genomics data globally is very skewed. And if you see from this slide, South Asia comprises 25% of the world population, yet only 0.8% of the global genomics data. So that means the drugs or the research being carried out globally is mostly based on the global databases. In Indian population is very less represented. And following this league, we started a Genome India initiative, which was to establish India as the genomic data hub. Before I run into my rest of the slides, I request a video to be played with the permission of chairs. Can you play the video, please? So this will just give you a flavor of what Genome India is.
India's population of 1.43 million people comprises more than 4,600 ethnic groups that harbor a large number of genetic variations. Indian population originated from a few founding groups. These groups share some genetic similarities as well as exhibit distinct variations. The Indian genetic variations are not well represented in global databases. This important gap is being filled by the Genome India project, funded by the Department of Biotechnology. A team of scientists from 20 institutions have collected blood samples from 20,000 individuals representing 99 diverse populations and performed whole genome sequencing of 10,000 individuals to catalog the genetic variations.
By decoding over 10,000 genomes, the Genome India initiative has created a national wealth of biological data that will be useful for embarking on next level basic research as well as targeted clinical interventions specific to Indian population.
The Genome India project was initiated in January 2020 under the leadership of Professor Vijay Lakshmi Ravindranath. The digital data thus generated will be precious national resource for scientific and medical communities. DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine. Human genome comprises 3 billion nucleotide pairs. The whole genome sequencing is a technique by which we can know the order in which each nucleotide is arranged in the entire genome. The Genome India project will create a reference genome for Indian population.
The Genome India program of Department of Biotechnology is an exciting opportunity for us to decipher the genetic background of our country, the sources. This will provide in future an opportunity to develop specific drugs, targeted therapies, genome-based solutions, and the future gene therapy programs that will help a variety of complex diseases that are prevailing in our country.
4 institutes that have collected the samples and performed the whole genome sequencing are CBI, CCMB, IGIB, and NIBMG. 9 other institutions which collected the blood samples include SKIMS, AIMS Jodhpur, GBRC, ISER Pune, NIMHANS, RGCE, ILS, Mizoram University, and IBSD. 7 institutions which are developing new methods include IIT Delhi, IIIT Allahabad, IIT Jodhpur, CDMD, IISc, NCBS, and IIT Madras.
Genome India is a unique study of its own kind because it takes into consideration the diversity of Indian population and creates a compendium of genetic variations of the Indian population. The project lays the foundation of genomic hub and an opportunity for India to enter the emerging field of personalized medicine.
The rich genetic data generated by the Genome India project will soon be a digital public goods that is being archived at IBDC.
Genome India is a flagship initiative for cataloging the unique genetic variations of the Indian population. Visionary funding by the Department of Biotechnology, DBT, and the dedicated involvement of 20-odd institutions has resulted in this remarkable success of the Genome India project, and I'm proud of the dedicated team of scientists, the Genome India Consortium, and I also want to congratulate them for their efforts. This is indeed a watershed moment for India that will genome-based medicine for various diseases, various ailment conditions in the Indian population.
As the scientific and clinical communities eagerly await the insights, outcomes, and digital data, Genome India stands tall as a beacon of India's commitment to deep science by advancing genetic research for the benefit of every citizen of our nation.
मुझे विश्वास है बायोटेक्नोलॉजी रिसर्च के क्षेत्र में ये प्रोजेक्ट एक मील का पत्थर साबित होगा। मैं इस प्रोजेक्ट से जुड़े सभी साथियों को बहुत बहुत शुभकामनाएं देता हूं। साथियों, जीनोम इंडिया प्रोजेक्ट भारत की बायोटेक्नोलॉजी रिवॉल्यूशन का एक महत्वपूर्ण पड़ाव है।
Genome India, genomics at its best for a healthy India.
Thank you. So many of you actually sitting in the crowd were part of this project. So maybe may I have a round of applause for all of you. Actually, you were all involved in it, and coming from the highest leadership actually means a lot, including Sunil sitting over here. Yeah. So as it was evident from the video, like the plan was to establish India as a genomic global data hub. And the target was the health of the people, the prosperity, as well as the security of the data as well, because we don't want data to go out of the country. So the vision was to sequence 10,000 genomes and build a reference database, as well as enable the precision medicine regime in India. We collected around more than 20,000 samples, and approximately 36% of the samples were collected from the rural population, 32% from the urban, and 31% from the tribal population. Target was initially the 100 populations, actually trimmed down to 99, but then during the refining of data, we could actually only limit ourselves to 83 populations finally. And as mentioned in the video, these are the numbers. They were this was a Herculean task involving more than 150 scientists actually working around the clock for 5 years, even during the COVID times. And a huge biobank has been created at CBR, ISC Bangalore, regarding all these samples, all these 20,000 samples. And as mentioned in the video, this was a milestone. This is an important milestone, and the data, Genome India data, was dedicated to the nation by Honorable Prime Minister last year on 9th of January 2025. The data is now accessible on Indian Biological Data Center. Talking about some key finding or some research leads in the Genome India projects, there has been identification of more than 130 million genetic variants. Mostly these are indel insertion deletion variants and small nucleotide variants, and there have been identification of more than 7 million novel variants which have not been represented in the global populations. Just to give you an example, that at least 10 new missense variants have been identified in LDRL genes linked to familial hypocholesterolemia in India, which were not actually represented in any of the global databases. So these are some of the key findings that we are which is going to affect the research, or I must say, which is going to shape the research of precision medicine, as well as in general genomics research in India. Again, our sequence reveals that novel insight into the Indian population, the sample populations capture linguistic diversity. As you can see, there is a correlation between the coordinates of the 83 populations wherein the sampling was done and PCA shows the clustering according to the language with the distinct north-south cline. Sorry. Okay. So we've coded the population just to be very sure as per the highest orders. And the broad patterns reveal the tribal population, particularly the code DT, A and A were the oldest population, but apparently isolated and minimal population growth. And TBT, which was probably in the south, is relatively youngest and fast expanding population. There are a lot of clinical implications associated with it. There were around 73 actionable genes identified, out of which 131 were associated with the pathogenic or likely pathogenic variation. So that means, and these were mainly linked to familial hypoclastemia, BRCA mutations for breast cancer, ovarian cancer. So if, this to mention you again, this was the study of healthy individuals, and in these individuals also these kind of variants have been found. So that means our healthy individuals are also associated with the disease profiling. Again, in PKG, Pharma GKB database, around 38 variants out of 18 have been identified, and again from the healthy individuals. So that means even we are less susceptible to the disease, the drugs being manufactured in the global west based on the global databases. And as it was mentioned that, we are trying to build our own reference standard, which is actually having an improved imputation accuracy than the TopMed as well as haplotype reference standard. Just to sum up the Genome India findings in one slide, I would say 33% of DNA changes have been identified, and many of these are associated with the disease even in the healthy population, and 1/4 of these are potentially disease-causing DNA changes and are novel. And 57% of the Indian population contains the variations which can lead to some disease burden, being the metabolic diseases. And the impact has been huge. Disease risk profiling is being carried out. We are targeting towards development of affordable diagnostics as well as precision medicine. A genome GI database has been developed by IGIB precisely. Dr. Farooq has worked on it, and this is mainly consisting the SNP based search, and this is available for public use. And again, imputation panel have been prepared. So keeping in view the importance of the data, at the same time, I'll just sum up in 5 minutes. Keeping in view the importance of the data, the translational research call was issued at the same time during the release of data. We received more than 400 proposals all over India. Again, it was a long process of identification of what to screen, what not to screen. Mostly proposals were actually very good. I mean, if some of you sitting in the crowd think that I submitted a proposal and it was not selected, it was that, the proposal was good, but maybe it was lacking the inference of Genome India data. Mostly, we could find that most of those proposals could be carried out without even the without even appreciating the Genome India data or without even considering the Genome India data. Those could be isolated researchers. And this call was mainly focused on utilizing the Genome India data, which was not evident in many of the proposals. So finally, 75 were screened and in these various domains. Another initiative which was done by DBT was Human Microbiome Initiative. Again, a very a very ambitious project being carried out initially by NCCS Pune, and where more than 4,000 individuals were sampled all across India. And this figure shows the microbial distribution of across the country, and it was attributed that mostly the microbiome profiling of the country is attributed to the dietary habits, specifically the spices. There has been a lot of data being generated on the spice profiling actually, and many of the microbial profiling has been associated with the spices being used in that region. An Umeed initiative, I mentioned this slide just because KIMS is part of this initiative, wherein we are screening rare and genetic disorders in pregnant women as well as newborn babies. We have adopted we've actually supporting 30 aspirational districts, and there are 30 Nidan Kendras also being supported throughout the country. Nidan Kendras are basically the high-end testing centers, and aspirational districts is where we are actually screening newborn babies, as well as antenatal screening has been done for these hemoglobinopathies and common metabolic syndromes. In first phase, we could screen around 3 lakh cases, and in this we are already into the second phase, and we are expecting to have around 10 lakh cases screened by that time. I know you may say that this would be a very small figure considering the volume or the our population of the country, but then we are envisaging that this would actually pave a way for a national screening program which may be taken up by the Line Ministry. And we need to control all this data, right? This is the genomics data, and genomics data is something which is a high value target, and it has immutable identity and exploitation risks associated. So the risk of losing genomics data is always high. Keeping in view of that, and we're learning from the global standards as well. I'm just rushing through the slides in interest of time. So keeping in view, we actually launched Biotech Pride guidelines as well as feed protocols, which are basically the enabling mechanism for sharing of biological data in the country. I'll again stress, it is an enabling mechanism only for sharing of biological data. It does not mention anything about generation of biological data. And to implement that, we have actually established Indian Biological Data Center, which is hosted at Regional Center Biotechnology, Faridabad, the NCR cluster, and Brahm HPCs, which were talked about in the video as well. So just to give you a heads up, and these are some of the portals which are available in Indian Biological Data Centers. Control Access Indian Nucleotide Data Archive is the place where Genome India data and microbiome data have been stored. Again, to mention something that we are now going to be the part of INSDC, we've got all the approvals from government as well as, I'm wrapping up. We've got all the approvals from government as well as we are in talks. 9th of June was the meeting where the assessment one was done, and probably once we're there, we'll be the part of INSDC coalition as well. So road to the future or genomics is huge, and we need to have, if we want to make India really leader in in genomics, we actually need to, strengthen our genomics ethics as well as our data archiving ethics, as well as we have to share the data amongst our own scientists for, carrying out this research. Thank you. Thank you, thank you.
And how they are collating the data. Yeah? Okay, so thank you so much. So now the talk is open for discussion. If there are any suggestions or questions, it may be please directed to Dr. Ritchie.
Thank you.
Thank you for, am I audible?
Yeah, very audible.
Thank you, Dr. Richi Mahajan. This was nice presentation. I have one query regarding this collection of data from different sources, different data centers. Did Department of Biotechnology categorize the data based on gender or based on age or based on different ethnic groups? Because there could be a lot of variation in terms of ethnic groups, in terms of disease patients or normal patients or normal persons. So how do we categorize it?
I'll answer this. Sunil, do you want to answer this? Since he was involved in sampling, so probably he can answer this better.
Thanks, Richie. So it was nice presentation. So actually we had collected only the controls. The idea was to make a reference map, and here the proportion we wanted to keep like males, females, and we cannot really collect all the 5,000 population.
Yes.
So that's why we have selected 83, where we know that based on earlier studies from IGIB, we made the map of like India properly grid, and then we decided the populations. So these populations really like a comprise of oldest like populations, endogamous populations, which are very close populations. The idea was to cover as many genomic variants that can differentiate and make a reference of Indian population. Definitely we need more populations to cover, to make proper reference map. But this is the start.
Oh.
And now the plan is to go for
Yeah,
the
we are actually moving, I didn't mention in my slides, since the things are under approval, we're moving towards Genome India Phase 2, which would actually cover around 1 million populations. So that's it.
I mean, I just wonder, how can we use this reference for different ethnic populations across the country? Is it possible?
Yeah, there are populations collected from all over India. So definitely it can be used, but if you are working on very small population, endogamous population, for which the reference is not there, then it would be difficult.
Okay, thank you very much.
Since I'm an oncologist and precision medicine is the cornerstone of treatment for oncologists, where have we reached as far as cancer genomics is concerned? Because you have mentioned breast, pancreas, oral, head and neck.
So as I mentioned, this data was mainly from the healthy individuals. We haven't actually right now taken the cohort studies or any disease-specific studies. That would be a very ambitious one, a very riveting one, and that would be part of our next phase, which will be soon available. We are actually formulating those lines. We're working on those lines, and probably once we come out with that proposal, because there's a huge amount for 10,000 genomes. It took around 150 scientists, 20 institutes. So you can see we are multiplying it 100 times now. So it's going to take some efforts and some times. We are trying to learn from the phase 1 proposals, schema, and then probably and definitely the cohort studies are going to be part of it. The breast cancer, the rare genetic disorders, CVDs, neurological disorders, all this is going to be part of those.
Yes, because I think a basic cancer genomic platform should be established, because most of the times the clinicians are facing the problems. We get so many labs, 150 genes for this, 130 genes for this. And most of the mutations, they are actually the passenger mutations. It is a very few chunk which is driver mutations.
Yes.
So I think India should be able to build up its own, cancer genomic platform against which we can, establish our own targeted treatments.
Yeah.
So if
I can add just one point here. You rightly said this thing, but till now we didn't had any reference. Now we have some reference. We can see that what are the variants we have in high frequency. At least we can compare with the variants that we are getting in our genomic analysis and say that, okay, this is in our population already, so this is not causal. So that we can negate.
So, Dr. Raghav, just to add to what, you were saying, that we have some references. I mean, does some references actually help, so that's the point. Is it not more confusing than solving anything? And the other thing I would like to just ask one question and have a little, since I am a basic science person, but I am in a medical institution. So what I see is that there are a huge number of samples we have. We have a wealth of samples there. We could utilize these samples, because at the end of the day, we just throw them away. That's also another issue with us, because how did you discard these samples? So why not use it for, we can categorize it there, use it properly. We have daily we have at least more than 10,000 samples daily, on a daily basis. So that could solve, if we rope in the hospitals, I think that should be able to solve the problem of sampling. I think
that's a good idea, yes.
I'll answer this one. The thing is, ma'am, actually, if you see, these 83 populations are homogeneous populations. This is not a mixed population. We've tried here to identify the homogeneous populations, and the reference was being prepared using all these homogeneous populations. When we say that we have hospital samples, we don't have categorization to those populations. That would make it very difficult to put it into what group to, when the patient is coming in. I'm just completing my point. Yeah. So this is what makes this study unique. Even in whole of Southeast Asia, such the variation of such homogeneous population was never ever collected. We are trying to, as I mentioned earlier, we are trying to increase this. I totally agree to it. For the size of 1.4 billion population, 10,000 is really a small number. US for all has around 5 million, or 5 lakhs, I guess, 500,000. So we are also trying to march towards it, but we had to make some start. And then we never had an experience for carrying out such a huge, India had never had an experience of carrying out. There was some, if I remember, when the global screening was done, it was done for just 1,000 population. And in that, 3 samples were collected all over India, from 3 spots. And when the final data was generated, India was not even represented in that population. So, I mean, we had to make some start somewhere. We made a start, and now we are marching towards, a bigger goal. Hopefully, there would be a time when, every Indian would be genetically screened, maybe when I'm sitting on that side of the chair. So
why I'm saying this is because some is not going to help, that all of us know very clearly. But see, the blood banks, hospitals have samples. So you need categorized samples. They are available there in the blood banks.
But that is the plan. So in the 1 million
just while you are planning, if we could know what the plan is then.
In the 1 million genome, the plan is that the cohorts already made and they are really defined, they will be taken into that. So that is in the plan.
I mean, it's still under preparation, so can't spill much beans, but...
That's why I didn't ask what the plan is, but I thought that what we deliberate here may add to the plan.
It's still the plan. As I mentioned, that we are going to venture into all the disease cohorts, as well as there would be some amount which is... And, even in the cancer, if you say, it's not only the type of cancer, but even the stages of the cancer where the cohorts have to be defined. Then there has to be a longitudinal studies as well, the follow-up studies as well. So all these things are being, because as I said, we actually learned from phase 1, and now we are venturing into phase 2 after the learnings of phase 1. So it would be more comprehensive and more kind of enlightening.
So cancer cohort we are talking about, but there would be also rare diseases.
Yeah,
they are a whole chunk of rare diseases.
As I said, there is focus on cancer, there's focus on neurodegenerative diseases, there's focus on rare genetic diseases, there's focus on CVDs, there's focus on, what else? There are 7 groups, I guess. So, I mean, there would be focus on a lot of, disease cohorts.
Thank you so much. And any more questions?
Yeah, please go ahead.
So by the time they ask, I have a question like the 79 variants that you showed. So these variants, are they restricted to certain, are they pan-India? The variants that you showed, novel variants
this
that you
was
identified?
done at around 6,000. Yes, this was done at around 6,000.
No, so these variants, are they localized to certain spots or they are...
So some variants are. I mean, some because, in case of, when I mentioned about 7 million novel variants, there are some diseases which are specific to a region and some variants which are present throughout the population.
Thank you.
So, sir, I have a question regarding the data accessibility of this Genome India. So, are the raw sequencing files available for reanalysis? For the
raw
researchers?
Sequencing, you mean VCSF files or what?
I mean FASTQ files. Are they
Why
available
do you, I mean,
for the
how would you, does your institute has capability of downloading so much of, why do you need FASTQ files? I mean, and tell me globally where the FASTQ files are available. Does NCB or even this UK Biobank allows you? No. I mean, it would be, why do you need so much of data when it is not required? I mean, it would be keeping my people busy as well as you busy with your system. You just have that kind of data which is utilizable. Let's just do what is required, and why to venture into something which is not required? Because I think FASTQ file transfer, it is around, 800 TB of data. It'll take 3 years. What do you do with that? By 3 years, we would have migrated way ahead. यहाँ रोज आते रहोगे दिल्ली डेटा लेने के लिए.
Thank you, Dr. Mahajan, for that forward-looking and deep.
One more question, I think.
Yeah, there's a thing. Sure.
Simple query regarding, we have given the extension to the MRUs, multidisciplinary research units. Is there any such plan for these Nidan Kendras as well?
Sorry, sir, I didn't get the question.
So we are running 2 types of these labs, multidisciplinary research units in various medical colleges of the country, and second is Nidan Kendras. You just mentioned about the Nidan Kendras. So multidisciplinary research units are being given extension. So is there any such plan for Nidan Kendras also? Because the scientists working there, they have a concern of this.
Extension of Nidan Kendras in tenure, you mean?
Yes, sir. Yes, sir.
So, okay. So Nidan Kendras is a very unique scheme where we take an undertaking from the institute. It's not a PI-driven project. It's an institute-driven project where institute has to give an undertaking that it would continue to run its Nidan Kendra even beyond the tenure of the project. So once the Nidan Kendra is established, it is established forever.
Okay. Okay. Thank you.
Thank you so much for...
Thank you, Dr. Mahajan, for that forward-looking and deeply inspiring account of India's genomic journey. May I now request Professor Madhuji Topadia to kindly felicitate our speaker, Dr. Mahajan. May I now request Dr. Sunil Raghav to felicitate our chairperson, Professor Madhuji Tapadia. May I request Dr. Ulfat Ara to felicitate Dr. Sunil Raghav. May I now request Professor Madhuji Tapadia to kindly felicitate Dr. Ulfat Ara. Thank you, everyone. We now move on to a brief presentation from our sponsor, Revity. And for this presentation, I would like to invite Dr. Deep Jyoti Das onto stage.
Good morning, everyone. So today I'll be speaking about TotalSeq portfolio from BioLegend. So TotalSeq is the complete protein profiling and single-cell sequencing platform from BioLegend, which is for surface, intracellular, and nuclear protein detection. My name is Deep Jyoti Das, and I work as a field application scientist for BioLegend. BioLegend is a part of Revity now. So today, the agenda of the talk is I will briefly discuss about the importance of single-cell proteogenomics, what is BioLegend's TotalSeq, and what are applications of BioLegend's TotalSeq, and few new products related to this portfolio. So what is the importance of single-cell RNA-seq versus bulk RNA-seq? So consider these as two different samples. So in the first sample, we have one RNA distributed in all the 5 cells. In the second sample, all these 5 RNAs are concentrated on a single cell. So if you do a bulk RNA-seq, you will not be able to differentiate between these two samples. So one more example. Let's say we have 7 red RNAs and 6 black RNAs. And in the second sample also, it's very similar. But if you do a bulk RNA-seq, you will not be able to differentiate between these two samples. However, if you do a single cell RNA-seq, so every cell will look different. So this is how a single cell RNA-seq data looks like. This is a t-SNE plot. So here, all these dots represent a single cell, and based on how these cells are correlated, so they have been clustered into different islands. So T cell form a different island compared to the myeloid cells and fibroblast. But what are the limitations of single cell RNA-seq? So there are few limitations of single cell RNA-seq as well. So first limitation is, so CD4 cells have different subpopulations of CD4 cells within the CD4 cells, like naive T cells, memory cells, and effector cells. Here we are not able to distinguish those particular subset of the cells. Similarly, fibroblast here is very difficult to separate or differentiate among the different kinds of fibroblast. The second or the third limitation is RNA expression and protein expression, they doesn't always correlate. So just an example here. So this first graph shows the PD-L1 mRNA expression levels post-PMA treatment, as you can see in the orange graph here. So after 20 minutes, the RNA levels keeps on decreasing. And however, the MFI data, the FACS MFI data keeps on increasing, or the protein expression level keeps on increasing even after 20 minutes. So why this is happening is because RNA and protein, they have different regulatory mechanisms, and they are controlled by different metabolic cell processes. So we asked this question, can we combine cell surface marker data with RNA data to get more defined cell populations? So this is where the TotalSeq portfolio or platform comes into play. So what is TotalSeq antibodies? In TotalSeq antibodies, we have replaced the fluorophore on the FC region with a DNA barcode. And this DNA barcode has 3 different parts: a PCR handle for sequencing, an antibody barcode, which is very specific to this particular antibody, and then we have the capture sequence, which will be modified for different formats of TotalSeq. So now suddenly you are not limited to the number of fluorophores or number of channels in your panel or like your machine. So we can plex up to 200 plus markers at one go in a single experiment. So this is what the typical workflow looks like. So first we have to stain the cells using these antibodies, and then it will go for cell singularization, and then you have to prepare 2 different libraries. The first library will be the ADT library based on this particular sequence, and the second library will be the RNA level library prepared from the single cell RNA-seq. So finally, we have to sequence the data, and we have to compile and analyze the data to get the analysis out of the entire experiment. So these are the different formats of TotalSeq. So we have TotalSeq A, B, C, and D, and the formats are they differ via the capture chemistry and the platform assay compatibility. So as you can see, all these formats have the same PCR handle and antibody barcode. However, the capture sequence keeps on changing according to the format. So TotalSeq A, B, and C, they are compatible with the 10x genomics, and TotalSeq D is compatible with Mission Bio Tapestry platform. In TotalSeq A, the capture sequence is a poly-A tail. In TotalSeq B, it's a capture sequence 1, which is compatible with the 3' universal and flex gene expression assays. And TotalSeq C is a TSO. TSO is template switching oligonucleotide, which is compatible to the 5' gene expression and flex gene expression assay compatible. So if we look into the entire ecosystem of TotalSeq, it comprises of 4 core reagents. So first is a single antibody. So here we have 2,000 catalog products. You can select your own antibody and you can build your own custom panel. However, the limitation of using this first product is that you have to optimize by yourself. So if you don't want to optimize or titrate your antibody, you can use the cocktails. These are predefined cocktails, lyophilized, and the plex number is very high. We can go up to 175 plex in this case. The third is the hashtag reagents, which is helpful to multiplex samples and to identify doublets. And the fourth is the secondary reagent that will be helpful for flexible detection with biotin or tag systems. So within the cocktails, we have several layers of complexity. So the basic immunophenotyping, one of the example is human TB-NK cocktail. It comprises of 9 antibodies. Then within the mid-range, we have targeted immunophenotyping comprising of human essential cocktails, mouse myeloid cocktail, and human heme oncology cocktail, which is basically a cocktail of 100 antibodies. And the most complex is the deep and exploratory phenotyping. Within this, we have the mouse and human universal cocktail comprising of 150 plus antibodies. Different use cases of TotalSeq. So these are the different applications of TotalSeq. It is very wide. It can be used for biomarker discovery, vaccine research, immune cell subset characterization, longitudinal and dynamic monitoring, therapeutic response profiling, rare cell detection, and deep tumor, microenvironment analysis. So basically, using TotalSeq, what essentially you are doing is you are zooming into the known population of cells, and you are getting all those subset of population within the known population, which you were not able to detect via either the normal flow cytometry or single cell RNA-seq. So few more use cases here. So here in this example, using a TotalSeq A panel, the authors have generated data from 315,000 bone marrow cells, and they have resolved 80 different population types, including stem cell, progenitor cells, immune cells, stromal and transitional cell populations. In the second example, using a panel of over 190 TotalSeq antibodies, the authors have identified novel immune cell populations that can emerge in patients experiencing moderate COVID-19, and this particular population, cell population, can expand in severe cases of COVID-19. In the third example, through the use of single-cell proteogenomics, the authors have identified a dendritic cell regulatory program that limits anti-tumor immunity and is induced by expression of IL-4. So within the cocktail, we have now a version 2. So it's called universal version 2 cocktail. It comprises of 175 plus antibodies, and it helps in improve dimensionality for deep immune profiling, and it is applicable across multiple tissue or cell types. Now in future, in late 2026, we'll be coming up even with a bigger plex. It will comprise of 300 plus markers, so it will be the biggest or the highest in the market. And how it will help is it will include markers spanning different tissue types and immune cell types, and it will be helpful for deep discovery and biomarker identification. And this is ideal for large scale screening and atlas generation. So TotalSeq antibodies are not just limited to the surface antibodies. Your antibody cocktail can be a combination of surface as well as nuclear protein, surface as well as cytoplasmic proteins. And in future, we are coming up with surface markers as well as phosphoproteins. So the reason behind it is that because different compartment within the cell have different, needs different fixation and permeabilization conditions, because they have different accessibility to epitopes. And with this, I will wrap up the presentation. Thank you for your attention. For more information, you can visit or you can email me. I'll be happy to take any questions. Thank you.
Thank you, Dr. Deep Jyoti Das. If you have any questions. Thank you. Thank you so much.
Thank you.
We now have the next session titled Infectious Diseases, Vaccines and Pathogen Biology. For this session, may I request Professor Clive Gray and Dr. Nazia Mahfouz from Sher-e Kashmir Institute of Medical Sciences, and Dr. Khalid Hussain from Sher-e Kashmir University of Agricultural Sciences and Technology to kindly grace the chairs. A quick announcement, please. We have the parallel sessions going on, so I request all the speakers to upload their talks in the respective halls. We have main hall here. Hall A and B are upstairs, and hall C is way back there. The first talk for this session is an online talk by Professor Sunil Kumar Arora, former head of PGIMA Chandigarh. His topic is Mycobacterium tuberculosis and HIV co-derive a dangerous synergy in co-infection.
This delay. At the very outset, I would like to congratulate and thank the organizers for holding this wonderful conference in this wonderful place which we all refer to as paradise on earth. And I also thank you all for giving me the opportunity to be amongst you all. So without further delay, let me start my brief talk. So as we all know, Japanese encephalitis, I think this is the only paper in this session on Japanese encephalitis. So we in Northeast India, the state of Assam alone bears 30% of the Japanese encephalitis cases of the whole country. And it is a zoonotic disease, and it is an important cause of viral encephalitis in Asia. And as per the literature review, about 1 lakh people suffer from JE globally, of whom 25% is the case fatality rate. And in the endemic areas, 10 in 1 lakh people suffer due to this disease. And this is the map showing the length and breadth of this disease occurrence. So it was long back reported in 1871 in Japan, from which it got the name Japanese encephalitis, and subsequently outbreaks occurred in large outbreak forms in Japan. And it is now an important cause of viral encephalitis in Asia, the virus being endemic to most Asian countries. We have found 5 genotypes of this virus.
G genotype 1 to 5. And in most of the places in India, the genotype 3 is prevalent, which is now gradually being replaced by the genotype 1. However, from the northeast, we have isolates from mosquitoes as well as humans, and still they belong to the genotype 3. Genotype 5 is less common, but has been detected in several regions and has gained attention in recent genomic studies. So, regarding the vaccination, the very first vaccine in India was developed by Kasauli, CRI, Central Research Institute. It was a killed mouse brain developed vaccine, which was around in 90s it was released, and then it could not go further because of the mouse brain inactivated strainers process, and due to various ethical concerns also which came later on. But it was a reasonably good vaccine which required 2 to 3 doses and further boosters annually. So at present 2 vaccines are currently available in India. They are GEVE and Genvac. These 2 again are killed virus vaccines. JE in India was reported in 1955 in Vellore for the first time, and gradually the government of India launched the SA1414-2 vaccine. This was a Beijing strain vaccine developed by Chengdu in China, and this vaccine was initially released for vaccinating 1 to 15 years children in endemic areas. Interestingly, after this vaccine, a rising trend of the disease was seen among adults, which became evident in India. And moreover, the introduction of term acute encephalitis syndrome, once we started giving terminology, the acute encephalitis syndrome, more and more cases got detected, because previously it was like a suspected JE, what we used to test. Then as the terminology acute encephalitis syndrome came, and syndromic approach, with the syndromic approach, more and more cases began to be tested, and therefore the case positivity also became high. So to check this spread of JE, the government of India introduced a mass JE vaccination campaign among the adult age group, which was a very rare phenomena in the 15 to 65 years age group. And the first pilot study was done in in Sibsagar district of Assam by the Indian Council of Medical Research. That was our center. And Assam was the first state of the country to administer JE. And rightly so, as I said, that 30% of the country's burden is in Assam itself. So our study was the nested study in line with the government's JE vaccine campaign. And this observational study was conducted to estimate the immunogenicity elicited by the JE vaccine in adults over 15 years, over a 12-month study period in the study area. So we found that the vaccine was quite safe, and with regards to the immunogenicity, a cohort of 970 adults were enrolled in the study. And we did the collected samples pre-vaccination, post-vaccination on day 28, 6 months, and 12 months. So we categorized this into, we did PRNT for all these samples, and we categorized into 4 categories. One was the seronegative titer, 3 categories. One was the seronegative titer, where the titer was less than 10, and a moderately seropositive titer, which had a titer of 10 to 90, and the third group was highly seropositive, where the PRNT titer was 90 above. So in this year-long study, it was found that a total of 95.5% subjects maintained the protective antibody titer up to 12 months. And this study was documented safety and immunogenicity by the vaccine in both the seropositive and negative. Interestingly, it was found that about 9.27% were seronegative. That means the whole vaccination attempt was there to protect this seronegative group of 9.27% in the population. So in this seronegative group, the post-seroprotection was observed in 85.5%. Okay, so it was further reduced, and ultimately it was 1.5% almost of the population that we were protecting by this mass vaccination. This study outcome was presented before the ANTAGI, and then this vaccine was rolled over for vaccinating the adult population wherever high JE cases were reported. So subsequently, the government of Assam also undertook the mass adult vaccination in 9 endemic areas of Assam. So after the vaccination, the cases did come down, and the vaccine effectiveness decreased from 91%, however, to 72% in 7 years. So we were monitoring this vaccine effectiveness throughout 7 years, and ultimately at 7 years, we found that this vaccine effectiveness was insignificant. And so a study on the JE vaccine coverage in adults and children was carried out in collaboration with the Department of Health, and vaccine coverage was the main issue here, because when we did a survey, we found that only 40% vaccine coverage was there. In spite of that, the cases came down to almost 50% of the pre-vaccination era. So vaccine effectiveness in subsequent years has come down, as I told, and so a second dose or a booster dose is essential at the end of this 7 years of vaccination, initial single dose vaccine. The apparent inconsistencies in the virus effectiveness, vaccine effectiveness trends beyond 7 years, including transient increase, is notable, and notably in certain years the upper confidence interval exceeded 1, resulting in negative upper confidence interval of vaccine effectiveness. Nevertheless, the waning of the immunity over the years is inconsistent from the trend. As I have a few seconds. So we also studied the role of HLA polymorphism with humoral immune response in vaccinated individuals, and I admit that number of samples was quite low, so there are quite shortfalls in this study. Here we wanted to see the immunogenetic targets. We looked for HLA-A and HLA-DRB1, and we found that the cellular interactions presents peptides in CD8 plus cytokine T cells, and we were looking for role in JV immunity of HLA, whether drives viral clearance and destruction of infected cells, and the HLA-DRB was indicating the essential for B cell activation, driving the humoral antibody response. So we found that HLA typing we did only for 87 positive cases and 61 age and sex matched controls. As I admitted that sample size was quite low here. We did this testing in the lab of Dr. Uma Kanga in the All India Institute of Medical Sciences. And the cohort demonstrated substantial HLA polymorphism, setting a highly variable baseline for JEV immune interactions. And 4 specific L types showed odds ratio of more than 1. The prominent odds ratio of 3.58 for HLA-A suggests structural variations in this L's peptide binding groove may lead to suboptimal CD8 T cell activation against JEV. And for the HLA-DRB1, 5 L types demonstrated that the L's present extracellular viral peptides, suboptimal presentation by high-risk variants like 03 and 08 may directly impair the T helper cell activation, resulting in the weakened antibody mediated. So this threw some light on the effect of HLA on persons who had severe symptoms and others who had milder symptoms. So the gaps, I will speak of what we had in the study. Effectiveness and schedule of currently available vaccines in pediatric as well as adults need to be studied. Because now we don't have the SA1414 vaccine. And now we have the Indian GV and all. So how this would act with these are kill vaccines, so how this would boost the existing immunity provided by the SA1414-2. This remains a matter of study. And as this vaccine are going to be introduced in UPI, Universal Immunization Programs, so they hold a lot of value. Length of effectiveness of the vaccines and decision about booster dose has to be taken, and the interval, of course, and low vaccine coverage among adults and the gradual decline in vaccine effectiveness highlight the need for revised vaccine strategies, including potential booster doses. Low awareness and vaccine hesitancy remain the major barriers which lead to the vaccine coverage. And of course, we all know vaccination among adults is always a challenge, mass vaccinations. So understanding these barriers is essential for designating targeted outreach, community engagement, and flexible vaccination strategies in endemic areas. Host genetics can no longer be hidden variable in endemic disease management. So practical problems faced during the immunization program, most important is as JE occurs in low-income groups, most of the time it is seen that people are averse to multiple vaccines. In that scenario, vaccines requiring multiple doses face risk of compliance. Thank you so much.
Thank you very much. So I have a question. From my understanding, there are 5 genotypes.
5 genotypes.
Right. And your vaccine was a monovalent. Could you highlight the genetic variability and the matching of the, you may have expressed it, but I may have missed it. Could you just articulate the success of the vaccine versus the failure?
Carried out not in my lab, of course. So it said that it had a limited effectiveness for the genotype 5. But we were in good condition because we had the vaccine SA1414 Zhengdu is genotype 3. So we had the same genotype here. So here we didn't have the problem of like matching or effectiveness.
Do you think that extending this to a polyvalent vaccine is the way to go in that case, so that you cover the genetic variation across the whole of the Oceania, Southeast Asia region?
Exactly. That would be ideal. And going a step ahead, I would suggest that having a vaccine of the JE serocomplex, including West Nile, that would have been fantastic. Because in Dibrugarh or in places of Assam and more areas, West Nile is getting reported. So West Nile reports the same JE serogroup with slight differences. So, but we looked with this vaccine, whether it cross-protects the West Nile. Some protection of 30% was found, but that has to be improved.
Okay. Questions from the audience? Yes, microphone here, please.
JE vaccine for childhood vaccination is effective for longer duration or not?
With the vaccine which we studied, SA1414-2, here actually the antagi suggested 2 doses. Why? At 8 months and 14 months? Because on both the occasions, the child was administered 5 vaccines, 4 injections and 1 oral. So the child might, there was a tendency of not agreeing to all the 4 vaccines. So at JE had been newly introduced, so there was another occasion. Otherwise, a single dose was enough for that. And we did not follow up the effectiveness in the child. For how long it works, but in the adults, we saw that the effectiveness lasted for 7 years.
So, have you inquired about the childhood vaccination amongst those who was seropositive or seronegative in your area?
No, our study was not done in the children group.
That may be in questionnaire.
Pardon?
That may be in questionnaire.
Yes. Yes. Thank you. Thank you.
Hi. There is with flaviviruses, there is always a risk of AD, antibody-dependent enhancement. So, with respect to JEB, have you looked into that aspect as well? Because you have taken live attenuated vaccine, right? Have you looked into the aspect of AD with respect, or are there any studies with respect to AD?
No, we did not look for that.
Is there any studies with respect to JEB?
Actually, JE cases have considerably come down, and so has come down the funding for JE, and therefore, I think it is for you guys to look into it. And Northeast is the ideal location, because wherever the vaccine coverage had been good, so in those areas I have seen the JE has come down drastically. But in Northeast India, as the coverage was very poor, and even we advised or presented our study to the government of Assam, and they did again second drives and third drives, but the vaccination never went above 50% coverage. That was a big issue.
Thank you.
So if there is no further question, we will move towards the next talk of the session, for which we have Dr. Umair Ahmed. He is a scientist C, ICMR, NIH. His topic for the talk is Breaking Barriers, Immunodeficiencies Behind Fungal Vulnerability. Dr. Umair.
Thank you. Thank you so much.
At the outset, I would like to thank the organizers for giving me this opportunity. I'm a medical scientist at ICMR, National Institute of Immunohematology, where we deal with inborn errors of immunity. So they are rare experiments of the nature where the children or the adults don't have different components of the immune system. And today I'll be presenting our cohort of patients with fungal infections. So we all know how our immune system fights against different fungi. Both the innate and the adaptive arm of the immunity is involved. And at each and every different aspect of the pathways involved in the regulation of the infection control, there are different monogenic diseases. And that is what we wanted to look for. So we established a cohort of patients with increased predisposition to mucocutaneous and invasive fungal infections, and also we wanted to understand the underlying molecular defects and the immunopathology of patients with increased predisposition to these infections. So we had a predetermined set inclusion and exclusion criteria where patients with recurrent mucocutaneous fungal infections or deep dermatophytic infections were studied, and also along with that, if at all there was any intracellular pathogen such as Salmonella, Strepto or Staphylo, then we recorded those findings. Of course, HIV being a secondary immunodeficiency was ruled out, and other immunodeficiency conditions were ruled out. So we performed a detailed immunophenotypic evaluation where we performed the CBC, because we all know that neutrophils play an important role in fighting against these infections. Then we performed an extended lymphocyte subset analysis that looked for the T cells, the B cells, the NK cells, and the expanded phenotype of the naive memory T cells. We looked at the serum immunoglobulin levels, the nitroblue tetrazolium test, which is an assay used to determine whether the patient has a defective phagocytosis. In the patients, wherever possible, we performed an whole exome sequencing, and as Th17 is an important cell implicated in defense against these fungal infections, we looked at Th1 to 17 assay. Because in adults we don't expect a monogenic condition, we evaluated the autoantibodies against these cytokines that play a role in fighting against the fungal infections. So total of 63 patients were evaluated in our study. As expected, the mucocutaneous fungal infection was the commonest manifestation, whereas almost 45% of them had invasive fungal infections. Invasive in the sense they had invasive aspergillosis, cryptococcosis or mucormycosis. It was the female to male ratio was 0.5:1. It was a male predominant cohort and the median age of presentation was around 7 months. So here you can appreciate that candidiasis was the commonest infection in the chronic mucocutaneous group, whereas lower respiratory tract infection was a common presentation in the deep dermatophytic infection. These patients, some of these patients also had endocrine deficiencies. So when we break the patients in the group of candidal infections versus the patients with other fungal infections, the deep dermatophytes had cyanopulmonary aspergillosis, that was the commonest manifestation, followed by CNS aspergillosis. And in the candidial infection, the oral mucocutaneous candidiasis was the common manifestation. This is the immunological phenotype of these, and you can appreciate that around 15% of the patients had low naive T cells for the age, and there were increased NKT cells in a significant number of patients. Of these, some of the patients had low naive TH cells and were diagnosed as severe combined immunodeficiencies. This is the immunological analysis where we performed a Th1-17 assay and Th1-17 assay. So when we look at the defects in the immune system, coming to the myeloid abnormalities, 7% of the patients had defect in the phagocytosis pathway, and they were diagnosed to have chronic granulomatous disease, which was confirmed by using a dihydrorhodamine assay and a genetic analysis. This is the molecular spectrum from the lymphoid and the other innate component, and as per the International Union of Immunological Societies, we classified these defects. The first and the foremost, the most severe one is the SKID and combined immunodeficiency disorders. Here you can appreciate RAG1 SKID, the way we have mice. Humans also have a monogenic RAG1 deficiencies. They had invasive aspergillosis. CD40 ligand, it is a type of hyper IgM deficiency that also had invasive candidiasis. The second most common category was diseases of immune dysregulation. So here in this category, there is a classical gene AIRE that causes apposite syndrome, and these patients have endocrinopathy along with presence of oral mucocutaneous candidiasis because of autoantibodies against Th17. Apart from that, the congenital defects in the phagocytes, they were predominantly because of these two genes, the NCF1 and the NCF2. And though we conventionally don't expect, but antibody deficiencies like BTK deficiencies, PIK3 delta deficiencies also had chronic mucocutaneous candidiasis. So we performed functional analysis wherever possible. So we have performed BTK expression by flow in patients with X-linked A-gamma globulinemia. In patients with apposite syndrome, where the patients had oral mucocutaneous candidiasis, there was significantly low Th17 cells. And in the patient with hyper IgM syndrome, the CD154 expression was absent in the helper T cells. You can appreciate from these monogenic diseases that most of the patients with AIRE deficiencies, STAT3 deficiencies and PIK3 delta deficiencies, all of them had a very significantly less number of Th17 cells. Coming to the panel of autoantibodies that we performed against IL-17A, IL-22, GM-CSF. So we observed that around 15%, 1/4 of the patients had autoantibodies against IL-17A, and only 6% of the patients had antibodies against IL-22. Conventionally, GM-CSF autoantibodies can be seen in patients with invasive aspergillosis, and in around 12 patients we observed these anti-GM-CSF autoantibodies. So this is the breakup of the clinical manifestation with the autoantibodies, and you can appreciate, as expected, the autoantibodies is against IL-17A is maximum in patients with chronic mucocutaneous candidiasis, but surprisingly the number is less in patients with IL-22 autoantibodies, and it was also present in the one of the healthy control that we had. Looking at the GM-CSF and the interferon alpha, which is seen usually in patients with invasive aspergillosis or other deep dermatophytes, the aspergillus patients had autoantibodies against interferon alpha, and some patients with chronic mucocutaneous candidiasis also had autoantibodies against both interferon alpha and GM-CSF. So here, this is the genetic spectrum of autoantibodies and its presence. You can appreciate these are the different genes involved. AIRE, that is responsible for the autoimmune polyendocrinopathy syndrome, that is Apposite syndrome. These patients had a significant number of autoantibodies against IL-17, and one patient also had autoantibodies against IL-22. And though not expected, but these monogenic conditions like the GAS11 to IL-12R beta-1 also had autoantibodies against IL-17. So this is the similar spectrum for the autoantibodies against GM-CSF and interferon alpha, where in patients with CGD and Docket deficiency, they had autoantibodies against interferon alpha. So to summarize, the study highlights significant immunological abnormalities in patients with disseminated and chronic mucocutaneous candidiasis. The genetic analysis reveals a spread across different categories. There are 10 categories of inborn errors of immunity affecting different aspects of the immune system, and it suggests that the genetics contribute to the underlying inborn error of immunity, and these patients are at an increased risk to develop recurrent fungal infections. Autoantibodies against IL-17 were there in around 40% of the patients with chronic mucocutaneous candidiasis, and it plays a significant role. Autoantibodies against GM-CSF were present in a third of patients with invasive aspergillosis. Autoantibodies against interferon alpha were there in significant number of patients with systemic fungal infections, emphasizing the role of these interferons against the systemic fungal immunity. The limitation of our study was we were not able to functionally validate these autoantibodies. There are certain neutralization assays that can be done that we were not able to do. The identification of genetic variants and autoantibodies provides a valuable insight into the pathogenesis of fungal infection, and it highlights the potential targets for personalized therapeutic medicine. These findings underscore the importance of genetic and immunological evaluation in patients with recurrent or severe fungal infections. Thank you.
Thank you, Dr. Umar, for your lucid presentation, well on time. So I would like to ask among the patients who are presenting with invasive aspergillosis, which was the most common presentation, whether they are presenting
Pulmonary.
Pulmonary. Okay,
Pulmonary,
fine.
followed
Among
by
the
CNS.
Pulmonary, what was the most common clinical presentation, like what was their clinical features, like hemopsis or like?
Sorry?
What was their main clinical presentation among pulmonary patients?
They were admitted in critical care unit, mostly in the KEM hospital with cough, fever and a classical pneumonia-like presentation. But when on further evaluation after imaging studies and after scopy findings, they were reported to have aspergillosis. Then in certain patients, bile galactomannan was also done.
Okay, fine. So with this, house is open for discussion.
Thank you for your nice presentation. I have a query regarding these genetic abnormalities. They are predisposing individuals to various fungal infections. So regarding genetically dominant and genetically recessive disorders, is there any difference between the two in terms of the predisposition for different kinds of fungal infections?
If the variant, like almost all of these variants were pathogenic, and the ones that were VUS, we tried to do the functional assays. So because ours is a pan-India referral center, what we observed that the patients who are referred from the southern part of India, they had more of autosomal recessive diseases like chronic granulomatous disease, the NCF1 deficiency, and in rest of the part, it can either be dominant or X-linked diseases like X-linked A gamma globulinemia or CD40 ligand deficiency. So per se, in the spectrum, dominant versus recessive might not make that much of a difference, but the actual gene that is responsible, it does vary the phenotype of the patient, how the patient and which fungus affects, like is it CMC or the invasive types.
Thank you.
Yes, we have one cardinine deficiency. So it was a very interesting case. The female had Exorophyllum rostratum. It is a very rare fungus. So the fungus was identified first because she had very bad exuberant exophytic nodules. And the dermatologist, they identified the fungus first, and then retrospectively when we did the exome analysis, we were able to identify cardinine deficiency.
I have a question. Thank you very much. What do you think the mechanism is for these auto-antibodies against cytokines, knowing that different fungi produce cytokine homologs, which are not quite cytokines, but enough to produce maybe antibodies? Is that a mechanism?
It's a very interesting question, and like many people have tried to identify, like Anfield from INSERM and Dr. Stephen Holland from NIH. They have over the years been trying to do that. So what they have described is that the total antibodies might not lead to the disease per se unless it is a neutralizing one. So like the downstream, like suppose IL-17 antibodies, a downstream STAT3 functional STAT3 assay where the patient's STAT3 phosphorylation is down-regulated, that may explain the disease, neither just the presence of autoantibodies, because it can also be seen in healthy individuals. Thanks.
Great, thank you very much. Okay, our next speaker is Dr. Alti from the ICMR in Patna. And I don't think I can pronounce the name of this. But he's going to talk about Leishmania peroxidases and M2 macrophage markers to control the infection. You can tell us how to pronounce that awful sounding name.
This one? कौन सी आपकी?
Is there a problem? We could move on to the next speaker and come back to you, Dr. Alti. Should we do that? Okay.
So as there is some technical hiccup with Dr. Alti, so we move forward with the next talk. It is by Dr. Ruqayya Nazir.
A very good afternoon and Assalamu alaikum to the whole audience. First and foremost, I would like to thank the organizers for giving me a chance to speak here at this forum. So, the title of the talk is Microbial Peptides as Immune Modulators, Clinical and Translational Perspectives on Bacteriocins. So AMR burden, I think we all know regarding the AMR burden. It's the scenario in India is highest in the world, I can say. So roughly 3 lakh people in India every year, they get AMR infections, and including thousands of newborns among them. So we all know regarding the main drivers of AMR and then the cost of AMR, it's increasing day by day. And here, basically, I am here to discuss regarding the innovations against AMR, for which WHO is highly giving funds and all, encouraging people to work on the innovative molecules, innovative therapeutics against AMR. So, mechanism of resistance, I won't touch, everybody knows it. What are the mechanisms? Like we have inactivation of antibiotics by enzymatic degradation, then we have decreased influx due to the altered cell membrane, we have activation of antibiotic efflux pumps, modification of the antibiotic target genes. So here our interest is basically the antibiotic alternatives. We have a number of alternatives to the antibiotics. We have phage therapy, we have probiotics, we have bacteriocins, we have fecal microbiota transplants, we have monoclonal antibodies. So different mechanisms are there where alternatives to antibiotics, they bring antimicrobial effect, like they bring bacterial cell damage, extracellular action, neutralization of the microbe-derived products, then even the production of antibacterial molecules, where they enhance the mucosal barrier, competitive exclusion by adherence to the mucosa epithelium, and even by the biofilm inhibition, and immune modulation by means of cytokines, by means of T-cell regulations, and other effector molecules. So here, our interest in our lab, the work that's going on, we work on probiotics, which are basically the lactic acid bacteria, which release the bacteriocins, that are the antimicrobial peptides, and they act as therapeutic antimicrobial agents against a number of pathogenic microbial infections, and which is an urgent command at the moment. So why lactic acid bacteria? There are many probiotic labs. They produce antimicrobial proteins in form of bacteriocins, which have a tremendous medical importance. And their exploitation in medicine is nowadays a novel approach against AMR. And thus the potential labs, isolated from cheap and economical sources, they have valuable health benefits as therapeutic agents and act as natural preservatives. So antimicrobial substances of lactic acid bacteria, we have number of them. Like we have the organic acids, we have the hydrogen peroxide, we have carbon dioxide, we have diacetyl molecule, we have the ryoterin, we have acetaldehyde, And we have bacteriocin-like inhibitory substances, and then particularly the bacteriocins, ethanol, and then others, acetylcholine, and then the ammonia. And here, our lab is basically interested in looking at the interventions through native labs and their derived bacteriocins. So, extensive use of antibiotics that has led to the antimicrobial resistance, dysbiosis, diarrhea, and so bacteriocins, they are potent alternatives to these antibiotics with a number of applications, like they have applications in food industry, they have applications in pharmaceutical industry, and even in agriculture. So we have other alternatives to antibiotics as well, like some of them are plant-derived bacteriophage, animal venoms, and then the secretions. So how these bacteriocins basically work, they have different mechanisms that facilitate the inhibition of pathogenic microbes through signaling quorum sensing and signaling immune cells, and thus modulating the host immune system. So a role of bacteriocins is not limited to only these. We have a number of roles with bacteriocins. They are anti-hypertensive, they are anti-diabetic, they even modulate our microbiota. They also lower the cholesterol, and even they play a number of roles in the immune modulation. So here we just understand that bacteriocins, how they act basically along the bacterial membrane. There are a number of mechanisms, like we have gram positive, gram negative, so I won't go much into the details. We have a number of immunomodulatory mechanisms of bacteriocins, and they have the antiviral and the antiparasitic actions also. So they have anti-cancer and the anti-inflammatory action also. So our lab, we have a multidimensional sample collection to get these functional immune modulators for the host health. These all samples are taken by different PhD students working in our lab. So this part of the work is carried out by the Hanif Mohammed and in collaboration with Dr. Zahid from SKICC. So here what we have done, we have taken the native chicken, and then we have isolated the lactic acid bacteria. We have pursued for sequencing and got some number of potent sequences. So from these labs, and further they have been checked for the safety assessment and the probiotic characterization. So this shows the antimicrobial activity of cell-free, supernatant, and neutralized cell-free supernatant of bacteriocin producing labs against chicken bacterial pathogens. The next table shows the bacteriocin activity, where NC10 and BC3 has shown highest activity. So we have selected the lab strains with probiotic potential based on the PCR analysis, and we found two standout strains, NC10 Legilactobacillus salvaris and the Enterococcus durans. And further, these were checked for different with the different treatments. So for purification, the characterization, we have purified NC10 bacteriocin, and it was found to be 7.2 kilodalton, stable at 100 degrees Celsius, proteinaceous molecule, highly active. And on LC-MS, it confirmed peptide sequence, and 90% homology was found with the known bacteriocins. So this was the best candidate, and we had implications. There was a promising antibiotic-free feed, which we can add in the poultry, and ultimately it reaches to the human race. So, I have already discussed some of the important points, like Legilactobacillus salvaris, NC10 was purified, and then further, these were some of the publications from the study. So next study was done by the Safoora Nisar in collaboration with Dr. Hasib from Department of Bioresource, University of Kashmir. So wherein we have done the probiotic characterization and the in vitro safety evaluation. And in this study, we have got some novel antimicrobial peptides, and which we are looking forward for getting them characterized and then move forward for the patenting. So here this study also gave us this piece of publication, and some more publications are ready to come. So these are some other publications from these studies. So the probiotic consortium studies here, the immunomodulatory study, the action plan is as such. So we are undergoing in vitro model and the in vivo model infection. We have already given the challenge with the bacteriocin in the THP macrophages cell line, and then the cacointestinal epithelial cell line, and then we have cells have been infected with the E. coli, Salmonella aureus, and then the Salmonella strains, and Staph aureus, sorry, and then the Salmonella strains. After infection, bacteriocin was added at different concentrations, and different cytokines were measured, and other parameters were measured. In vivo model study is still continuing. We have given the model that is still going on in the lab right now. And further, we are looking forward for getting the results for the cytokines and the other parameters. So the possible mechanism here for the immune modulation is downregulation of NF-kappa B during infection, upregulation of the anti-inflammatory cytokines, reduction of infectious inflammatory mediators, and modulation of the TREG-TH17 axis, and the production of mucosal tight junctions and barrier integrity. So the expected significance here, if the bacteriocin shows the immunomodulation, which is quite expected as we have already checked in the in vitro model, and the same is expected from the in vivo as well. It can serve as the dual action therapeutic, antimicrobial as well as the anti-inflammatory immune balancing. Potential applications are gut infections, sepsis, systemic inflammatory disorders, adjunct therapy alongside the antibiotics, and we can introduce a new candidate postbiotic with the therapeutic relevance. So these are the list of ongoing PhD scholars with these topics, and these are already awarded students, and a few of them have submitted. Thank you.
Thank you, Dr. Ruqayya, for your elaborated presentation. Definitely this antimicrobial resistance is a burning topic and needs urgent attention. We need to devise strategies to reduce the incidence of AMR. I would like to ask, can you give us some practical tips out of your research for our clinician and physician so as to reduce the incidence of AMR in our community?
Very nice topic. Actually, we are recently working on one of the manuscripts with the same kind of question as you have asked. Like basically, what is needed for the clinicians? I can say that they must educate. They must try to educate the patients to restrain from getting the over-the-counter top medicines, and the surveillance is very important, and which is important that for the different funding agencies, like we have ICMR, we have DBT, we have many other funding agencies, which can put forward more and more grants towards the surveillance things, so that people will come to know regarding the AMR, because even in India and in remote places, people don't know that how they are getting affected with the over-counter medicines and all.
Definitely, thank you so much. So with this, house is open for discussion. Mic, please.
RNA. Genome sequencing, 16 RNA we have done. We have.
So you have mentioned other parameters, and in that you have included survival analysis.
Yeah.
So
safety assessment.
Yes.
With that, we have done the virulence genes. We have done the virulence genes. We have done the hemolytic tests also.
No, did you perform survival analysis?
Survival analysis will be done in vivo.
In vivo.
That will be done in vivo. That I was telling that that is still undergoing.
Okay. Undergoing. Okay.
So, okay, if there is no more question, we will move towards our next talk. It is by our speaker, Dr. Shukla Das. Oh, Dr. Alti.
So we're going to go back to Dr. Alti, who is he in the house? Yeah. Sorry, madam, we're going to go back to Dr. Alti.
Okay, all right.
So not just yet. Yeah. Thank you. The floor is yours.
Good afternoon. Thank you for giving this opportunity. I am Dr. Daya Karalti, Scientist C from ICMR RMRMS Patna. Today's my presentation. Today's my presentation is azardione targets Leishmania peroxidases and M2 macrophage markers to control the infection in silico and in vitro insights. This compound azardione derived from neem leaf ethyl acetate fraction. It is a tetracyclic triterpenoid compound. Talking about Leishmania, it exists in two life stages. One is bromastogote, survive in the sandfly vector. Second one is amastogote, resides in the host macrophages. When these amastogotes replicate inside the host macrophages, we can see the blown up symptoms like irregular bouts of fever, anemia, weight loss, deadly symptoms like hepatomegaly and splenomegaly. If left untreated, these patients will die within 2 years. Coming to the statistics of Kala Azar in India, especially in 2026, between January to March, already 60 cases are reported. 9 are HIV co-infected cases, 13 are post-Kala Azar dermal leishmaniasis cases. Still, this disease is existing in some endemic pockets in Jharkhand as well as Bihar. After implementation of National Kala Azar Elimination Program in India successfully, the cases are significantly declined between 2014 and 2025. Whereas the global scenario is really pathetic, still the disease is prevalent in more than 70 countries, and every year 4 lakh new cases are being reported to WHO with 20,000 deaths. Yes, there are several challenges in the Kala Azar treatment that includes drug resistance, drug toxicity. The big challenge is co-infections, especially the Kala Azar is more opportunistic in HIV and TB patients, and lack of pharma interest to develop new drugs or vaccines. These difficulties are ultimately demanding for alternative, safe, affordable and effective anti-leishmanial therapies. Our hypothesis is that neem could be possible drug target for the treatment of Kala Azar. We have initiated working on neem long ago. We have made 4 different fractions from the neem leaves using 4 different solvents. Among them, ethyl acetate fraction was very effective to kill the leishmania. From this fraction, we identified around 14 compounds by performing the LCMS-MS analysis. Interestingly, none of these 14 compounds, at least one of them is also not evaluated for anti-leishmanial activity, but few of them were already evaluated for anti-inflammatory, anti-malarial, antifungal, anti-cancer activities. Among them, azardione is also there, but it is not yet explored against leishmania. Our research question is that can azardione kill leishmania? Don't know any. If it kills, what are the molecular targets? And how does it affect the host-parasite interaction via immune modulation? To address these research questions, we have designed the methodology in 3 parallel tracks. We performed in silico studies by conducting the molecular docking and simulation with azardione by targeting leishmania trypanothione metabolism enzymes, which is unique pathway in leishmania. And in the second track, we performed in vitro studies on bromastogotes, where we performed growth inhibition assay, genomic DNA fragmentation, reactive oxygen species estimation by flow cytometry, finally, leishmania drug target gene expression by reverse transcription PCR. In the third track, we performed in vitro studies on intracellular amastogotes. Before going to the intracellular amastogotes, we evaluated cytotoxicity levels on the host cells. Here, the THP1 macrophages are working model. After that, we measured the rate of infectivity using microscopy. Then M1, M2 macrophage specific markers were evaluated at gene level and protein level by real-time PCR as well as ELISA methods. Coming to the results parts, molecular docking is predicted that leishmania peroxidases are the potential drug targets for azardione. Followed by, in the peroxidases, there are 2. One is ascorbate peroxidase, second one is triparedoxine peroxidase. Ascorbate peroxidase is reported for high docking score, followed by triparedoxine peroxidase. So, apart from these high docking score, they are unique to the leishmania. These 2 factors are motivated us to move ahead for the evaluation of anti-leishmanial activity of azardione at in vitro level. As I told earlier, we have performed a simulation. This is done for almost 100 nanoseconds. Throughout the 100 nanoseconds, azardione was stably interacting with the active site of ascorbate peroxidase. That was confirmed by stable RMSD, minimal RMSF, and stable SASA, solvent accessible surface area, and consistent radius of gyration and sustained hydrogen bonding between the ligand and enzyme. Then we move to the in vitro validation of the azardione for anti-leishmanial activity by testing against bromastogotes. We used 3 different concentrations. With increasing concentration, azardione is inducing structural deformalities in the parasites. At the same time, we observed At the same time, we observed decrease in the viability with increasing concentrations. On the other hand, we also measured propylium iodide positive dead cells using flow cytometry. With increasing concentrations, they were found to be upregulated. So from these three experimentations, we calculated IC50 around 17.09 micromolar. At this concentration, we wanted to test the DNA fragmentation that was clearly visible on the gel. And we also measured intracellular ROS levels that was found to be upregulated within the parasite. From these results, we demonstrated that azardione is inducing ROS-mediated apoptosis in the parasite. To validate the in silico prediction of Leishmania peroxidases or the drug targets for azardione, we measured their gene expression levels, both ascorbate peroxidase and tetrodotoxin peroxidase. Their levels were downregulated. That downregulation is correlating with increased ROS levels. This ROS is correlating with the apoptosis death in the parasite. Then we move to the in vitro studies on amastigotes. As I told before, first we have measured the cytotoxicity on the host cells. So with increasing concentrations of the azardione, the viability of the macrophage was gradually reduced. From here, we calculated the CC50 concentration, 50% cytotoxic concentration, around 56.32 micromolar. Then we move to the infectivity studies, where macrophages were infected with different concentrations of azardione within the CC50 range. With increasing concentration of azardione, the rate of infectivity by means of number of intracellular amastigotes per 100 infected macrophages were gradually downregulated. So from here, we calculated 50% EC50 concentration around 11.67 micromolar. You can see here a representative image of Giemsa-stained macrophages, where A represents uninfected, B represents infected controls, and C is infected plus azardione treated macrophages. From here, we demonstrated that azardione is biocompatible in nature to the macrophages while inhibiting the intracellular amastigotes growth. As we know, macrophages are immune cells. We wanted to measure the M1, M2 macrophage specific markers. Generally, M1 macrophages are associated with host production. M2 macrophages are associated with the disease progression. Here, M1 macrophages are interleukin 12 and indivisible nitric oxide synthase. These two genes expression was downregulated during the infection, but after azardione treatment, they were significantly restored. On the other hand, we also measured M2 markers like interleukin 10 and arginase expression. These two are significantly upregulated during the infection, but after azardione treatment, they were significantly downregulated. Among the cytokines, IL-10 is a key determinant for the disease progression. Those IL-10 levels were measured in the culture supernatant by ELISA. We found it was upregulated during the infection, but after azardione treatment, it was significantly downregulated. From here, we inferred that azardione is modulating the host microenvironment in the favor of the host to combat the Leishmania infection. So these are consolidated results. On promastigotes, we found IC50 around 17.09 micromolar. On host cells, it was around 56.32 micromolar. Intracellular amastigotes, the EC50 concentration around 11.67 micromolar. From CC50 and EC50, we calculated selectivity index around 4.83, which is moderate when compared to the effective drugs. But in case of leishmaniasis, it is highly considerable. The notable finding from this study is that EC50 is lower than the IC50, where it indicates azardione is modulating the host microenvironment hostile for the Leishmania. So from this study, the key findings are molecular docking and simulation predicted Leishmania peroxidases are the possible drug targets to azardione anti-Leishmanial activity. In vitro studies on promastigotes validated ROS-mediated apoptosis in Leishmania donovani by downregulating peroxidase expression. In vitro studies on intracellular amastigotes demonstrated azardione leishmanicidal activity by inhibition of M2 macrophage markers and upregulation of M1 macrophage markers. Currently, we are doing animal model studies for the evaluation of safety and efficacy of this drug, and we are also planning to do the combinational therapy with existing drugs to cut off the toxicity by cutting down the dosages. Thank you. This is my research institute. I have established lab very recently. They are my team members. Thank you.
Thank you very much, Dr. Alti. Open to questions from the audience. Yes, microphone there, please.
So, your compound is a natural compound, no?
Yes, sir.
Is there any alternate compound if suppose shows some sort of the drug resistance, then what's the alternate compound?
You are talking about natural compounds, sir.
Yeah, natural or synthetic?
Yes, sir. Yes, sir. We have already our drugs are developed resistance. We are using amphotericin B and miltefosine. They already developed resistance.
Okay, thank you.
Yes.
So I have a question. In your macrophages, your in vitro studies, did you? It wasn't clear. How did you isolate your M1 and your M2? How did you gauge what AZD was doing to those cells, the different polarities of those cells?
Yes. No, sir. We didn't do any polarization studies. We didn't use any surface markers to differentiate M1 and M2. We directly measured the ELISA, gene expression levels of M1, M2 associated markers. So in animal models, we will do all these things, sir.
But what did you take? Peripheral blood cells from the patient?
Sir?
Or what was your starting material?
I didn't get.
What cells did you take?
I've taken THP1 macrophages, sir. The THP1 macrophage
Cell lines.
Cell
THP1s.
lines.
I see. Okay.
Yes.
Thanks.
Okay, if there is no more question, we will move toward our next talk of the session. It is by Dr. Shukla Das. Ma'am is a director and professor and head of University College of Medical Sciences, GTP, Hospital, New Delhi. The topic of her talk is Host Immune Landscape in Dermatophyte Infections, Insights from Transcripts.
Good afternoon, everyone. Good afternoon, Chairperson. And I thank Dr. Dilap Rose Madam and her team for inviting me. So my topic is host immune landscape in dermatophyte infection, insight from transcriptomics. I take you through this. So this disease, superficial dermatophytosis, is now taken a pandemic situation in our country. And although it is superficial, but it has now become chronic and recalcitrant, and it is causing a lot of relapses. Globally, it has almost taken a pandemic form. And globally, in fact, there are 25% of population are infected by superficial fungal infection. This is known as tinea infection by common name. Dermatophytosis in the earlier days used to be, considered to be treatable, and a clinician never asked for any laboratory diagnosis. By looking at the clinical manifestation, they could treat. But what has happened over the years, that not only the dermatophyte has undergone a change taxonomically and anthropophilic, it was zoophilic considered, now it has crossed species barrier, and it has become chronically adapted to the skin keratinized tissue. So it is an infection of the superficial keratinized tissue. And this diagram just tells you that various factors have led to its chronicity and persistence in India and globally. Few are pets, tight clothings, and one of the things which are very common in India is a fixed dose combination use of steroids with antibacterial, antifungal. So steroid has given the fungus to establish itself and produce chronicity and persistence of their infection. So this is just giving you the epidemiological data, which tells that dermatophytosis can occur any part of the anatomical sites, but tinea cruris and corporis, these are the areas which are maximally affected, mostly familial, and has been found to be more affected in male than females. This is just a picture microbiologically where it resembles the original T. mentagrophyte, which was about 20 years back. We were identifying them as a very harmless pathogen, but now it has taken a very dangerous situation, and a clinical, for the clinician, dermatologist, has become a therapeutic challenge. This is globally has been found to be even challenging because it's terbinafine, which was the drug of choice, resistant. So it has shown amino acid mutations at 493 and various other mutations which are in the map. And these have led to squalene epioxidase resistance and dermatophyte. These ones are not getting cured by terbinafine. And this is an example of one of the cases where you see lesion starts from a small lesion and they are so extensive and large and concentric lesions, and they extend from the trunk down to the genitals. And you can see the number of antifungus that was given to the patient. Finally, there was a combination of itraconazole and terbinafine which resulted in cure. Now, this is where we wanted to ask that since it is a polymorphic disease and superficial fungal infection, which is immunologically, it keeps a very low profile. Are there any immunological dysregulation which is occurring in this infection? So, just to talk about the virulence, they have 500 and more proteases and enzymes that they secrete. Once the conidia attaches to the keratinocytes, they secrete large number of proteases, and then they establish and start reproducing. These are a few example of endo and exoproteases, and we also did first study the pathogenicity of these mentagrophyte. Taxonomically it is called T. mentagrophyte interdigital complex, and a new nomenclature has been given as Taenia indotini, which is terbinafine resistant, highly virulent, originating from India. So we took some isolates and we saw that these subtilisins and these endoproteases are extensively produced as compared to the previous isolates. This is an immunologically low profile disease. The immune responses has not been studied very vividly. Animal models are not existing. Guinea pig is supposed to be, but very hard to establish such infection. And innate immunity, if it is intact, so antimicrobial peptides and beta defensin can clear the infection if it is targeted at an early date. And this, as our earlier speaker said, that these are certain deficiencies genetically. If they are identified, then this can take a serious mode of infection. Now, innate immunity, we know that this is very polymorphic, and scientists have said that if suppose innate immunity is intact and the patient comes with a very small lesion and it is treated, the trigger goes away, then the innate or the Th1 response can take and build up a memory system, and even if there is a relapse, the infection gets cured. So it was identified that there can be a quick epidermal turnover, phagocytic activity and antimicrobial peptide if it is intact. Then it can be cured if it is treated early. But adaptive immunity is very important. So we identified that cell-mediated immunity is very important for even this superficial fungal infection. This is just giving you a picture, a cartoon, where we know that there are various antigens, mannan, glucan, all these antigens have a tendency to attach to the receptors and produce various toll-like receptors, and then they produce the infection, Th1 and Th17 responses. But if Th1 was robust, then why this infection persisted? So we wanted to identify this, and if Th1 was protective, why they are suffering from this? Is there any role of Th17 and T regulatory cell? So the adaptive immune response, I'm not going into the details. It is like an orchestra. Some are down-regulated, some are up-regulated, and we wanted to find out what exactly is happening in these patients. So we started with a trichophytin antigen. We developed this antigen and purified, and we started doing these intradermal antigen testing, and we found that delayed hypersensitivity reaction was numbed, was suppressed. They all had immediate hypersensitivity reaction, suggesting that there is a predominance of Th2 response and Th1 was absent. So these are the key players, and we wanted to look for both CD4 T regulatory set, Th17 and Th1 responses, and for this we had a paper where we identified the responses of Th17 and T regulatory, and in this methodology we took healthy controls and these patients, PBMCs. These were stimulated with trichophytin antigen, and the PBMCs were stained with various CD4 antibodies and the various markers, cell surface markers, and cytokine expression was studied on the supernatant. This is how the experiment went about, and very surprisingly we found that Th17 was very high. T regulatory was also was found to be very high. These were the gating experiments, and this is what we found. And trying to figure out, this experiment just told us that Th1 and Th17 were competing for each other, and interleukin, this shows that interleukin gamma interferon, even if it was high, the IL-17-23 axis maintained a very high Th17 population, and Th17 was suppressing and subverting the protective immunity that Th1 could give. And simultaneously, there was interleukin-4 was also increased. That means they were giving an effect of energy and tolerance to this fungal antigen. So Th17 and T regulatory players were the key players in this infection. And because TGF-beta was found to be very high, probably TGF-beta was converting the T regulatory cell into Th17. So we all know that there is Th17 T regulatory cells, 5 to 15% in circulation, and they maintain homeostasis. But if they go unchecked and there is high exacerbation of T regulatory cells, then they have a potential to cause collateral damage. So this is what probably was happening. The T regulatory cells being more plasticity, so they were converting into Th17, and inflammation was exacerbated in these population. Then we also checked the MDSC's population. The MDSC population also keeps a check on the T effector responses. We found that both granulocytic and monocytic MDSCs were high, and they were suppressing the Th1 but accelerating and exaggerating the Th17 responses. This was our last ICMR project, and where we did the RNA-seq sequencing. And just to conclude that whether Th1 and Th17, which one is predominant in this dysregulated immune responses in this patient, we took 6 patients, confirmed T mentagrophyte species complex, and we took the PBMCs and isolated the RNA by the RNA-seq preparation. So we found that once polarized CD4 T cells acquire a stable phenotype, cytokines can again reprogram and change their phenotype. IL-6 can convert Tregs to Th17. Further, Th17 upregulated the TBX21 and gamma interferon response to Th1 polarizing cytokines. So this was our results, and what I'd like to conclude by saying that global transcriptomic changes that we saw in dermatophytosis, 120 differentially expressed genes, 93 genes were upregulated, 27 genes were downregulated, where we found that IL-17 was predominant, Th1 was following this. We also found there was metabolic reprogramming, innate lymphoid cells were also dysregulated, and chemokine signaling pathways were also very extensively working in coordination for maintaining the Th17 population. And we also found that there was an epithelial damage and epithelial leakage. So to conclude, early predominance of Th17 response is followed by a Th1 activation. The ROR gamma T STAT3 signaling is associated with fungal clearance. And gamma interferons are linked with sustained antifungal immunity. FOXP3 downregulation suggests a dysregulated T regulatory. So dysregulated means the T regulatory were converting into Th17 and acquiring the ROR gamma T phenotype. So this is a volcano picture which is showing how many genes which I just expressed, that there were several genes which were upregulated and many genes which were downregulated. So Th2 response is minimal evidence, and upregulation of IL-33 was showing that Th2 response is maintaining the T cell energy and the tolerance to this fungal infection. So there is a complement. This is the conclusion that innate lymphoid cell activation was observed. Upregulation of certain molecules, trained immunity signals was also upregulated, complement activation was upregulated, and there is barrier remodeling showing that there is an epithelial leakage in these patients, telling us that these patients are into chronic inflammation. So these are the key implications. And conclusion is this, that in such patients who are suffering from chronic persistent dermatophytosis infection, the immunological parameters does not come back even with treatment. So now the clinicians have understood that the treatment should be continued even if there is clearance of lesions and even if your microbiological evidence becomes zero. Even then, for 4 weeks, continue the treatment so that your immunological parameters come back to normal and Th1 can be regained and Th17 and T regulatory come back to their normal levels. So thank you this is my team of PhD workers and postdoc.
Thank you, ma'am, for your lucid presentation. I would like to ask, which is the most common cause for the recalcitrant nature of this disease?
So in India, we have found that I told you that fixed dose combinations. So you see in the advertisement, they use the ring guard. So now clinicians are trying to ban that cream, which has steroids, antibacterial and antifungal. So with steroid, you are suppressing the Th1 to actually act and bring down the infection. So over the counter taking ointments is a big challenge, which a lot of Indian populations do so.
Very well said.
And then keeping pets also. And actually, if it is a familial infection, dermatophytosis, so you have to maintain a lot of personal hygiene. And this tinea indutini has become very adapted to the skin keratinocytes. And it's it's found its own niche and is very difficult to remove. That's why treat as early as possible.
Thank you, ma'am. Second point is that what is the sensitivity and the specificity of that skin testing test that you have mentioned in your presentation?
So, trichophytin antigen testing, we just did to find out, to convince our clinicians that there is cell-mediated immunity is also there. In fact, everyone knows that superficial fungal infection, why should we require a CMI? Because the innate immunity is good enough. So just to prove this, CMI is depressed in this. This intradermal skin testing was done, which shows that it was immediate hypersensitivity. That means there was a Th2 predominance in these patients. And they were more atopic, and very high IgE response was also there. And humoral antibodies, as we know, is not at all protective in these patients.
Thank you. With this, house is open for discussion.
No, only transcriptomics we did only on 6. Otherwise, we had taken about 50 samples in the initial Th1 and Th17 samples, Th1 and Th17 on PBMCs. That was done on around 50 samples, but the transcriptomic was only done on 6 samples because of the cost. So we are going to continue with further taking more recruitment of more patients and establish more data on this. Yes, for the flow cytometric. So for flow cytometric.
Okay, thank you very much. We'll move on. I've just been told we need to stick to time. So the next speaker is Maju Chanda from Manju Jain. No, that was the person just spoke, right? Oh gosh. Manju Jain.
So with this, we'll move towards the next talk of the session. It is by Dr. Manju Jain. She's an associate professor. The topic of her talk is atypical leishmaniasis, a neglected disease phenomena of concern towards sustenance of VLN elimination. Dr. Manju Jain. This one.
Yes, a very good afternoon, all of you, the chairs. So I thank Immunocon 2026 and Dr. Dil. I can see her finally here in the hall. My deep-felt thanks to give me an opportunity to present a piece of the work carried out in our lab. So I'll try to make the presentation less data intensive and would like to highlight this phenomena of atypical leishmaniasis, which is different from the visceral leishmaniasis, about which some of the talks were, carried out yesterday, and today Dr. Dayakar has already covered some of the basics. So, as a thrust area of ICMR in alignment with the WHO roadmap, for the elimination of neglected tropical diseases, the VL has been successfully taken care of in context of, the number of different blocks in the country depicted by a red dotted area in the northeast. The prevalent form of the disease that we have been traditionally aware of, caused by Leishmania donovani, the classical form which we are aware of. So already the country is declared with the status of VL elimination with the achievement of the threshold of less than 1 case per 10,000 population in the different 633 blocks in the afflicted areas. And right now we are in the consolidation phase, almost at the end of consolidation phase, where the validation of this elimination threshold of less than 1 case per 10,000 has to be sustained. And we are about to transit into this maintenance phase, wherein we ensure that the VL elimination status happens on a sustained level. So to take care of that aspect, different components which are ongoing under the National Kala-Azar Elimination Program are like the newer endemic regions, the new relapsed and asymptomatic cases, the PKDL cases, and the HIV-VL co-infection cases that present as active reservoirs for the Leishmania donovani per se, which are actually included in the policy healthcare management at present, and it has to be taken care of during the entire maintenance phase. So what I'll be focusing on today is more on atypical cutaneous leishmaniasis. Why I say atypical? Because it is like the cases are having peculiar localized cutaneous lesions, but the disease is caused by Leishmania donovani. And still these cases, which are like it's an emerging disease form, is not included in the national reporting. So as you can see, okay, let me tell. If you look into the India map, apart from the classically L. donovani mediated VL cases represented by the red dots, we see the atypical CL cases are coming up in parts of India and Kerala. And we are projecting that HP is the newer endemic state, Himachal Pradesh. Very newer studies are coming from Nepal also, and Sri Lanka is already an established country for this disease, atypical disease phenomena. So coming to our study area, that is Himachal, we see that the number of these atypical CL cases caused by L. donovani, they are on an increase. And also there is an expansion of newer endemic pockets in the state. Majorly the cases are concentrated along the Satluj river belt, and newer endemic pockets are moving towards the western side of the state. So being a basic biologist, I would be interested in knowing as to why Leishmania donovani, traditionally known to cause the VL disease, is actually pushed towards causing the cutaneous manifestation. So we looked into specific components of the parasite biology and the host immunity that determine the disease outcome. So with the clinically confirmed cutaneous leishmaniasis cases, as we can see the LD body positive cases with the typical cutaneous lesions, we typed the causative parasite species, and it came out to be ITS, based on ITS1 and 6-PGDH initial screening for the species identification, we can say that the causative parasite is the Leishmania donovani itself, causing the dermotropic manifestation. And these isolates are very distinct from the classical VL causing LDs. So with this basic background, we further wanted to know, based on the whole genome sequence analysis, regarding the origin, genetic relatedness to the VL causing LDs, and definitely the biology based on the whole genome sequence variations that can possibly explain the dermotropic capability of LDs in these specific regions. So we up till now we have sequenced 3 isolates from the region. They are a bit fastidious to grow in the lab conditions. So it is like CL66, 71, 76. One of them, all 3 are based on the parent haplotype analysis. We see that they are tracking back to Nepal. And that too, they doesn't belong to the classical Indian subcontinent core genomes that have already been sequenced, but actually they belong to the ISC1, a divergent clade from the ISC core. There also they are more closer to the further divergent LDBPK 512 among the ISC1 strains. Among these 3, one of the LD came out to be actually an intra-species hybrid, such that 2 LD strains, they cross to create something, the hybrid strain which was isolated from a ACL case. And then the other 2, again, they are more closer to the ISC1 as I specified. So based on the whole genome sequence analysis, in comparison to the VL causing reference strain, we could see a large number of ploidy changes, copy number variations, indels, single nucleotide polymorphisms, and based on the the gene variant annotation enrichment, many gene variants, a humongous amount of these gene variants, they related to phenomena like genotype, virulence, adaptation to host stress, growth, metabolism, motility, and processes involved in genetic information like replication and chromatin remodeling. So with this derivation on at the genomic level, we found these CL causing LDs are very, very different from the VL causing LD in terms of large number of gene variants that are associated with some of the important biological phenomena that actually they can actually drive the disease outcome. Phenotypically, I give a small snapshots. These isolate, they are much slower in their growth as promastigotes as well as amastigotes. In fact, the promastigotes are transforming to amastigotes on a very slow time scale. Infectivity wise also they are compromised based on our in vitro studies on THP1 and PEG based infectivity assays. In vivo mice based studies, we could see that these atypical CL isolates could also disseminate to the different visceral organs, just like LD Bob, the VL causing parasite culture, and even by the parasite specific PCR, we could say it's disseminating to the different organs. Importantly, these ACL isolates could also be transformed successfully from the mice to the pro-mass culture. And based on semi-quantitative and quantitative data also, which I have not put here, although the parasite load seems to be lesser. It's systematically present, but the load seems to be quite low. Again, coming to being a biologist, I would really like to further get into the details of why the LD variant is a more of like dermotropic nature rather than viscerotropic. So we have taken out specific genes that relates to metabolic pathways, chromatin remodeling, and also the interesting genes that are getting altered in terms of the copy number variations, and also the variants in terms of non-synonymous SNPs. We have some key immunodominant antigen specific genes, and that does sound quite obvious because the pool of antigens which these LD variants will present to the host system will be quite different. We can see like the RK39 based test, we have VL positive, very clear, strong bands, but in some fraction.
Could you conclude?
Oh, I'm already over. So they are systemically present, very, very important. They are attenuated, they seem to be attenuated parasite, but systemically present. And immunologically, if we compare, the VL patient and the CL patient, just a minute I would take. If we compare them, then we see that the major axis that is modulating the disease outcome is the Treg axis, wherein the Treg response is actually compromised in this ACL, such that the TH1722 parasite killing activity will be more in these ACL cases, and can lead to the cutaneous outcome rather than the VL. Specifically, I focused on the leishmaniasis project which is going in our lab, apart from the more interests that are being taken up, because I see in JNK there are recent reports where again the CL cases are there. In Nepal, it's a very, very prominent phenomena, and it has become a part of the national reporting system. So I thought it will inform, the researchers in the field in this area regarding getting into further details of these CL cases. And again, as I was traveling, I could see the Chenab river valley. So possibly, it can give further cues as to whether this phenomena is much more widespread than what we are thinking. And we are actually trying to work towards, based on whole genome sequence analysis, if we can develop a strategy where we can differentiate the VL, ACL, and PKDL causing LD variants to be used in molecular surveillance. So that is the long-term goal, and I have students from the lab presenting posters with much more details. I would request people who are interested to please go through their poster presentations. Sorry for taking a bit of more time.
Thank you. I'm sorry we have no more time for questions, but they can go to your students' posters. Yeah. Thank you.
Thank you.
So with this, we will move towards the next talk of the session. It is by Dr. Madhav Chanda Mohanty. He's Scientist F, ICMR NIV Mumbai. Topic of his talk is immune dysfunction and altered microbial networks defining long COVID in Indian population.
Yeah, good afternoon, everyone. And it's a pleasure to be here at the Immunocon 2026 at the mesmerizing city of Srinagar. And my sincere thanks to Dr. Dil Afroz and the whole entire organizing team for the invitation. I will be presenting some ongoing work on long COVID immune responses in an Indian cohort. So the title of my talk is Immune Dysfunction and Altered Microbial Network Define Long COVID in an Indian Cohort. So as we know, long COVID is a major healthcare challenge globally because of its lasting, continuing conditions, and also because of more than 200 symptoms affecting multiple organs and already affecting millions of people worldwide. So there is no certain diagnostic markers, there is no biomarkers, and there is no treatment facility available so far globally because of the heterogeneous population. And you can see in this picture, this is not working. So you can see the challenges in establishing diagnostic biomarkers for long COVID. There is a massive clinical and phenotypic heterogeneity among this population. There is an overlapping divergent pathophysiological mechanism. There is a dynamic episodic symptoms which is difficult to follow up, invasive sampling and hurdles for key sites. So this heterogeneity actually dilutes the experimental condition, diluting the statistical power, and whenever there is a trend found, that is also can signal some emerging patterns. So even the modest trend can have large scale consequences when millions are affected. So what is long COVID? The definition is signs and symptoms that develop during or after an infection, which is consistent with COVID, continuing for more than 4 to 12 weeks and are not explained by an alternative diagnosis. So there are hypothesized mechanisms of long COVID pathogenesis. It can be by the reactivation of some of the viruses, mainly the herpes viruses. Then there can be impacts of SARS-CoV-2 microbiota, and there can be autoimmunity, which is primed by the immune cells, and microvascular blood clotting or dysfunctional signaling of the brainstem. So we conducted a study in an Indian cohort, as we know that there is a very less awareness of long COVID in India, as there are no systemic study or no long COVID clinics available in India. So it is the first of its kind study in Indian, that's a cross-sectional study with in a multi-centric involving 4 different hospitals, in which we are studying the different parameters, starting from immune dysregulation, innate immune gene expression, antibody levels, T cell response, cell death mechanism, cytokines, and also the effect of the microbiota. So this is our study plan, in which we have taken the confirmed SARS-CoV-2 infected patients, which are having persistent symptoms like fatigue and etc. And there we are comparing these results with the confirmed SARS-CoV-2 infected patients, which are not having any kind of symptoms after SARS-CoV-2 infection. So this is a study cohort, which is, you can see there are more than 100 patients in each category, and you can see that there is no difference between the study population while in the terms of age, gender, and vaccination status. However, you can see there are more number of people who are being hospitalized and had oxygen therapy during their active infection are there in the long COVID patients, and hypertension is significantly more in the cases of patients who are suffering from long COVID. This is a kind of comorbidity which is seen in more percentage in case of long COVID. So after that, if you check the treatment status, more number of participants in the long COVID group have undergone remdesivir and oxygen therapy, most importantly the mechanical ventilation and the nasal cannula. And these are more there in our long COVID participants than the recovered patients. While categorizing them as per the symptoms, you can see the symptoms like respiratory, cardiovascular, and psychiatric are decreasing over time, and we have taken the patients who are infected 1 to 2 years, 2 to 3 years, up to more than 4 years. And if you see the general symptoms, which are comprising of fatigue, weight loss, tiredness, body ache, etc., they are constant or there up to this after more than 4 years also. Incidence of general and ENT symptoms similar across duration, whereas the other symptoms are decreasing. So fatigue is the most reported symptom, followed by cough and anxiety, which is seen in more than 80% of the participants which were included in our study. And if you compare according to the variants, the way they were infected, during the time period when they were infected, we took the data from INSACOG based on their infection date, because we have the evidence of their infection. And then we can see that there is no difference in the variant status of the infection at the time of COVID infection. However, if you'll see the number of symptoms, you can see that there are more than 10 to 20 symptoms seen in case of Omicron affected patients, but then that can be due to bias because Omicron is the recent strain, maybe they are showing the infection, so we cannot conclude on that. So if you check the gene expression status in PBMCs and nasal epithelial cells, we could find there is a very high significant level of TLR7 rebel in long COVID patients, which is other way around actually, and also the type 1 interferons. And if you separate the fatigue cases, we have significantly more TLR7 in fatigue cases. After that, we did the immunophenotyping with TBNKs and all, and we found that T helper cells are significantly more in the patients which are having long COVID, and the frequency is lower in NK cells. It is little bit lower if you take the total cells, total population. If you divide it according to the duration of infection, then you will see that they are up to 4 years. There is an elevation of NK cells, frequency of NK cells in RC, that is recovered, and lower in case of long COVID patients. And if you check the, after that we further check the dysfunction of NK cells if they are at all, and we found that the cytotoxic and cytokine producing NK cells are lower in this NK cell population, and however the receptor, activating receptor, which is like NKG2D and the inhibitory receptor anti-4 NK is more in case of. So then we check the immune exhaustion markers. It is the PD1 level is higher in CD8 population, and interestingly the CTLA4, LAG3 and TIM3, which is the inhibitory molecules markers, exhaustion markers, they are lower in case of this long COVID patients. And if you check age-wise also, you will find the similar trend. However, in CTLA population, you will see that the more than 4 years, it is actually very less after more than 4 years. So while we tested the apoptosis, presence of apoptosis in the PBMCs, we found that the late apoptosis phase is lower in case of, is higher in long COVID and lower in LC. It is up to 2 to 3 years of infection. And serum cytokine sequence, we have covered 30 more cytokines, and from that some are elevated, but the significant ones I am presenting, IP10, eotaxin and IMIP1B are significantly more in case of long COVID. So also the autoantibodies to complement 1Q. Then this part showing that there is a dysregulation because of the activation of type 1 interferons, TLR7, PD1, etc. And then prior to the studies, I will go to the microbiome study, wherein you can see there is an increase in certain microbiota which are pathogenic in nature, and there is a decrease in beneficial microbiota. And although there is no difference in the diversity, you can find the network is different, wherein the LC group had lower vulnerability index, reflecting increased rigidity and lower adaptability to the microbial community. And we found there is a certain pro-inflammatory and autoimmune parameters like IP10, IL-8 and C1q autoantibodies were highly connected with gut microbiome. You can see the association with the heat map, and you will see that here there is a positive correlation in the microbiota with whatever factors we have found significantly more, that is the IP10, MIZ, etc. So in the summary, long COVID is associated not just with alteration of microbial composition, but with a broader reorganization of the gut microbial ecosystem, and that can be targeted for the therapeutic implications. And this is the overall summary of our study, which shows that there is a TLR7 activation both at nasal and PBMCs. Then there is a type 1 interferon effect by increasing netosis in PR3 autoantibodies, C1q, and then is a decreased CTLA4, increased PD1, then NK cell dysfunction, leading to cell death, inflammatory burden, and then dysbiosis also plays a role for the systematic fatigue and the symptoms of long COVID. That's all. Thank you. And this is my long COVID team and the clinical collaborators who are actually collecting the recruiting the patients. And this is Dr. Vikrant Bohr from National Institute of Research in Women's Health, who has contributed a lot for the microbiome.
Thank you very much.
Thank you very much.
We have time for one question from someone in the audience.
Thank you, ma'am, for your interesting presentation. I have one query. As far as your study is concerned, you have shown that the patient who are having multi-comorbidity, they are more prone to develop long COVID. Have you subjected your data to multivariate regression analysis to find out any independent risk factor for developing long COVID?
Yeah, there, as I have shown that comorbidity like hypertension is much more. The data actually is so vast. I mean, you can imagine that there are so many comorbidities and so many factors, symptoms and all. We are actually proposing an AI analysis by machine learning so that we can find out something with this huge data. So this is, is going to be very difficult, but you are seeing multiple panels of cytokines. You have to have a multi-panel biomarker. There is no single marker available, like that comorbidity also. So we are going to do that, and after that we will be seeing that whatever factors are responsible.
Thank you, ma'am. So if there is no question, we will move forward with the next talk of the session. It is by Dr. Dimpu Gogal. He is Scientist C, NHR Debru. His topic is TIM3, respiratory viral infection, a potential immunoregulator.
A hungry man is an angry man. And thank you for your patience. Good news is I will try to be as brief as possible. Okay, so here, respected all, I will be talking about TIM3 in respiratory viral infection, a potential immunoregulator. Now, I won't go to details about immunopathology associated with emerging and re-emerging viral infection, respiratory viral infection like recent COVID-19, which my previous speaker has spoken. So the disease severity is not because of the virus per se, but it's because of the immunopathology, which is mediated by excessive immune response. And virus specific T cells produces lots of cytokines, and also all the nuts and bolts of immune cells get exaggerated, which cause severity of the symptoms. Now, in case of respiratory viral infection, lung injury is the most typical symptoms which causes seriousness in the patient. Now, we have drugs to decrease the severity of the disease, but it comes with their limitation, like drugs like steroid, like glucocorticoid or monoclonal antibodies such as tocilizumab. It reduces the viral load. It causes, it hampers in the reduction of viral load, and it causes undesired side effects, and it's not recommended for the pregnant patients. Now, in my today's presentation, I will be talking about H1N1 and some of the in vitro experiment which we have carried out. So here now H1N1 pandemic was another popular respiratory viral infection, which is also known as swine flu virus. Now, the pig has a biological properties to acquire viruses from human, pig, avian, which makes it as a mixing vessel, and thus it also causes generation of new type of viruses. And respiratory viral infection like H1N1 can bind to alpha-2,3 sialic acid, which are present in upper respiratory tract and lungs, and then it activates the nuts and bolts of immune system, and which causes disease severity. Now, this kind of study are well known in mice model. In human, it's difficult. It's understudied. So this pandemic H1N1 viral infection, now it comes as a sporadic outbreak infection, even in our locality. So what we did was we confirmed the cases by doing real-time PCR, H1N1 positive cases. And in H1N1 cases, the unique property was that this virus causes severity of the symptoms in the children and young adult, not in the older cases. So we took the patients who are between 25 to 36 years old, and then we look at their T cells percentage, and we found that H1N1 patients, they have lesser percentage of total T cells and also CD4 T cells. However, they have increased percentage of V delta 1 T cells. V delta 1 T cells are known to provide mucosal immunity. And we also found that the activation marker, expression of activation marker, CD69 and CD25, they were very high in the T cells from the H1N1 patients, showing that they are activated, although some of the T cells are diminished. And these patients had less symptoms. They were mild cases. So we thought, although there is activation, but the symptoms are very less. I mean, they are the mild. There are patients with mild or moderate symptoms. So we thought, let us check their immune checkpoints, and we check all the 4 different checkpoint inhibitor, TIM3, LEC3, CTLA4, PD1, and we found that TIM3 expression was significantly higher on T cells of these patients. So, and we think that it is possible that this increased expression of TIM3 might be dampening the immunopathology, which could be associated with aggressive T cells, because these patients were with mild symptoms. Now, to check this, now it's difficult to get the patient at the time where you want to prove something. So what we did is we took an in vitro system where A549 cells, they were stimulated with viral mimic, which mimic the TLR3 ligand, poly-IC, and then we stimulated it, and then we use agonist antibody, FC protein against TIM3, CTLA4, and PD1. And then we initially stimulated with poly-IC, then co-culture with PBMC, then agonistic antibody were added and immunophenotype. And we found it, so the y-axis is HLA-DR, late activation marker, and x-axis is TIM3 and y-axis is HLA-DR. Now for these 3 different subsets, CD4, CD8, gamma delta T cells, we use this agonist antibody alone or in combination. And not to confuse you, wherever TIM3 is there, you can see there is a increased percentage of double positive T cells and also TIM3 positive T cells. And we found that in all these T cells like CD4, CD8, and gamma delta T cells, when we are adding the combination of TIM3 and CTLA4, the single positive HLA-DR are getting significantly decreased, and the single positive TIM3 cells were significantly increased in CD4 and gamma delta T cells. So this is what we got now. So it could now again it's possible that this TIM3 might be kind of a kind of novel mechanism by which we people can utilize it for reducing the immunopathology which occurs during respiratory viral infection. And this is what we think, and this is work in progress. And this is picture is from our lab. This is like this has both my lab members and my family members. We go for regular outing, and we get ample chance to take group picture. And the towards your left hand side, JP, now he is continuing this work. With this, thank you.
Great, thanks very much for keeping to time. Questions from the audience? Any comments, questions? I have one comment, I guess. It's not really a question, but how big was your flow panel? How big was your flow panel, your flow cytometry panel?
Yeah. So thanks for asking this. We have also looked for perforin.
No, but how big? How big?
How big meaning?
How many markers?
Yeah. So total, I will just read whichever I remember. So all the T cell subsets, and then we had activation marker HLA-DR CD25 as well, and also for the exhaustion marker, as well as. So this was for one panel. Another panel, I have not shown the data. We have also done for granzyme B as well as intracellular cytokines. So that was second panel. So that data I'm not showing it here.
And TIM3 was in both panels?
Yeah, TIM3 was.
So I would comment that you need to do some cluster analysis of your data, which may yield more information that you have here.
Yes.
More insight.
Yes.
Thank you for your positive feedback, and we are planning to do that. We are in a process of getting the flows of software. We don't have.
Good.
Thank you.
Anything further? Over here.
Yeah. So
There's one
yes.
over here.
Please go.
Microphone, please.
No, but we are planning for it. We are planning, not yet done. This is the kind of very preliminary data we are planning for. Yeah.
Because influenza, there are more studies in vivo model.
Yeah, that's
as compared to the in vitro.
Exactly. So that is the plus side for us. In vivo model there are many. For human thing, it was less study are known. So that is the plus point for us, that we could show the human data. Nevertheless, with the TIM3 study, we would like to continue with the in vivo model also. We would like to go for bronchioalveolar lavage and lung tissue also.
As you were saying that you were not getting the exact time the patient you would like to evaluate.
Yes.
So in vivo model, that is upon your choice.
Yes,
you can do that.
Yeah. Thank you. We will be doing it. It's there in our back of mind. Thank you.
I'm interested in influenza because I have done my PhD on influenza.
Oh, is it?
Yeah.
We will talk again. We'll talk.
Sure.
Thank you.
Great. Okay, thank you very much.
Thank you so much.
And I think it's now lunchtime.
Yes. So thank you so much. With this, we come to the end of this pre-lunch session. A quick felicitation ceremony for all the speakers because they've been waiting for so long. May I request our worthy chair to come forward? May I request Professor Clive Gray to felicitate Dr. Dimpu Gogoi? Dimpu Gogoi. It's there. It's there, man. They're like sleeping. Thank you. I now request Professor Clive Gray to felicitate Dr. Daya Karalti. Daya Karalti. I request Professor Clive Gray to felicitate Dr. Ruqayya Nazir. Thank you. I request Dr. Nazia Mahfouz to felicitate Dr. Manju Jain. Thank you, ma'am. I request Dr. Nazia Mahfouz to felicitate Dr. Madhuchanda Mohanty. Thank you so much. Thank you all. We now proceed for a quick lunch session. We will resume after half an hour, 30 minutes, for 3 more talks from eminent scientists. They are continuing as a chair post lunch. We will do it there. We are not leaving you. Please proceed for the lunch. We are coming back at 3 o'clock sharp. Thank you.
We'd like to thank the organizers for the invite. My lab at IIT Madras works largely on the infection biology of the pathogens, where we are trying to understand the microbial entities and strategies leading to disease pathogenesis. With tuberculosis as a model system, disease caused by Mycobacterium tuberculosis. Apart from the very, very well-known clinical scenario, it's not just the pulmonary form of the disease, but it is also causing several extrapulmonary forms. And beyond the clinical spectrum of the disease, it's a very, very interesting intracellular bacteria to chase at a very fundamental biology level as well. And the immune response to Mycobacterium starts with Mycobacteria and its components interacting with the innate immune cells, which in turn send the message to the cells of the adaptive immune system to initiate adaptive immune responses depending on the cytokine milieu. So we are particularly interested in characterizing what these components are in different forms. And if you look at the mycobacterial cell wall cross-section, and you'll see that it's very, very enriched with very different antigenic molecule. We are particularly interested in this particular molecule that is enriched at the periphery of the Mycobacterium. It's a glycolipid called mannose lipoarabinomannan, which I'll be referring henceforth as MANLAM. And MANLAM is a very abundantly present antigen in the periphery of the mycobacteria, as I mentioned, and very unique feature of the pathogenic mycobacterial species alone, and shown to be a ligand for DC-sign and mannose receptor, and is also secreted from the infected cells. While some roles of MANLAM is known with respect to the inhibition of phagosome, inhibition of phagolysosome formation in macrophages, but its effect on the inflammatory landscape is not known. That's precisely what we are pitching our work on. And the first set of cells that we were wanting to address with respect to inflammatory landscape characterization of MANLAM are B cells. So B cells are prominent cell type in the TB granuloma, and they are not just antibody producing cells, but they secrete a lot of cytokines, chemokines. They are antigen presenting cells and therefore have a very profound influence on the immune response outcome to infection. And B cells have been shown to be steadily increasing in the granuloma post-infection in humans and in as well as in different mice model and guinea pig models, including the recent literature suggests B cells have a very enigmatic roles in TB, which is yet to be understood. So that's precisely where we were pitching our question to. And when we checked the effect of MANLAM on human B cells, CD19 positive total B cells, we saw that human B cells proliferate in response to stimulation by MANLAM. And MANLAM induced B cell proliferation seemed to be mediated through TLR2, because on blocking TLR2, we saw the B cell proliferation going down even in the presence of MANLAM. So next we checked the modulation of the MAP kinase pathways in B cells induced by MANLAM, and we saw that MANLAM differentially modulates MAP kinases, where we saw early activation of ERK, we saw inhibition of P38 MAP kinase pathway, which is important for inflammatory, pro-inflammatory responses. And very interestingly, we could see that the MANLAM-mediated downregulation of P38 MAPK could be rescued on blocking TLR2, meaning confirming the role of TLR2 in MANLAM in mediated modulation of B cell functions. Further, we checked a cytokine's profile on B cells on Mandlam induction. We saw that there is a prominent IL-8 and IL-6 upregulated, and very, very marginal increase in the other cytokines. And the Mandlam educated B cells in co-culture with T cells induced a very, very good T cell proliferation. And the T cell cytokine profiles looked interesting with respect to enhanced IL-4, IL-5, and enhanced IL-10 with reduction, downregulation of interferon gamma, suggesting Mandlam-educated B cells polarized T cells towards Th2. The next cellular cell type that we wanted to check with respect to the action of Mandlam was on dendritic cells, and CD14 positive dendritic cells differentiated in vitro. And when we added Mandlam to the cultures, we could see that Mandlam induces dendritic cell maturation with respect to upregulation of the maturation markers. And Mandlam induced NF-kappa B activation in dendritic cells, and interestingly, blocking DC sign downregulated some of the maturation markers. So, and further, on checking the inflammatory profiles, Mandlam did induce IL-8, IL-6 indices, but then IL-12, which is a sentinel pro-inflammatory cytokine, was not induced. And with respect to the chemokine profiles, we saw CCL17 and CCL22 upregulated, which are very important for Treg and Th2 recruitment. And also we saw the CCL19 downregulated, which is very important for Th1 recruitment to the site. On checking the T cell polarization of the Mandlam-educated DCs, we could see that there was a very profound IL-4, IL-5, and downregulation of interferon gamma and IL-10 induction, and very, very indicating a Th2 polarization. And very interestingly, the Mandlam-educated dendritic cells also induced CD25 positive, FOXP3 positive Tregs, and also there was a CD274 upregulation in the DC post-Mandlam treatment, which was downregulated post-blocking CD274 antibody. So suggesting Mandlam-educated disease promote Treg expansion as well. So what we had tried to show in our work is that Mandlam shredded from the infected cells and as well from the bacteria could work on D cells, DCs and B cells, and promote Treg Th2 via axis, and which in turn blocks Th1 response. So using DC and B cells as a niche, Mandlam chokes the very protective immune response required for TB infection protection. So be it active TB or be it risk of reactivation in TB, both are associated with imbalance in the inflammatory landscape. It is not just about the immune response induced, it's about what kind of immune response is induced and when. So with respect to that, we see that Mandlam has a role in driving inflammatory imbalance in TB. And very interestingly, Mandlam is not just present on the cell membrane, but is also shredded into the bodily fluids. We are in an engineering institute, so we kind of collaborated with biomedical engineering people where we could develop biosensors for sensitive detection of Mandlam in the bodily fluids. So in TB diagnosis, the host-based biomarker does suffer with the differences in the host responses, but the bacterial-based biomarker suffers with, though it is very specific, suffers with sensitivity with respect to limits of detection being really low. So we developed in collaboration work, of course, a biosensor, a sandwich assay-based biosensor, which could detect Mandlam in urine up until 5 femtograms per ml. Of course, this has been done in PBS and in synthetic urine samples and spiked urine samples. Of course, we have not yet checked it on the patient samples. So the take-home being a single glycolipid of MTB acting as a molecular rheostat, working across different pattern recognition receptors, tuning the cells and can paralyze both myeloid and lymphoid axis, mediated protective immune responses. Therefore, MTB, a very, very, very fascinating bacteria, does not simply hide from the immune system, but instead proactively rewires the immune synapse for its own advantage. So this is the team that I work with in IIT Madras, and this is our campus, which we share with the ones that you see on the photo. So if any of you are in Chennai, you are welcome to visit us. We'll be glad to host you. Thank you so much.
Yeah, we can have questions, please.
One of the early cytokines actually. So it differs, so the time point differs with respect to the concentration of antigen, the type of cells that you are looking at, and the ratio of the cells that you are looking at, etc. And of course in vitro assays. But it's a notion that the pro-inflammatories come much early on actually. So it's early time point cytokines, yeah. Right.
May I ask one question, please? Here.
Yes.
You talked about that man-lam detection. Why do you want to do that? Why can't we actually detect antibodies against the man-lam, which is more sensitive?
That's a good question. The antibodies have been detected in patients, of course, against man-lam, that being a carbohydrate again. So one thing that we wanted to also do was that check the ratio of free man-lam versus antibodies against man-lam, not just go with the antibody titer because it is sequestering of man-lam, right? So and that might therefore the readout might vary in different patients. So we wanted to kind of do a paired analysis of the free man-lam in the samples versus the anti-man-lam titer as well.
So is there any concern about the specificity of man-lam, that it is specific to mycobacteria?
One of the concerns is that the Manos cap numbers differ in different pathogenic species, and therefore probably it might differ in the clinical strains as well. Yeah, that is one of the.
So wouldn't it be better to actually check whether it is specific for MTB?
It is specific. The Manos capped liporabinamenon is specific. Yeah. Otherwise, the other species have pilam, aralam kind of stuff. Other cappings. Yeah.
Okay. So do you have any more questions?
Yeah, nice talk, Vani.
Thanks.
It was a somewhat similar question because is man-lam specific to this MTB?
Right.
And do you know what difference is there with versus other pathogens? Because man-lam is present in other pathogens as well.
The lam is present in other pathogens, but the Manos capping is present only in pathogenic species.
Only in MTB?
Pathogenic species.
Pathogenic mycobacterium only?
Pathogenic mycobacterium only,
okay,
as far as I know. Yeah. Thanks. Thank you.
Okay. So with this, we'll move towards our next talk. It will be delivered by Dr. Sunil K. Raghav. He is a senior scientist of Institute of Life Sciences, ILS, Odisha. His topic is single cell immunogenomics, identify NCOR1 as a potential prognostic marker for TB therapy response.
So very good afternoon. Thanks, Chair, for introduction, and thanks to the organizers, Dr. Dil, for inviting me and then giving this opportunity to discuss our work that we are doing. So the work that I'm going to present is completely unpublished, and we are doing it, and it's quite interesting, so I would like to share it, and that if somebody wants to give feedback, it would be most welcome. So my group at ILS works on most host pathogen interaction, and we use immunogenomics and systems biology approach to try to understand how the immune responses, they are finely fine-tuned at the transcriptional level. So we do high throughput immune profiling of immune cells in clinical settings. Okay. Yeah, so today I'm going to talk about an interesting protein that my group is working on almost like a decade now. And we started working on dendritic cells on this protein, and this protein is known this is a nuclear core repressor protein, And these are two homologous proteins, ANCO1 and SMART. It is known as ANCO2 also, this SMART. And these are global transcriptional co-repressors. So when we started, we were looking into the function of them and how they really regulate the immune responses. So just giving you an introduction that these nuclear receptor co-repressors, they form complex with nuclear receptors and some other transcription factors, and they form co-repressosome complex. Okay. And this co-repressosome complex basically leads to recruitment of histone deacetylases, and that represses the target genes. The interesting thing is, these co-repressors, they keep the genes primed. So they are not basically completely repressed chromatin. So I will show you here, because we and others showed that this co-repressor is present on almost 60 to 70% of PU1 bound regions on these dendritic cells as well as macrophages. So what happens when any activation signal comes, this co-repressosome complex is degraded, and because of activating signals like activating transcription factors or ligands, the genes get activated. And this is a cyclic process. When the ligand is off, activation goes down, then again the genes get repressed. And we know that that is a very important feedback regulation. So what we found here in dendritic cells and in macrophages also, we know that. So if you see that when we do chip sequencing for ANCO1, we see almost like a 56% of these regions are bound by this ANCO1. And PU1, we know this is a pioneering factor for myeloid cells, macrophages, dendritic cells and all. And when you activate these cells just by CPG, like TLR9, you can see a lot of these regions bound by activating transcription factors like NF-kappa B, AP1 factors and all, at the same regions. And this is very activation specific. Like if you activate with CPG, you will get like inflammatory response. If you will activate with TLR3 ligand like PIC, poly-IC, you will get antiviral responses. So the ANCO1 moves from those regions and then those genes are activated. So, and these proteins were, co-repressors were known only in the muscle cells, fat cells and all, and they were known to regulate metabolism, mostly oxidative phosphorylation and fatty acid biosynthesis. So when we, in ANCO1 knockout mice, what we observed that when you deplete this co-repressor protein, you see that instead of making inflammatory Th1 cells upon any challenge, they had a lot of Tregs. So removing ANCO1 or depleting ANCO1 leads to activation of IL-10, CD83, IDO1, all these immunosuppressive markers, and the balance perturbs toward more of regulatory. Okay. And then Kaushik, one of the PhD students who looked into the metabolism, because the idea was to target it, and what we found that ANCO1 depleted cells have increased glycolysis and fatty acid oxidation, and if you use synthetic inhibitors, you can perturb this Th1 Treg balance in vivo as well in mice, and you can control that, fine-tune it. We did a lot of epigenomics. I will not go into those details to look into where these ANCO1 bind, how the TLR9 activation is regulated, TLR3 activation is regulated, and where the more regulation is happening, fine balance. Interestingly, in the same animals, when you give infection with Mycobacterium tuberculosis, we found that these animals have very high load, and this is in cell, but in animals as well. In macrophages, if you see that there is heavy load of Mycobacterium. And when you overexpress this protein in the same cells again, you can rescue it back. And I'm not going into details of this because interesting thing is coming later on after this, follow-up of that. So what we identified that this protein is a very important sensor of this AMPK ATP responses. So what happens when you deplete ANCO1? Glycolysis in OXPHOS goes so high, ATP is so high in the cells that lead to decrease in AMPK. And ultimately autophagy is completely suppressed. So there is no autophagosome maturation and this lysosome biosynthesis. It's drastically reduced and the Mycobacterium survives very easily in these macrophages. Interestingly, we are looking into now this angle, how this happens. So when the NPB infects, how the NCO1 is kinetics is regulated and the ATP changes happen, what are the metabolic pathways that are regulated? So that we are looking into, but interestingly, side by side, when we look into the patients, we found that during the treatment, during acute infection, this protein goes down significantly. And then upon treatment for 12 months, this protein comes back to the normal. So it shows that when the load is high, this protein is low, and there is some correlation. And when the treatment happens, mycobacterium dies, the protein comes back to the normal. Okay, so it looks like somehow this NCO1 turnover controls MTB clearance in macrophages and hopefully in tuberculosis patients. So what we did a fine kinetics to really look at how the NCO1 is regulated in these animals and alveolar macrophages. So you see here MTB CFU in the lungs. Day 7, day 10, you see sudden increase, sharp rise in the CFU. And then day 17 is maximum, then it goes down, and then it becomes constant. So we did till day 90, and we see that the bacteria survives like that. More like latent, but in C57 black 6 mice, latent infection is not well defined. But still we see till day 90, it survives, mice doesn't die. If you look at the NCO1 expression now in the same scenario, this is uninfected one, okay. Initially it goes up at day 7, but then it starts dropping, okay. It goes down and then becomes like a minimum at day 21, and day 43 it starts a little bit up. So it follows exactly in the same pattern, and if you look at the correlation, regression basically, is minus 0.84. So basically, it shows that the level of MTB inside the macrophages somehow is correlating with the NCO1 level in those cells. So this was very interesting. So now the questions are, how this NCO1 dynamics during MTB infection is regulated, which upstream signal is sensed by NCO1 during infection that leads to these changes. Either this is metabolic thing or mycobacteria is secreting some metabolites that is leading to this. Do we see similar NCO1 kinetics in TB patients? Because we wanted to do parallel things along with the mechanism. So we started follow-up of patients also. But before that, I will just show you some mechanism, what is happening during early kinetics. So what we did fine kinetics at early time points only, like day 0, day 3, day 7, day 10. We infected animals with 100 CFU, and we did single cell transcriptome of alveolar macrophages at these time points. Bell fluid basically, bell cells, not exactly alveolar macrophages, because there are some other cells also. So we took all the cells from the bronchial alveolar lavage. Now you see that in NCO1 knockout animals, you see there is more infiltration from the early time point, from day 3 itself. You see there is more infiltration in the lungs. Okay, and there is always higher CFU in knockout animals. So the bacteria survives easily, that I showed before as well. Now you look at these, these are all the cells, okay. So based on this single cell R labels, you define that which cells are what. So you see that maximum are macrophages, that is expected because this is a bile fluid. But you see a lot of infiltrating cells are coming. So this is from all the days, okay, day 3, uninfected, all the days are there. Now when you segregate this and look into how is the proportion of cells in control mice at different days compared to uninfected and these knockout animals. So you see clearly here at day 7, there is strong infiltration of neutrophils and these inflammatory cells inside the bile fluid from the lungs, okay. And the load is high, it is expected, there will be more infiltration. But what is happening in these macrophages, okay. Now interesting, now you look at these metabolic changes that are happening. So you see day 3, day 7, there is some increase in these metabolic, and 0 is basically the uninfected, okay, basal state. So when you see the glycolysis, fatty acid oxidation, beta oxidation, you see all of them shut down, day 7 to day 10. It's almost like they're less than basal level. Glycolysis, still you see there is a little bit high, but all other metabolic pathways are almost shut down during this time point. Except, interestingly, you see that phosphagen system, creatinine kinase and all, I will take still like 2-3 minutes. So the phosphagen system, which is basically the ATP that is generated from phosphocreatinine and the muscle, that goes up very high here. And this is only the control mice I am showing. In knockout mice, the metabolism is completely different. I will not go into much details. You see the NCO1, it basically goes a little bit high and then drops down suddenly at day 10. And these are the gene clusters, which are basically specific to knockout, specific to your wild type animals, and only during the infection. Now we are looking into these details. I will not go into much details here. Interestingly, what we see here is the NCO1 knockout animals have more module-like features of M2 macrophages as compared to M1. And they have more features of foamy macrophages, more score module of foamy macrophages, as these genes are highly expressed in this. Now we are trying to understand the kinetics, but I will not. So I will just show you the human patient data now, that we just tried to follow this NCO1 in the human patients during the treatment cohort. Okay. So what we do, and I will not show you the blood data here, only the sputum macrophages. So what we do, we collect at before treatment and after treatment, month 1, month 2, month 4, month 6, patients, follow up exactly same patients, and try to see what is the change happening in sputum macrophages. Now you see here, this is the chest X-ray score of the patients. You see that this is, because the treatment is happening, you will see that they are responding to the treatment. Interestingly, you see here, the NCO1 behaves in the same way. When the treatment is working, the NCO1 is increasing with time. And at the same time, you see that IFN gamma IL-10 cytokine is known to really tell about whether the homeostasis is established or not. You see that at day 0, IFN gamma is so high, and then it goes down because IL-10 starts increasing. Okay. If you look at the ROC value here, you see that NCO1 can predict IFN gamma IL-10 cytokine ratios in the patients around 75%. Because there is no marker yet that can really tell how much IFN gamma IL-10 ratio will be there. So but just by looking at NCO1 in the macrophages by flow cytometry, you can tell how is the response of IFN gamma IL-10. And at the same time, how the chest X-ray response is there. Just by looking at NCO1. Though it's not 90% or something, but in case of MTB, just by one marker, reaching around 80% itself is a good thing to see. Because at least you can know whether, and here we have not divided responder versus non-responder, because the patient size is still 30. So we need to really increase it, and then we can divide and see that maybe in responders, the NCO1 is behaving properly, and non-responders are giving a background here. Just a conclusion, final slide. So I showed here that NCO1 expression is dynamically regulated during MTB infection, and the level of NCO1 protein is inversely correlated with MTB load in macrophages. By single cell transcriptomics, we clearly see that there is a change during in these metabolic pathways, or metabolic rewiring happens. Now we want to look into how this MTB is really triggering that. That's a very difficult question, but we are trying to find out. And NCO1 level in sputum macrophages appears to have a strong prediction for IFN gamma IL-10 ratio as well as chest X-ray score. So this is the team. Students are here. Subhashis and Divya, they presented their posters as well. Some of you might have interacted with them. So Divya is working on this human follow-up patients and all. And I didn't show the data. We followed exactly the treatment in mice also, because there you can control it properly. And we see there much more strong prediction. It's almost like 90%, 85 to 90%, because there the variability is not there. Subhashis did the initial, this early kinetics and single cell transcriptomics. And Divyanshu is the person who analyzes the single cell data, looking into those mechanisms. So these are the funding agencies from where we have the funds. And these are the clinical corroborators who provide these samples, and we work with them. And the patients that really give their consent for the samples. Thank you. And sorry for the delay for.
It's okay. So do we have any questions from the audience? Okay. So we have time for just one question, please. Just one question.
Yes.
You please.
Very interesting finding. I have a question regarding your neutrophil data. So NCOR 1 is going down, neutrophils are coming up. What do you think why the neutrophils are only mobilizing, or majority of them are mobilizing than others?
It's known that in MTB infection, neutrophils infiltrate in the lungs. That is a well-known phenomenon.
No,
but here, in NCOR depletion condition, I will tell you interesting thing, because what we observed that in NCOR depleted cells, it's not like inflammation doesn't happen. There is IL-12, these inflammatory cytokines go up, but on the other side, IL-10 also goes up. So maybe the balance, inflammatory cytokines or chemokines are there, they are recruiting it, but the IL-10 is creating a...
but what brings the neutrophil more?
I don't know.
Yeah, okay.
So I can't, maybe we will look into neutrophils separately, the gene expression and all. It might give some idea. Thank you. It's an interesting question.
So, okay. I think we'll end this session.
Okay, thank you so much. With this, we come to the end of this post-lunch session. A brief felicitation. May I request Professor Clive Gray to felicitate our speaker, Dr. Sunil Raghav. Please. Thank you, sir. Thank you, sir. May I request Dr. Nazia Mahfouz to felicitate Dr. Vani Janakiraman. I now request Dr. Nazia Mahfouz to felicitate Professor Clive Gray. Yeah, it was done. Okay, so yeah. Thank you. I now request Professor Clive to felicitate Dr. Nazia Mahfouz. Thank you so much. With this, we come to the end of this session. We have an important announcement for you. We'll be here at 4:30 p.m. for an online talk by Professor Vijay Kuchru from the Harvard School. So I request you all to be present in the hall by 4:30. For the moment, we disperse for the poster presentations which are being held upstairs. Students need your guidance, your input, so please kindly make it convenient to visit them. Thank you so much.
IL-17 is one of the very important subset and the cytokine which is actually play a major role in all organ-specific autoimmune diseases. And neutralizing IL-17 is kind of shown very nice therapeutic in the therapy in the psoriasis and many more autoimmune diseases. The other thing, after this, during this time of T-cell differentiation, his lab continuously keep producing new subset. And I remember I was, one of the author in the paper where we discovered the Th9 cells as the fourth subset of the T-cell subset. Then the TR1 cells, how IL-27 can induce IL-10 producing FOXP3 negative T cells, which is important for maintaining the immune tolerance. So I can keep going on and on about his discovery and the work he has done, and he has opened up the new field. And not only opening up the new field, he was involved in start of many companies. CoStim is one of the startup he has done. Then he's in the board of many companies like Biocon, Syngene, and many more Indian companies. So here his involvement in the field of immunology, taking from one horizon to the other horizon, and making the name of Kashmir worldwide. People take this pride that he's from Kashmir, and he has done pioneer work. And as a president of Indian Immunology Society, it is like I'm so fortunate that I am actually chairing his session, and my EC has decided to give him the Lifetime Achievement Award to Professor Vijay Kuchru. We have his citation and the medal, which I think I have to post to him, or he will be coming to Delhi. Then I will hand over to this citation and the medal to Professor Vijay Kuchru. So I will not waste more time, and then I think I will hand it over to Professor Kuchru.
Molecule that's on the surface of Th1 cells. We immunize rats with a Th1 clone, generated about 20,000 monoclonal antibodies, and identified 4 different monoclonal antibodies.
His research focuses on T-cell biology, cancer immunology, infectious diseases, vaccine development, and translational immune therapeutics. Through his pioneering work, he has significantly advanced our understanding of immune regulation and the development of innovative immunotherapeutic strategies for cancer and chronic diseases. Professor SKICC has authored over 242 research publications in leading international journals and has contributed to approximately 108 patents and translational innovations in immunology and vaccine science. He'll speak on tumor microenvironment remodeling of cold murine tumors with prime target neoantigen vaccination. Professor SKICC, we are delighted to welcome you to Immunocon 2026.
Thank you very much for your introduction. And I'd like to thank Dear Lafrose and the rest of the organizers for inviting me to the conference here today. It's a wonderful conference, Immunocon 2026, and it's also a wonderful place to host this meeting. So keep going like this, guys. You always have wonderful meetings. What I'm going to talk to you about today is about using a vaccine strategy to elicit immunity in tumors which are very hard to treat. And I like to stress that because if you go into the major articles like Nature Science and you look at immunotherapies, 90% of the literature uses tumors which are quite immunogenic. So CT26, MC38, or tumors that express OVA. And these are relatively easy to treat tumors. But when you get to trying to treat tumors like mesothelioma or pancreatic cancer, those are very, very tough to treat. They're very immunosuppressive. So we started with the premise of trying to make a vaccine, a neoantigen vaccine, that targets the mutations in the tumors, like pancreatic cancer or mesothelioma, and can we make a vaccine that's potent enough for these type of tumors? And what I'll present today is about how this strategy that we have, where we prime mice with a vaccine by subcutaneously vaccinating, peripheral immunity, and then when we follow this with intra-tumor delivery of antigen, and this is very important, the neoantigen in the tumor, this is very important for us to start seeing effects. And this technique, we called it prime target vaccination. We prime the immunity, and then we target the immunity to the tumor. So if we look at the immunity cycle, or the immunity that we would like to be seeing in tumors, right, tumor antigens are released when cancer cells die. The antigen is taken by dendritic cells to the lymph nodes. You prime the immune system there. You get T cells that will recognize these mutations, if you want, in the tumor, and they circulate. And of course, then they go back to the tumor and they can exit and overcome T cell dysfunction or suppression, and actually get more killing. This is the ideal setting that you would like to have when you reject the tumor. Unfortunately, in the real world, and especially in the hard-to-treat tumors, this cycle is disrupted. So one idea has always been, well, if we can vaccinate, we can stimulate these type of T cells that we want. I don't know if we can see my pointer here. Nope. Sorry. So if we can use a vaccine, we can prime the first half of the cycle, if you want, induce lots of T cells, and then hopefully by priming the immune system, we can get these T cells to go into the tumor. Now that sounds like a great idea. We can, rev up this cycle by vaccination. The problem is we've had more than 100 clinical trials in tumors. Half of them have been done with peptides that target neoantigens. About a third of all the trials has been done using an adjuvant called poly-ICLC, which is an attenuated form of poly-IC. It's a TLR3 agonist. These have, even though we've done so many clinical trials, most of these have failed. And why have they been failing? Well, the major reason is basically if you try and look in the blood of the patients that have been vaccinated, you find very small, minuscule responses often. The ex vivo responses for CD4 T cells is less than 1%. Often you cannot detect the CD8 T cells ex vivo. And often what's reported is, which is, I think, is a misnomer, is in vitro stimulated cells, which really means in vitro expanded T cells after 14 days of culture, and then you can start measuring these. And there has been no clinical benefit to date with a lot of those peptide vaccinations. We are seeing some first, maybe you want glimpses of hope with some of the messenger RNA vaccines. But again, the cautiousness here is a lot of the trials that are being done are not matched trials with proper control arms. And that's a very important one, because what we're doing in a lot of these vaccine trials is we're throwing everything against the patients: surgery, radiotherapy, mRNA vaccines, anti-PD-1, and there is never a control group in all of the studies that we see published. So we need, more studies to prove these, and the future will hopefully show that some of the messenger RNA vaccines can actually deliver the goods. So why are these vaccines that target neoantigens fail? Well, there's a number of reasons. One is the vaccine immunogenicity itself, not picking the right neoantigens, the right mutations to target, right? The second important issue, which has been a real problem in the field, is we've been using adjuvants which are good for B cell responses mostly, but not very good adjuvants for T cells, except for TLR3, which has been used in the last 5-10 years. Previous vaccination studies were all done with things like montanide, for example, which isn't really a good T cell adjuvant. So we need better adjuvants. But then there's the issues of how do you get those T cells inside the tumor, right? And how do you prevent these T cells getting exhausted? If they keep seeing antigen in the tumor, they will get exhausted and then they become dysfunctional. And then finally, the tumor microenvironment will suppress these tumors. So these are all obstacles, if you want, to overcome if you want to have a vaccine that will work against the tumor. So our goal was to make a neopeptide vaccine. We call it neopeptides. We use 20 amino acid peptides that have inside them the mutation, the difference in the single amino acid that the tumor carries and not the healthy cells. And we wanted to use this type of vaccine strategy in, as I said, cold immunosuppressive tumors. And we've been working with a mesothelioma mouse tumor and also a pancreatic cancer. And classically what we did is there's a number of ways that you can identify the neoantigens. I'm not going to go into those details. We chose to do whole exome sequencing and RNA sequencing. And by doing so in the tumor and in healthy tissues, you can identify the mutations, you can identify whether they're expressed, and you can select the mutations that you think will be immunogenic. We make 20 amino acid peptides. We call them neopeptides. And then what we do is we functionally validate those. So what we do is we make a bunch of neoepitopes, and then we immunize mice. And we select the ones that are immunogenic. So we want verified immunogenic peptides to use in our vaccines. And we spent a few years testing a bunch of different adjuvants, and we came up with this combination of a TLR9 and a STING agonist combination, which gives us very potent T cell responses. And this is just showing you here that when we compare it to what's considered the best-in-class T cell adjuvant poly-IC, if we immunize subcutaneously 3 times a mouse, we harvest the spleen 1 week later. You can see here in red are the T cell responses that we get when we use our combination of adjuvants with our neopeptides. They're about 10 times higher in an ELISpot assay. And we look at the CD4s and the CD8s, they're from 5 to 10 times higher in the periphery of the animals. This is for the B16 melanoma neoepitopes. I'm just showing you here that these responses are very strong also when we use epitopes, neopeptides for mesothelioma. So very good, potent T cell responses, and much better than what poly-IC itself gives. But the disappointment here was, even though we get very strong T cell responses in the periphery of the animals we vaccinate, so we did a great job making a very strong vaccine, when we gave the tumor at the same time when we start the vaccination, see if I can get. No, sorry. Well, when we give the tumor at the start, and then we start vaccinating on the other flank, the mouse. We do 3 or 4 vaccinations subcutaneous. I showed you the very strong T cell responses, but as you can see here, this is the mesothelioma. We get absolutely no protection. No protection in tumor growth, no protection, no survival advantage. So if we go back to this cycle of 7 steps that we had for the tumor cancer cycle, we've managed, by priming with a very strong combination of adjuvants, to get very strong T cell responses. And we can find these responses in the spleen and in the blood, so we can generate the T cell response and we can get it circulating in the blood of the animal. But somehow we're not closing that circle, the second half of the circle. So we're not getting any benefit in terms of controlling the tumor. So we went back to the drawing board and we said, what are we doing wrong here? What can be happening? Well, the first thing is, of course, if you can look at step 5, is can we get the T cells into the tumor? The first step that we need, obviously, is to get the tumors from the blood into the tumors. And so we vaccinated animals, we gave them the tumor, vaccinated them, and then 7 days after the last vaccination, when we have very robust T cell responses in the blood, we took the tumors out and digested them. And we looked inside the tumors in animals that were sub-Q injected with saline or sub-Q injected with a vaccine. We compared the T cells inside the tumor, and you can see here on the left dot plots, you can see that basically there's no difference in T cell numbers. And if we actually look at the neopeptide-specific T cells, we can do intracellular cytokine stains, and we can count the number of T cells that recognize the vaccine. We see very, very, very few cells inside the tumor. So we have lots of T cells that recognize a vaccine, the tumor in the blood, but they don't seem to be getting inside the tumor. So this led us kind of back to the drawing board. We started thinking about this, and we thought of an old experiment that we had done a few years earlier. So a few years back, we had asked the question, do effector CD8 T cells require antigen and co-stimulation in order for them to expand? And we'd asked this question back then because when you take mice and you infect them with flu, 5 days later, you find 500,000 dendritic cells inside the lungs of these mice, right? And what are they doing there, right? Theoretically, if the dendritic cells were important only for starting the immune response, why would they be accumulating inside the lungs of an infected animal? So we suspected maybe those dendritic cells were doing much more and they were delivering signals and maybe presenting antigen. So what we did is we took a mouse, which is a Thi 1.2 mouse, and we transferred in it naive Thi 1.1 OT1 cells. And the OT1 cells are transgenic. They recognize the ovapeptide synthecal. And using Thi 1.1 and 1.2, we can distinguish the donor cells from the host cells. And then we infected these animals with a flu virus that expresses the peptide for ova, the synthecal peptide. That's the peptide OT1s recognize. And when you infect mice actually with flu, the peak of the immune response is usually 9 to 10 days, right? So what we did is we took the lungs at day 8. That's the time point when the CD8 T cell response starts to rapidly take off and expand in the lung of the animal, right? We digested, we isolated the donor OT1 cells, and we transferred them into a new mouse that had been infected a few days earlier. So what we did is we took a new Thi 1.2 mouse. We transferred those effector cells. They're from a day 8 lung, infected lung, right? OT1 cells. And we gave them to these mice on day 5. And then we harvested the lungs at day 10 to see what happened to those effector cells when we put them in a new infected mouse. And we did this trick. We infected the mice some mice got infected with a PR8 flu virus, which is the flu virus that doesn't have the ova peptide, and some mice got infected with a WSN ova virus, which is the flu virus that has the ova peptide. The first thing you'll notice on the bottom left is that if you transfer the day 8 effector OT1 cells, isolated from the lung, into mice that are not infected, these T cells, these are the red color animals there, each dot is an animal, they just don't expand, right? So you need to have an infection going on. Now, that doesn't tell us that that's antigen, right? If you transfer them into a mouse that has been infected with WSN ova, expressing the ova peptide, you see there's a huge expansion in the lung of these animals. So the question, of course, there is, well, WSN ova does two things. One is it's giving antigen, but it's also causing inflammation. So maybe it was one or the other. Well, we did the experiment also in transferring these cells into a mouse that was infected with PR8 flu. And as you can see here, when we do that, these effector cells don't expand. They only expand when we have WSN ova, when we have flu infection with the peptide. So what tells us is that these effector cells, these day 8 effector cells, need to have antigen in the lung in order for those effector cells to keep expanding. So that was, if you want, an irrelevant experiment we did years ago, asking a different question, which got us thinking what should we be doing here with our tumors and getting the tumor T cell response to, let's say, close this cycle that we are interested in closing to get some efficacy. And we asked the question, well, we know that these neoantigens, these neopeptides, are very rare actually inside the tumors. If you do, and we've done this, if you do immunopeptidomics and you pull down the MHC class 1 to look for peptides that are mutant peptides in the tumor, you will get 300 peptides, which are all self-peptides, and if you're lucky, you'll get one peptide that's a neoantigen. That's how rare they are on the surface, right? But remember, you only need one peptide on the surface to kill a cell. So we thought maybe if we change this stoichiometry a little bit, if we deliver antigen into the tumor, we can get this robust expansion of effector cells and maybe the recruitment. And that's what we did. We basically gave a tumor to a mouse, which is the red arrow. We subcutaneously vaccinated 3 times to get a good T cell response in the periphery, and then we do a single injection with the vaccine in the tumor. And 7 days later, we basically harvest the tumors and digest them and look, do we get T cells in these tumors? And we did a bunch of combinations. We wanted to exclude the effect of potentially the adjuvants, increasing the T cells, right? So we have the blue dots are sub-Q vaccination with just adjuvants alone being given inside the tumor. We have the sub-Q vaccination with IT saline, and that's strong response in the periphery, but only saline in the tumor. That's the gray dots here. And the open circles are basically saline, sub-Q IT. And as you'll notice here is when we do the double vaccination, when we give antigen inside the tumor, the red dots, you see we have many more T cells. And when we look for the neoantigen-specific T cells, they're greatly increased when we give both antigen and adjuvant inside the tumor. So delivering the antigen intratumorally, the neoantigen, it's critical to get those T cells, and especially the vaccine-specific T cells, inside the tumors. Okay, so we wanted to know what's happening here and how is all this happening, and these are the questions we're going to try and address here. We took the mesothelioma model. We did exactly the same what I just showed you now for counting the T cells, but in this situation, we let the tumors grow a little bit more to become 100 cubic millimeters when we gave them the IT intratumor vaccination, and then we did spectral flow cytometry and single-cell sequencing 7 days later. And by spectral flow, what you'll notice is in the last combination, which is the combination of the sub-Q and the IT vaccination, you can see there's a nice red population on the bottom left. Those are CD4 T cells. There's lots of CD4 T cells showing up in the tumor when we do the double vaccination. And all the other combinations, you see varying degrees of changes. These are all relative numbers, of course, because that's what we're looking at here. But we can actually go and count these cells. And what I'm showing you at the bottom, I think the most important part is the right side, bottom right, where we've counted the number of cells per gram of tissue of tumor. And you'll notice here that with the sub-Q saline, IT saline, and the sub-Q vaccine followed up with an intratumor saline, there's no increase in Th1s. There's a very weak decrease maybe for the Tregs with the sub-Q vaccine, and the CD8 T cells really don't change. But when we do the PT vaccination, the double vaccination, you can see that we have very large numbers of Th1 cells in the tumors, and the Tregs go down in absolute numbers in here. So what's happening is, with the intratumor vaccination, we are expanding Th1s, or we're recruiting and expanding, and the Tregs go down. The overall CD8s, by this flow cytometry, don't seem to be changing, because here we were only just looking at total numbers of CD8s. We did some immunofluorescence on the tumors, and here you will appreciate the purple are the CD4 T cells, and you can see here that the tumor, when we do the double vaccination, literally fills up with CD4 T cells all over the place. One of my colleagues wants to tell me that there are some micro niches in there, but we haven't really proven that. And we're trying to do that histology to see that if there are any little clusters being forming. But the point here is, whichever way we look at this, we see a lot of CD4 T cells showing up in the tumors. We did single cell sequencing, and we looked at the T cell clusters here. And basically the finding is that by doing the intratumor neoantigen vaccination, we get a recruitment of Th1 cells and non-exhausted CD8 T cells in the tumors. And I think if you just look at the bottom, the bar graphs, you'll see that the Th1, the group number 3 is the double vaccination. You'll see that we have very large numbers of Th1s inside the tumors. There is an increase for the effector CD8 T cells, and what we call effector is healthy non-exhausted CD8 T cells in the tumor. Those go up, but they're not statistically significant with this test that we're doing here. The exhausted cells in the tumor don't change. Whatever exhausted cells were there, the number of the cells per gram of tissue of tumor, they don't change. They stay the same. We looked at the TCR clonality, so we wanted to see the clones of these T cells. Are we doing anything different here? Are we changing something in the clones? And I'm showing you here basically for the Th1s, the effector CD8 T cells, which is the red, and the green is the exhausted CD8 T cells. On the blue side, the Th1s, the first set is a number of animals with a sub-Q saline and the IT saline. The second group is the sub-Q vaccine, and the third group, the red line underneath, is the double vaccination. And you'll appreciate here we have many, many more clones. There are some shared clones with the sub-Q vaccination. So these are relative numbers, but if you look at the numbers inside the boxes, those numbers are numbers of cells in the single cell sequencing that share the identical TCR. So there are many more clonal cells in the TCR, in the double vaccination animals that share those clones, which are not present in the non-vaccinated animals. So these are vaccine-induced TCRs. And yes, sub-Q vaccination induces some of them in the tumor, but the double vaccination, you have many more cells, clonal cells, and expanded. And you can look at the circus plots. It shows a kind of a sharing, how this sharing is in the top. The black arcs are actually the saline mice, and you can see they're barely visible there because there are just none of these Th1 cells being clones in those animals. They're all vaccine induced, right? And the very large numbers are in the red arcs, which are the double vaccination. Same is true for the effector cells. But if we look at the exhausted CD8 T cells, what you'll notice is in the green group at the end there are some clones that are shared, especially there's one very dominant clone which is present in the tumor, the control groups with saline vaccination, and it's still there when we do the sub-Q vaccination, it's still there when we do the double vaccination. So that clone is the dominant CD8 T cell clone against the tumor. It's there from before our vaccination, and it stays there, it doesn't go away. So we can't really change that CD8 T cell, exhausted CD8 T cell. It seems to be staying exhausted. What we're doing is we're recruiting new CD8 T cells and new Th1 cells. Well, we did a measurement of clonality, and what we find is that the intratumor neoantigen delivery basically increases the T cell clonality in the tumors, which is kind of obvious from what I just showed you, but you can do this in a more fancy way. We use the Gini index, and you can see here that the Th1 cells, the red dots here show you that there's a very large increase in the clonality of the Th1s, an increase in the effector CD8 T cells, but there's no difference, no change in the clonality in the exhausted cells. But it's important to zoom in a little bit and say, okay, do we have very large populations that have expanded? Do we have large clonal populations? And what we do find is that new intratumor vaccination with the neoantigen, that neoantigen delivery will induce very large clones in the Th1s and the effector CD8 T cells. I'm just showing you the control mice on the left here. The dark green dots are clones where you can find more than 20 cells in the single cell sequencing that share that clone, right? And what you'll appreciate is that most of those dark green clones are in that bottom left exhausted CD8 T cell population for the control animals. So the control animals, if you're going to find larger clones, they're all pre-existing, if you want, exhausted T cells. Those green dots are still there with the sub-Q vaccination. You're starting to see some large green dots in the other two populations, the Th1, which is the blue population circled, and then there's that salmon color effector CD8 T cells. But we see a very large increase when we do the double vaccination of those large clones. And we can count those. We call them highly expanded clones. We can get the numbers of those per gram of tissue. And you will notice here that with the PT vaccination, we have very large numbers of the Th1, of the effector CD8 T cells, large numbers of expanded clones, numbers in terms of the size of the clone, right? But for the exhausted CD8 T cells, we just really aren't affecting the clonality of those cells there. They just don't go up, they don't go down. Whatever's in the tumor just stays there. We're just bringing other cells in when we do the absolute counts. Remember, these are absolute counts, and that's what we really should be looking. If you look at relative numbers, the exhausted cells go down, right? But those exhausted cells, the numbers are there. They just, there's many more of the other T cells that come in. All right, so if we look now, I showed you that the Tregs go down, and I'm just going to show this very briefly. If we take the ratio of Th1 to Tregs, or the ratio of the CD8s to Tregs, or if we take the ratio of the Th1s to the M2 macrophages, I didn't show you that data, or the effector CD8s to the M2 macrophages, you will see that with the PT vaccination, we have a dramatic change of that ratio. A dramatic change to anti-tumor T cells versus the suppressive T cells, whether it's Tregs or M2. So we are having a very, very dramatic change, if you want, in the tumor microenvironment towards an anti-tumor state. So we asked the question, okay, we know we have more T cells, but when is this happening and how fast is this happening? So we did the similar experiment I showed you before, but we just looked at the tumors one day after intratumor vaccination. And if we do that experiment, already we have very large numbers. We have large numbers of CD4 cells and CD8 T cells inside the tumor. So IT vaccination is doing something within the 24 hours that's pulling in T cells, right? And those T cells, once they're in, they start expanding up to day 7. And what we believe, I use the word expanding rather than continuously recruiting, is because we see that there's a very large increase in Ki67 CD4s and CD8 cells in the tumors after the IT vaccination, the double vaccination. Now, typically the textbooks tell us a lot of things are happening in the lymph nodes, but we're trying to see whether is everything happening in the tumor or not. And basically, if you look in the lymph nodes, there is no difference in the lymph nodes. There's nothing is happening in the lymph nodes. It's all in the tumor. It's CXCR3 cells that are being recruited. We know that by counting the cells in the CD4s and the CD8s inside the tumor. And we looked at, are we inducing factors that recruit these cells? And this dotted line that's in these figures is basically the baseline of these chemokines and cytokines in a control tumor that's received saline. And we're comparing here sub-Q vaccinated animals. Sub-Q vaccinated animals that they got the adjuvants in the tumor, because it's very important to see what's the adjuvant effect, right? And the double vaccinated animals, the red. And you'll see here that we're reducing, these are log 2-fold axis here, and we are having 4 times log 2 for CXCL9, CXCL10 is going up, interferon beta, interferon gamma. All of this is happening within 24 hours. If you look at day 7, those changes in the chemokines and also the cytokines start to go down. So what we're doing is, by doing the intratumor vaccination, we're getting a very rapid induction of chemokines that will attract more T cells inside the tumor. So I won't go through these data because the time is already up. But, does this vaccination work? I will just show you here. We've tried every combination you can imagine with the PD vaccination. And you can see the red line here. We have survival and reduced tumor growth in the A17 model. If I show you an individual experiment, you'll appreciate here that In the last red, 6 out of the 15 mice become tumor-free, which is quite unprecedented for these type of tumors. We looked at the pancreatic tumor. You can see here that the PT vaccination induces a very potent, if you want, control of the tumor. With individual animals, 4 out of 5 animals will become tumor-free. This is really quite remarkable protection against these tumors. If we take a different antigen, because someone might say, well, is it really the neoantigen or is it just any antigen we deliver in the tumor will do the job? If we use pathogen peptides, very strong peptides from a bunch of different pathogens, you can see we get a partial kind of protection as long as we do the vaccination. That's what these orange lines are showing here. But once we stop, the tumor just starts growing very fast, and very few animals are tumor-free. I think it's like 1 out of 10, which is basically the background for this model. We can do a protection. There's memory there, long-term protection. So the mice that rejected the A17 mesothelioma, you can see this is a very long experiment. We re-challenged these animals at 110 days after the first tumor. We waited to be sure that the tumor is not going to come back, and at 110 days, we re-challenged the animals. And the red line here, as you can see, none of the animals grew a tumor. The naive animals, as you can see, as you expect, in 2-3 weeks, they're already reached the endpoint. So with that, I just wanted to say that with a sub-Q vaccination, we can use lots of T cells to circulate, and that's typically what we've been doing all these years, I guess. But with the intratumor delivery of the antigen, what we're getting is rapid production of cytokines and chemokines. We have a kind of an idea of a cascade, but this allows us to now pull in the T cells into the tumor and make T cells proliferate, recruit more T cells, and also reduce the suppression in these tumors. And with that, I would say that we've kind of closed this circle that we were trying to close, the cancer circle, the immunity circle, this way. But the intratumor vaccination makes the tumor explode here, as you can see here. And as acknowledgements, the funding is the Dutch Cancer Society, the University of Tokyo, and the work has been done primarily by Ko Hyoki, a wonderful Japanese postdoc, and Melissa Castro. She started the work, and Marlus is a technician who's done a lot of the work in the group. And with that, I thank you, and I'd take any questions.
Thank you, Peter, for a wonderful talk. In the interest of time, we can have one question. So, yeah.
Yeah, thank you. Thank you, Professor Peter, for a very innovative talk on this concept of new antigen generation. So, my just 2 questions, very small. One is, you have used only localized tumors, this strategy, or in a metastatic disease?
So these tumors, because they are subcutaneous tumors, they don't give metastatic.
For example, you have used the model of mesothelioma and pancreatic cancer.
Yes.
So
but when you give those tumors subcutaneously, they don't give metastasis. We're trying to do an orthotopic pancreatic. And we've done the first experiments we did. We know that when we do the orthotopic model, and we're not doing that in the Netherlands, we're doing it in the US because we can't do it in the Netherlands. We get many more T cells in that model. That model does give metastasis, and we're trying to look at that.
Yeah. Another question is that you want to transform a cold tumor into a hot tumor to make it immunologically, increase the immunogenicity. Have you used immunotherapy?
We've done anti-PD-1 in this.
Yeah.
I didn't show you the data. The problem is with when we do the vaccination, we get very good protection, and when we add PD-1, we get an increase, but it's not huge because the animals are nearly protected. So we get a slight increase, but I think that's the limits of the protection that we can get.
Thank you.
I think Peter will be here, so if anybody has a question, they can discuss over the tea or the dinner. Because of the time, interest of time, we would like to conclude this. Thank you very, very much, Peter.
Thank you, Professor SKICC, for the compelling lecture. I would request the chair and co-chairs to please come forward for felicitating the speaker. I'd request Professor Mohammad Jamal Dar to kindly felicitate our speaker, Professor SKICC. May I now request Professor Katsakis to kindly felicitate our chairperson, Professor Mohammad Jamal Dar. May I now request Dr. Amit Awasthi. May I now request Dr. Amit Awasthi to kindly felicitate Dr. Mohammad Hussain Mir. May I now request Professor Katsakis to felicitate Dr. Amit Awasthi. Thank you, everyone. We now move on to our next keynote, and to preside over this session, may I respectfully invite Professor Satya Das, All India Institute of Medical Sciences, New Delhi. Professor Rajiv Goel, LHMC, New Delhi. Professor Rajiv, is he around? And Dr. Riaz Ahmed Mir from AIMS. That's fine. Sir, मैंने बुला दिया सकता था. Rajiv Goel is not here, sir. Please sit. Our next keynote speaker is Dr. Pravesh Gupta, who is an assistant professor at the Mayo Clinic, Phoenix, Arizona, United States. His research expertise spans neuroimmunology, cancer biology, brain tumor microenvironment, innate immune signaling, and translational immunotherapy. Through his work, he has contributed to advancing our understanding of the complex interactions between phagocytic immune cells and tumor progression within the central nervous system. Dr. Gupta has authored approximately 33 research publications in leading peer-reviewed journals and has made significant contributions to the fields of neuro-oncology and immunology. Dr. Gupta will speak on unlocking the potential of phagocytic circuits in the brain tumor microenvironment. Dr. Gupta, the podium is yours. We warmly welcome you.
Is the laser working? वो बोल दिया ना ये जस्ट
Can it be a little quick?
Can you hear me? Is it audible? Is it audible now? Okay. Sorry for this technical. So, just to start with how we have learned, brain immunity in the fundamental books, through our PhD and postdoc days. I'll walk you through some anecdotes, through the centuries. It was in 1787 that Paolo Mascagni first described brain lymphatics through paintings and drawings, but his observations were dismissed. And there were 3 to 4 more, neuroanatomists who described this, but, it was almost as if it didn't exist. Until 2015, when Jonathan Kipnis' lab, there's a postdoc who was doing staining on the brain dura mater, there was an accidental discovery of brain meningeal lymphatics. So with this discovery, spanning, so you see the disconnect about 2 decades, no, not 2 decades, 2 centuries, we were not able to appreciate the highway of immune traffic from the periphery to the brain. And how this all, clouded our observations over the years is lymphatics were absent. That's what we were taught. And blood, and there was a concept which was seeded in our minds, blood-brain barrier. So I'm going to just break some paradoxes, as I speak. And most of the times, we don't question these, fundamentals because we have a large body of literature supporting this. And in brain, we have the dominant cells, which are the microglia. And this is the large family of phagocyte which we understand. And when it comes to brain tumors, we have terms like tumor-associated macrophages, glioma-associated macrophages. All these, types of terms have been used to describe them as, immunosuppressive cell types which tone down the immunity. And I would advocate something very different as I walk through this presentation. And I think the previous speakers, Professor Peter as well as Professor Kuchru, already touched a lot on what is the role of T cells in cancer immunotherapy. So my job is easier not to touch this topic. So what has been really shown, at least until date, what we understand is brain tumors are cold tumors and they have low tumor mutation burden and neoantigens. And there's a T cell scarcity. So if T cells are less, and I'll show you with the data that we have from human patients, if T cells are less, what are we trying to rejuvenate is a fundamental question we asked. And CAR T is one of the modalities which has worked wonderfully in liquid tumors. And we have been enthusiastically trying to, make it applicable to the tumors, solid tumors. And what we know as of now, that none of the approaches of CAR T, whether single CARs or dual CARs, against the tumor-specific antigens or tumor-associated antigens, in any of the combination have resulted in long-lasting tumor responses, anti-tumor responses. And so we were just trying to think, in a lab, if, let's say, we even reintegrate these T cells, what will happen? There's already a tumor flare, the inflammation which is caused by in the brain. And we are throwing T cells inside. And what will happen is, brain being a tolerogenic environment, will shut it down. That's exactly what is witnessed in the clinical trials. And yet, we are running after T cells. And today I'm going to tell you something against the wave. Brain immune microenvironment is myeloid cell rich, and we have shown it, not only us. There are many leading labs across the world with the advent of the single cell technologies. They have characterized the predominance of myeloid cells in the brain ecosystem. Not only adult brain tumors, but even the pediatric brain tumors. Regardless of whatever kind of inflammation it is, I think God gifted brain with something which is very beautiful, which is the innate immune cells. They're fast, they're first line of defenses, and they can do the job. It's just that we have not been able to identify what's the trick. And I think this I already described, the richness of the myeloid microenvironment, right? So if we understand fundamentally what is the biggest immunological process, the abundant immunological process that clears abnormal cells in the body, phagocytosis. Every day, about 0.3 trillion cells are engulfed, and cleared silently without any bystander inflammation. If that's the case, we need something, a process like this, to clear the tumor silently without inducing bystander inflammation that causes a lot of side effects. And since it's more dominant as phagocytes, so what are the molecules that regulate this phagocytosis phenomenon? There are some molecules which have been identified, a family of cytokines which is don't eat me receptors and eat me receptors, and they facilitate this. What has been the status in the clinical trials? The punch-1, which is the CD47, has not worked in clinical trials at all across most of the solid tumors. Then what we do is we try to pair it with PD-1 or pair it with some combination therapy, hoping we will be able to rejuvenate this arm. Punch-2, which is the CD24, because people think, it's not people think, we at MD Anderson, Betty Kim has shown that CD24 is another don't eat me receptor. If you neutralize this, maybe you can tackle the refractoriness of the mechanism that is compensating for the punch-1. So we identified this gap in knowledge, and when I was in my postdoc at MD Anderson Cancer Center, I asked a simple question. Can we understand the diversity of immune cells in the human brain through a tumor standpoint and also using only patient samples? Let's not go to mouse samples at all, because what happens is the phagocyte family in the brain of mice, there's no one-to-one correlation with the humans. And we had this simple hypothesis: do we even have immunoreactive phagocytes, or all of them are immunosuppressive, right? So we created a very, very reverse translational brain tumor immunology program, under the flagship of MD Anderson Moonshot program, where we had this simple question: let's just nail the cells and the molecules in as a part of the brain immune system, and maybe we find some leads. And to do this, we just put a suite of complementary technology pipelines from single cell transcriptomics to proteomics to spatial and functional proteomics and liquid biopsy. And once we are able to create this, phenospatio functional insights in the cells and the molecules in the human brain, then we can go back to the mouse. That was the idea. So with a multi-omics approach, as I described before, we started with patient samples, and we used this as a discovery cohort where we looked at the low-grade gliomas, which are the IDH mutant of the primary and relapse subtypes, and also the high-grade gliomas, which are the IDH wild type. And we dissociated the cells to enrich for immune compartment with Perkol gradient, and identified that with the differential expression of CD45 high and low, and high being the infiltrating ones and the low being the resident ones, we were able to, differentiate this first time in the humans because this was never proven. Only in mouse this differential expression of CD45 high and low was shown, but never shown in the human brain concepts. So, and we had this very, very, fantastic genomics and computational biology team, and it was a very multidisciplinary approach. Once we were able to understand the cellular transcriptome, we validated this with multispectral cytometry. And this, I have already shown it before, this diagram, this UMAPs, and also the quantitation. The message here is the relapsed gliomas show higher lymphoid infiltration compared to the newly diagnosed. And this is just 3 to 4 samples each subtype. I don't show the flow cytometry data here, where we profiled 55 tumors. And, regardless of IDH mutant or IDH wild type glioma category, we noticed that T cells were increased. And this is the entire cell atlas that we show here. All kinds of phagocytes. It ranged from microglia as the dominant players, and we had MG-like cells, which are the monocytic derivatives, as well as we had monocytic derivatives of macrophages, dendritic cells of CDC1 and CDC2, which I don't show here. But you can appreciate if brain was immune privileged, where did we find all these cell types? There are about 22 distinct cell types in human brain tumors in the inflammation scenario, and they mediate the glioma immunity. And if we look at what are the specifics, what is the cellular perturbations? What we found is whenever you have a relapse of glioma, whether IDH mutant or IDH wild type, there's a decrease in microglia. And these are the major phagocytes in the brain. And if these cells are decreasing, what is compensating for the decrease, with the decrease of these cells, we are seeing concomitant increase of microglia-like cells. What do you mean by microglia-like cells? This is a very vague term, but just to make it very specific, these were CCR2 derivatives of microglia, which differentiate in the brain microenvironment. And we looked at even the T cells, as I described before, T cells were increased during glioma relapse. So you see there's a lot of T cells, during relapse, yet the patients are dying. So just to switch gears, when we understood all this different, cell types, in different kind of IDH glioma subtypes, we just wanted to understand what is their functional implication. If these are the only 2 dominant cells which are shifting in high-grade and low-grade gliomas and during relapse, we characterize for all possible molecules, PD1, TIM3, granlysin, granzymes, and perforins. We are not able to see any differences across all the 4 categories of glioma subtypes. So I'll get to some molecular discoveries later, but let me finish this characterization phase. So another question we had is, the field always struggles with M1 and M2 classification of macrophage polarization. Although we know this is very, very primitive classification, which was done with in vitro assays, what we did is using a single cell data that we had, we wanted to understand how many modules exist beyond M1 and M2, and what we are able to identify are 10 different meta modules were present in the polarization spectrum, of which beyond the M1 and M2, you will always see either hybrids of multiple states. It is never like you'll have one M2 and it doesn't have any other flavors of functions. Okay, so it's a very spectral phenotype. And we provide this community guideline how you should go about it. And just to give you a methodology how we did it, our idea was simple. There was a paper by Zhu et al. In 2014, and they used human monocytes and subjected them to 40 different cytokine perturbations in different combinations. whatever was the flavors of polarization patterns, we just superimposed on our data. And if we are able to do across cancer, we will get the leads basically, what is this, spectrum? And, it was very heartening to see one of our collaborators was able to show this in an in vitro study, and we are following it up, for the existence of these subtypes in vivo. Second clarification that we provide, and it is also a clinical application for, tumor biologists or, people who are working in translational front, is we use this LM22 signature. This LM22 signature is derived from blood. And we try to use these signatures to do an immunogenomics deconvolution of the tumor microenvironment. If you have an incomplete reference, I'm not sure how we are going to capture the breadth of different cell types. So what we did in order to create a methodology, we had the single cell data and we had performed bulk RNA-seq on the same data. And our collaborators had the tumor signatures. When you take a tumor from the surgery room, it's a mix. It has a tumor and the immune compartment, both, also the stromal compartments. If you don't subtract from the TCGA data the tumor compartment, what you will see is an impure data. So what we did is, from our immune-rich data, we extracted the TCGA data and subtracted the tumor. So now you have something which is correlative with immune pure populations. And we describe, at least for the brain, something very different. So blood does not have microglia. So if you deploy the LM22, you will not see those compartments. So here we describe 36 different signatures transcending from microglia, macrophages, DCs, CD4, CD8, and their functional states. And if somebody who is trying to study, brain and immunology in the brain, if they deploy these signatures, probably they will have better leads. And not only, with this small subset of 16 or 21 samples that we had overlapping data, we also extended this to GLASS cohort, which is this study design that we had, primary and recurrent gliomas of IDH mutant and wild type. So we deployed this, and if you see on the left, this is what LM22 will give. And the one that you see on the right, this is the gliotime 36, right? So that's the difference. So if you take an incomplete reference, you will not get the information that you desire in the clinical trials when you're looking at immunogenomics deconvolution. So this is a summary of this part of the cell atlas that we created. Let's get to, 3 discoveries that we made beyond the ones which I previously showed in the eat me receptors and don't eat me receptors. So, and this was very fascinating because we started with humans. This was a total surprise. So we identified granulysin expressing microglia. And for me or anyone who is looking at this, it is going to be a total surprise because granulysin is conventionally expressed on the NK cells and the T cells. And why were people not able to discover this was because they were starting from mouse. And mouse does not have granulysin expression. And since we started with patients, we were able to discover an inflammatory population of microglia expressing granulysin. And we also, created this, different functional states of microglia amongst the inflammatory microglia groupings. And we were able to, if you see all these different patterns of inflammasome and also the interferon family, these are very distinct flavors of the functions among the inflammatory microglia. You just cannot say this is one type of inflammations. There's a lot of diversity here. And since this was very new, we wanted to validate it in existing literature. And if you see this, we validated the same in a publication from Antunes et al. And we were able to again find this granulysin expressing microglia. Okay, this is transcriptomics. It can mislead you. So we really wanted to be sure is it really there in the tumor microenvironment, or is it some artifact? We did the Codex assay, which had about 20 protein markers. And if you can see here, it showed the TREM2 co-expression with granulysin. And this correlated with the transcriptomics discovery that we were able to chance upon. And we also show it with flow cytometry. And this rare population is observed in high proportions in the relapsed patients of IDH wild type. And we also identified APC-like microglia, which many people have described. There's nothing new over here, but this was also confirmed with our study. So what does it do basically? Granulysin expression. What would it do? So since we had this 40 parameter panel where we had all these granulysin and checkpoint molecules and also different functional states of phagocytosis, we just did a correlation study, and what we found is whenever you have a granulysin, there was high expression of phagocytosis marker. And this phagocytosis marker spanned from CD86 to CD163, which is a scavenger receptor. I know people would question CD163 is an M2 marker, but functionally it's a scavenger receptor that clears abnormal cells. So yes, this is all correlative studies. Does it really do it? Yes, it indeed does. So basically what we did is we took glioma stem cells from patients, and we took this and we showed that this indeed does regulate phagocytosis. Similarly, we identified TREM2 to regulate phagocytosis in glioblastoma, and regardless of, if you see on the right, regardless of wherever the TREM2 is expressed, it regulates phagocytosis. So this is the second molecule that we discovered in this, the brain microenvironment. And I talk about the third molecule, and then I'll discuss basically what is the use of these novel molecules. As Dr. Khashru mentioned, that TIM3 is a checkpoint molecule, and what we wanted to do is, from our single cell data, we wanted to see what are the interactions of microglia with lymphoid cells. And what we identified is a galectin-9 as a signaling hub between all lymphoid cells. And TIM3 was one of the suspects. But we thought, okay, so this is in a trance. Here we are talking about what is the function of galectin-9 on microglia rather than what it does to the T cell, because brain microenvironment, there's no T cells or it's very scarce. And even if T cells are present, they will not migrate to the tumor because there's a trafficking problem. And we show this that galectin-9 is expressed on microglia and not the tumor cells. And again, we did a galectin-9 positive and galectin-9 pathway analysis, and we found all different processes of phagocytosis were upregulated. And we did again the same assay with the glioma stem cells by neutralizing the gene as well as the protein with antibody. In both the settings, we were able to identify that galectin-9 is a pro-phagocytic regulator. So if you see this entire reverse translational immunology program that we developed, in 5 years, we have been able to identify 3 novel regulators of phagocytosis. So what? Is there any application about this? So what we talk about as a futuristic roadmap is, at least for the brain and the tumors where you have high abundance of phagocytes, what we want to create is a family of phagocytosis rejuvenating immunotherapies with a very novel non-viral genome insertion tool which can achieve multiple transgenes as a cassette. So whatever we discovered, we are trying to put it in different synthetic circuits. And with this, we have generated iPSC-derived synthetic circuits in combination with CARs and without CARs. And we have a very promising data where, one of the circuits where we engineer with TREM2 and also a checkpoint molecule, we have seen survival of the mice, 100% of the mice, they survived. And even in the re-challenge setting, they were not, they were able to reject the tumors. So in a nutshell, what we are trying to do is, if we deploy these synthetic circuits to rejuvenate phagocytosis in cold tumors across a family of solid tumors where, tumor-associated macrophages are dominant, I think they should be able to do the trick of eliminating the tumor. And that's our pursuit, hoping that we could create next generation of phagocytosis driven immunotherapies, which would be allogenic, and they would be of a very low cost. And the better part of this immunotherapy is it has innate memory, and it can even prevent relapses. That's the idealistic roadmap we have as a team. Yeah. Thank you for your time and attention. I would like to thank all the people who contributed to this program. We have Shivangi and Kenzie, both are PhD students, and they were instrumental in doing the characterization studies of the cytometry. Single cell team, we had from Nicholas Naveen group, Tuan and Zhonzo Lee. They did the single cell, library preparations and things like this. And, the computational genomics streams, which helped us navigate this complex, computational framework. And the clinical collaborators led by Frederick Lang, and also our collaborators at UTMB, who were able to create human microglia, the data which I showed. And we were able to, test the phagocytosis with human brain tumors. And the funding agencies, the MD Anderson Moonshot Program, CIPRIT, and lastly, the most important player is the patient owners. Thank you for your time and attention. I would like to have any questions, if any.
Thank you, Dr. Gupta, for your nice talk. And since we are late, there will not be any question. Dr. Gupta is available.
Yeah.
So you can ask him if you have any question during dinner time or maybe tomorrow. Thank you very much.
Thank you.
Thank you, Dr. Pravesh Gupta. May I request Professor Satya Das to felicitate our speaker, Professor Dr. Pravesh Gupta, please. Thank you, sir. Sir, please take the chair. Yeah. Now we have a lecture from a giant of innate immunity and peripheral tolerance research. Professor Chandrasekhar Pasare is chair and professor in the Department of Immunology at the University of Texas, and professor and director of the Division of Immunobiology at Cincinnati Children's Medical Hospital. An internationally recognized immunologist, Professor Pasare's research focuses on innate immunity, peripheral tolerance, checkpoint molecules, dendritic cell biology, and the mechanisms that regulate inflammation and autoimmunity. Through his pioneering discoveries, he has significantly advanced our understanding of how immune checkpoints and innate immune signals shape adaptive immune responses and maintain immune homeostasis. Professor Pasare has authored over 86 research publications in leading international journals and has contributed to several translational innovations in immunology and immune regulation. Professor Pasare will speak on organizing principles of peripheral tolerance, role of checkpoint molecules in regulating inflammation. Professor Pasare, we are truly honored to have you in Immunocon 2026. Thank you.
Thank you so much. I'm trying to figure out the glitch here. All right, thanks for that introduction. Thank you, Delafrose, for inviting me to this beautiful valley. It's been a great experience, and Thank you for all the troopers that have stayed back for the evening. So I know this is kind of late in the day. All right. So what I thought I'll do today is talk a little bit about some of the recent work in my lab that deals with understanding checkpoint molecules and really how tolerance works in the periphery. My lab is primarily interested in understanding how dendritic cells recognize pathogens and how that information is related to T cells, but that has also forced me to start thinking about how T cells regulate themselves in the absence of any microbial infections. So this is sort of the current understanding of how tolerance works. Central tolerance especially works in the concept of when you have low affinity peptides in the thymus, you have positive selection. But when you have intermediate affinity peptides, you have generation of Tregs, and when you have presentation of high affinity peptides from the periphery, you especially with the transcription factor A through MTEX, the self-reactive T cells are deleted. But we know that while central tolerance works really phenomenally well, that peripheral tolerance is an important aspect of maintaining homeostasis, and nothing really exemplifies that more than this paper from Sasha Rudensky's lab where he expressed a diphtheria toxin receptor and the control of FOXP3 promoter, and just one dose of diphtheria toxin leading to deletion of Tregs leads to profound autoimmunity in the periphery, right? So these mice have undergone proper positive and negative selection in the thymus. They have peripheral immune system, but just removing the Tregs from the periphery leads to profound splenomegaly and lymphadenopathy that, then eventually the mice succumb from autoimmunity. People generally don't die of autoimmunity, but this is an extreme case. But mice with mutations in those, the scurvy mice do die and don't make it very long. All right, so if peripheral tolerance works really so well, how do you actually get immune responses at any juncture? So one of the early papers that I had published as a postdoc was really trying to understand this mechanisms of how really Treg suppression is overcome, and we had demonstrated that when DCs recognize pathogens through pattern recognition receptors like TLRs, they make cytokines, and especially IL-6 sort of overcomes this Treg-mediated suppression, leading to priming of naive T cells into expanded antigen-specific T cells. So the question still remains, right? So when you have microbial infection, you can overcome Treg-mediated suppression, but why does removal of Tregs lead to this profound autoimmunity? Which suggests that many of the T cells, in fact, in the periphery are self-reactive. So in that specific case, the T cell intrinsic tolerance really becomes really very important, right? The two famous molecules that you've heard of today are CTLA-4 and PD-1, and they work a little bit differently. CTLA-4 competes for binding to CD80 and 86, and PD-1 has ITAM motifs and sort of inhibits ZAP70 phosphorylation, etc. Right? So there are therapeutic benefits for blocking negative regulators of T cell activation. This famous paper from Jim Allison's lab back in 1996, where he showed that using anti-CTLA-4 you can essentially cure these implanted cancers in mice, and very similar work from Tsuko Honjo's lab where blocking PD-L1 leads to recovery from tumors, right? So this is elegant demonstration in mice, and these molecules have not been translated. I don't have to tell you guys how effective cancer immunotherapy has been. So what has been interesting about PD-1 is that when early on it was discovered in Tsuko Honjo's lab, it was really sort of, he was really looking at activated T cells, but seminal work from Rafi Ahmed's lab really established PD-1 as an exhaustion molecule, right? So this is a very interesting experiment if you pay attention, where mice were infected with acute and chronic LCMV, and then if we look at day 6, there is really no difference in antigen-specific T cells expression of PD-1. But once the antigen is cleared, when the mice don't have infection anymore, which is the acute strain of LCMV, the gray bar, the PD-1 expression on T cells comes down. The red bar is where the mice are constantly infected and there's presence of antigens. So even at day 40, these antigen-specific T cells are really expressing PD-1 on the surface. And this is what led to the idea that exhausted T cells express PD-1, and then blocking PD-1 obviously leads to relief of exhaustion and treating of tumor cells. So So this is sort of the established paradigm right now of how mechanisms of checkpoint blockade work, right? So that CTLA-4 is rapidly upregulated and activated T cells. It's able to compete with CD28 for binding to CD1886. And that, you know, that really inhibits the ability of T cells to get primed beyond what is needed. And really that blocking PD-1 relieves the effector phase of T cells to kill tumor cells, right? So CTLA-4 basically working during priming and PD-1 really working in the periphery. So there's obviously benefits to blocking PD-1 and CTLA-4, but there are many undesirable outcomes of blocking these negative regulators of T cell activation, right? So there's lots of immune-related adverse events that happen in these patients. You know, you can go from all the way from encephalitis to enteritis. Every kind of pathology happens, right? Every itis is listed there. From uveitis to vitiligo and myocarditis and pancreatitis, etc. And this is not surprising given the genetic data from the mice that if you don't have PD-1, you develop lupus-like autoimmune disease, and that was actually due to antibodies that were generated. But if you don't have CTLA-4, those of you that have worked with CTLA-4 knockouts know that you get lymphoproliferation and multi-organ tissue destruction. And this was work from Arlene Sharp's lab along with Jeff Bluestone. So as far as I'm concerned, sort of the unanswered questions in this field are what are the relative contributions of PD-1 and CTLA-4 in driving efficacy of anti-tumor immunity? And what are the mechanisms that underlie immune-related adverse events? You know, are the same events that are leading to killing of the tumor cells, are they also responsible for causing damage, in various organs and tissues? And what exactly are the roles of PD-1 and CTLA-4 in homeostasis? One of the adverse events we decided to focus on, we can't really study all these diseases. It's really almost impossible to understand the antigens that are expressed in all the different organs. But one of the observations we had made in my lab is that if you simply take regularly housed mice and give them anti-PD-1 and anti-CTLA-4, you get splenomegaly and lymphadenopathy, which very much resembles Treg depletion that I showed earlier from Sasha Rudensky's lab. We are really interested in sort of going after myocarditis, basically because a MD PhD student joined my lab and she was really interested in understanding why blocking PD-1 and CTLA-4 and specifically blocking PD-1 causes myocarditis. Here are some of the more important clinically relevant reasons. So the incidence of ICI cardiotoxicity is estimated to be about 2%, which is if you're giving it to 100 people, 2 people develop cardiac toxicity. If you're going to live longer, that's an acceptable risk. But the problem with that is there's 50% mortality. So half the patients actually die, and they can die as early as 10 days. And there is an increased, what is interesting is that there is an increased risk when it's a combination therapy with PD-1 and anti-CTLA-4. And it's sort of tracked with early data from Honjo's lab where autoimmune dilated cardiomyopathy was actually reported in PD-1 deficient mice. And as I mentioned earlier, it was because of autoantibodies against troponin 1. So T cell specific, there's a paper recently showing that alpha myosin specific CD8 T cells which drove myocarditis in PD-1 and anti-CTLA-4 administered mice. So that's, Kathy that joined my lab about 5 years ago, wanting to work to understand why humans or mice develop myocarditis. So we decided to make a new mouse model. So I'm not going to go into the details of how this mouse works. Basically, it's ovalbumin that's expressed in the heart. It's a DOX on and DOX off system. So we can express the antigen only in the heart and not in other tissues, and we can express it when we want it. So on the left, the mice, if you take doxycycline off, they start expressing the protein. And as you can see here, you can see my pointer. Only heart has ovalbumin and not the other organs. So this was really created a very elegant system where the antigen expression was restricted to the heart. So what we decided to do is instead of activating endogenous T cells, the endogenous T cells were negatively selected and deleted because I think it's a little bit leaky, which actually turned out to be a good outcome because then we could just transfer a specific number of T cells in vivo and ask what happens to those T cells. So here is sort of the outline of what I'm going to show you. So we basically have the mice we give it on doxycycline. The antigen is not expressed. So about 5 weeks we take doxycycline off. Ovalbumin is now expressed, and we transfer about 1,000 to 100,000 OT1 T cells, which are specific to this infectious peptide that the antigen expresses. And we give 3 doses of anti-CTLA-4 and anti-PD-1. And then basically look at what happens on day 9, right? So this is sort of a very targeted regimen in mice, but probably a little bit different than what patients get. But this is what we see. So when OT1 T cells are transferred and mice get checkpoint immunotherapy, their heart weight to body weight ratios go up, so the hearts are getting hypertrophied, and you have a lot of T cells, CD45 positive cells in the heart. Right, so there's a lot of inflammation. So if there is no ice, these are mice that receive OT1 T cells but no checkpoint. So remember, these hearts are actually expressing the antigen. We're giving OT1 T cells. The OT1 T cells are in fact reactive to the antigen being expressed in the heart, but only when you have anti-PD-1 and anti-CTLA-4 do you start seeing pathology. So when we started looking at the heart in wild-type mice versus mice that are expressing ovalbumin in the heart, if you give anti-PD-1 and anti-CTLA-4, of course, nothing happens in wild-type mice. In mice that receive OT1 T cells, you don't really see that many OT1 T cells in the heart. But when mice receive both anti-PD-1 and anti-CTLA-4, you see this massive expansion of T cells that happens in the heart. And that's quantified here on the right. So we decided to really look at what is happening in the mediastinal lymph nodes, which is what drains the heart, versus what's happening in the heart. And this is sort of a kinetics experiment. So no ICI, you're not really seeing any expansion of T cells. On day 5 in the mediastinal lymph nodes, the OT1 T cells start expanding, and there's more expansion on day 6, and of course there's more expansion on day 9. So this is gated on CD8 T cells that are expressing the alpha 2 and CD45.1s, and that's the quantification. But if you start seeing in the heart, you can see here that T cells are expanding in mediastinal lymph nodes, but you're not seeing any T cell expansion in the heart at the same time. So the idea is that they're getting primed here and really start trafficking into the heart and then start accumulating in the heart, right? So the priming is actually happening in the draining lymph nodes, and we can actually sort out DCs and see the peptide being presented. So all right, so then what really is happening? The experiments that I've shown you so far, includes giving both anti-PD-1 and anti-CTLA-4 together. So we decided to really look at how these individual molecules are working. So when we blocked CTLA-4, and this is a very surprising result, when we blocked CTLA-4, it really did not change the expansion or accumulation of T cells in the heart. And we gave anti-PD-1, it led to massive recruitment of CD8 T cells to the heart. But what was really interesting is that the combination blockade really led to humongous expansion, or recruitment, I should say. The expansion is not really happening in the heart. The priming and expansion is happening in the draining mediastinal lymph nodes, and the T cells are trafficking to the heart. And that's what's quantified here. So anti-PD-1 blockade is what's leading to priming, and CTLA-4 blockade is what's leading to expansion of that priming. And this sort of, this forced us to really start looking at the kinetics. This is all CD8 T cells, by the way, of PD-1 and CTLA-4 expression following T cell receptor ligation in vitro. And we've also done these experiments in vivo. So this is basically ligating the T cell receptor using the peptide or anti-CD3 and looking at CD44 expression. And as you can see here, as cells start dividing at about 48 hours, they express CD44. But what is really interesting is that T cells that haven't even entered cell cycle start expressing PD-1, right? So, and even here, as you can see, only about 60% cells are divided, but almost 90% of the cells are expressing PD-1. And almost all cells are PD-1 positive at 72 hours, which is very different than CTLA-4. So the CTLA-4 expression is not detected about 72 hours. So sort of it's reversing the way we think about this checkpoint molecule. So PD-1 was originally discovered as an activation marker and became sort of stuck as an exhaustion marker, but PD-1 is really sort of comes up much earlier than CTLA-4 on cells. And I think this explains why blocking PD-1 and CTLA-4 together works, but blocking CTLA-4 by itself doesn't work, because T cells are, when the T cell receptor is getting tickled, PD-1 is coming up and blocking that really relieves that negative regulation, and CTLA-4 is expressed a couple days later. So blocking CTLA-4 after blocking PD-1 really induces the synergy between the two molecules. And this tracks with what we see in vivo. So this is an experiment where we transferred OT1s into the transgenic mice and really looked at PD-1 expression. And this is about 12 hours after transfer in the transgenic mice. Once the T cells are seeing the antigen, they're expressing PD-1. These cells are not entering cell cycle. So what we think is happening is that T cells see the antigen, they express PD-1, and they're really held in check by PD-1. And once you block that PD-1, PD-L1 interaction, the T cells really take off. So, so this is sort of the idea, that my lab primarily works on, looking at how pattern recognition receptor activation of dendritic cells really drives cytokine production and co-stimulatory molecule upregulation to lead to T cell priming, the classical 3 paradigm signal, right? MHC, TCR, co-stimulation, and cytokines. But what we think happens in autoreactivity is that you have naive T cells that if they come in contact with dendritic cell presenting self-peptide, they upregulate PD-1 pretty rapidly, and the DCs express PD-L1, and that really keeps that T cell in check. So that's a major way in which peripheral tolerance works. And if you remove that inhibitory signals, then those T cells take off, right? So, and these signals are not really required when the microbial activation happens, because the co-stimulation molecules really overcome these checkpoint molecules. So then this begged the question, what is the role of CTLA-4 in peripheral tolerance? So we decided to now look at the innate immune system. So if you look at the panels on the right here, when we block PD-1, we see T cells and myeloid cells in the heart, but really that expansion, recruitment of myeloid cells really goes up when you give anti-PD-1 and anti-CTLA-4. So the combination blockade not only expands T cell activation, but really changes the quality of those T cells so that you're getting much more innate inflammation in the heart. And these are markers. I'm not a cardiologist, but so this was a collaboration at the molecular cardiology labs. So you have expression of all these molecules that are associated with damage to the heart when you have both anti-PD-1 and anti-CTLA-4 blockade. And we're checking, we're sort of ascertaining if similar principles apply to other tissues also. And one important observation of this experiment is, the original thought was the CD8 T cells are going to the heart. You have all these myocytes expressing the protein, and they're just damaging the heart by, killing the myocytes, but that's not what we discovered. What we saw, in fact, is that the peripheral knockout T cells were even worse in creating inflammation. So this is not the classical mechanism of CD8 T cell mediated toxicity. Peripheral knockout OT1 T cells were in fact sort of induced much more pathology in the heart than wild type T cells, suggesting that this was not CD8 T cell mediated killing of cardiomyocytes. So going back to the myeloid part of this, of what I was describing earlier, what we saw was that when we blocked both PD-1 and CTLA-4, we saw a lot of induction of innate cytokines in the heart. So these are basically transcripts from the total tissues. You can see IL-1 beta mRNA here and IL-6 mRNA here. So PD-1 blockade does lead to some innate inflammation, but blocking both PD-1 and CTLA-4 leads to much more profound innate inflammation. So one of the focuses of my lab in the past has been to really understand how naive T cells get activated, which is what I mentioned before, that microbial signals are detected by receptors like TLRs, and that information get conveyed to T cells. But in the past few years, we sort of have started looking at how T cells can act as microbial surrogates. So when you have memory T cells in the periphery, when they come in contact with dendritic cells that are presenting peptides that they've been primed against, T cells sort of start acting like microbes and start activating dendritic cells to induce pro-inflammatory cytokine production. So we've primarily shown this for CD4 T cells. So we're really interested if this analogy applied to CD8 T cells, whether the flow of information is going from T cells to DCs to drive this IL-6 and IL-1 beta production. And we also shown that TNF superfamily ligands that are expressed by T cells, TNF coming from T cells, and Fas ligand by expression by T cells engages respective receptors on myeloid cells to drive production of IL-1 beta and IL-6 and IL-12. And this work was, again, as I said, was primarily done with using CD4 T cells. So one of the things that we saw was that when we looked at the heart, there was a tremendous amount of TNF that was present in the heart. So what we decided to do was, whether this TNF we normally associate with myeloid cells, microbial activation of myeloid cells leads to production of a lot of TNF. So we generated OT1 T cells on TNF deficiency. So these are OT1 T cells basically are able to kill but cannot make TNF. They can kill because of perforin granzyme and fast ligand expression. So it's basically the same experiment that I showed you before, except we are now transferring wild type OT1 T cells versus TNF knockout OT1 T cells, right? And the results were staggering. So the production of innate inflammatory cytokines completely went away if the T cells were no longer able to make TNF, right? So transferring OT1 T cells was leading to production of IL-1 beta and IL-6 in the heart, and that completely went away if those OT1 T cells lacked the ability to make TNF. And more importantly, there was no difference in the ability of those OT1 T cells to be primed. So if you looked in the mediastinal lymph nodes, the T cells derived from wild-type mice or TNF knockout mice were equally expanded in the mediastinal lymph nodes. But what was interesting is that if you looked in the heart, the T cell recruitment to the heart was compromised, and then all the other innate inflammation was completely gone. So the idea is that T cells are getting primed in the mediastinal lymph nodes because of blocking of PD-1. They're trafficking to the heart, and they're basically making TNF, which potentially activates endothelial cells and other TNF receptor expressing cells, and that brings in more neutrophils and myeloid cells, and that basically perpetuates the innate inflammation. So if those T cells lack TNF, they can go to the heart. They're not really killing myocytes, but if they're not making TNF, they're not really triggering the innate inflammatory response. So basically, T cell derived TNF is required for ICI-induced myocarditis and mortality. So we tried to increase the number of T cells to look at mortality. So if we instead of transferring 100,000, if we transferred 1 million OT1 T cells, actually these mice drop dead after receiving checkpoint immunotherapy. But if we block TNF at the same time when we give these T cells, we're able to completely rescue the mice. So single dose of anti-TNF is able to prevent the ability of these T cells to cause pathology. This was sort of news to me that you can actually do electrocardiograms on mouse hearts. So if you could transfer OT1 T cells and do not give checkpoint immunotherapy, you have this nice cardiac waves. But as you can see here, if you give OT1 T cells and give checkpoint immunotherapy, you have this arrhythmia, right? So these P and Q waves are really sort of not regular, as you see in wild type mice. But what is really interesting is that if you do the same experiment where you're giving now TNF knockout OT1 T cells, your heart function is restored. Mice don't die, and their hearts are totally fine. And we have tracked these mice up to 6 months, and they live totally fine. And the same thing happens when we therapeutically administer anti-TNF antibody. All right, so TNF can, as I mentioned earlier, can be made both by myeloid cells and T cells. So when you activate cells like macrophages and dendritic cells, they make soluble TNF. And soluble TNF mainly functions by activating the TNF receptor 1. So TNF receptor 1 has a much higher affinity for soluble TNF, and TNF receptor 1, drives apoptosis, necroptosis and apoptosis in addition to activating NF-kappa B. But TNF alpha, that's membrane bound, primarily activates TNF receptor 2, which doesn't have the death domain, so it activates NF-kappa B. So we then decided to check, which receptor is the TNF that's coming from T cells is acting through. T cells primarily express membrane-bound TNF. It eventually gets cleaved, but it remains on the membrane for a much longer time. So if you give checkpoint immunotherapy, mice drop dead. If you give anti-TNF receptor 1, there is really no difference in the ability of the anti-TNF receptor 1 blocking antibody to protect mice. But if you block TNF receptor 2, then you get this therapeutic benefit where the mice don't die. All right, so all of this is great, right? So you're able to block TNF, you're able to inhibit immune-related adverse events if you're able to get rid of the ability of the T cells to make TNF, or if we are able to specifically block the ability of TNF to signal through TNF receptor 2. But what if this actually also prevents the ability of checkpoint immunotherapy to treat tumors, right? So I started this talk saying, can we really uncouple the ability of the T cells to kill tumor cells versus the ability of the T cells to cause immune-related adverse events, right? So this is an interesting experiment where we actually started using the transgenic mice, express OVA, and now inject MC38. It's an immunogenic tumor, like Peter pointed out, but really highly responsive to checkpoint immunotherapy. And these are not expressing OVA, by the way, so these are non-OVA expressing MC38 cells. Now you give OT1, you transfer OT1s and you give checkpoint immunotherapy, with and without TNF receptor 2 blockade. So if you give ICI, the mice, like I said, drop dead in about 15 to 20 days. But if you block TNF receptor 2, you're able to rescue the mice. And and again, with TNF receptor 2 blockade, you're able to restore the function of the heart, and there is this arrhythmia that you see with checkpoint immunotherapy, especially when you have heart-specific T cells present. So then what happens to the tumors? So I maybe focus at the bottom first. So if you don't give any checkpoint immunotherapy, the mice develop these large tumors. You give checkpoint immunotherapy, mice, it's not 100% penetrant, but you get therapeutic benefit. But blocking TNF receptor 2 really doesn't affect the ability of the T cells to kill the tumor cells, right? So really sort of revealing two different aspects of how these T cells are functioning. That checkpoint blockade is really allowing the T cells to kill the tumor cells. But blocking TNF receptor 2 doesn't interfere with the T cells' ability to kill the tumor cells, but really prevents their ability to cause pathology in the heart, right? So these are again tumor growth over a period of time, no ICI. This is ICI, and this is ICI plus anti-TNF receptor 2 actually works better than ICI. Then I went back and looked at the literature. It turns out that actually Treg cells express TNF receptor 2. And blocking TNF receptor 2 sort of takes away their ability to suppress. So blocking TNF receptor 2 seems to have dual outcomes. One is that it prevents checkpoint mediated immune-related adverse events. It also makes Tregs less active. So it works through two different functions in making checkpoint immunotherapy even better. All right, so the summary of what I've presented so far is that PD-1 is rapidly upregulated on T cells following interaction with cognate antigen. PD-1 inhibition seems to facilitate CD8 T cell priming even in the absence of exogenous microbial detection, and with this we think is most relevant to self-antigens. CTLA-4 inhibition alone, to my great surprise, is insufficient to prime CD8 T cells. It drives increased innate inflammation and exacerbates cardiac injury when PD-1 signaling is inhibited at the same time, and CD8 T cell induced innate inflammation is dependent on T cell intrinsic TNF. I didn't have time to show the data, but PD-1 and CTLA-4 inhibition actually do not enhance any T cell priming when DCs are previously activated with pattern recognition receptor ligands, clearly suggesting that these molecules really are there to regulate tolerance to self rather than regulate T cell expansion and differentiation following microbial infection. So that's basically the model that PD-1 blockade leads to priming in the mediastinal lymph nodes. Those T cells traffic to the heart, make TNF, and that potentially leads to enhanced inflammation. So just to acknowledge the people in my lab and Kathy who did all the work, and this is a collaboration with Jeff Malconton at Cincinnati Children's, who is the division director for Division of Cardiac Molecular Cardiology. We still have this institute thing called NIH. I don't know how long it'll survive, but we still have funding from NIH, also funding from American Heart Institution, and as well as funding from Cincinnati Children's. And thank you for your attention. Happy to take questions.
Thank you, Professor Pasare. Actually, I'm delighted that you shared this talk. When our Honorable Governor were here, he specifically emphasized on this thing that myocarditis is there. These patients have heart attacks, and you touched upon this. So we are really glad. We don't have time to take questions, but we will, once we are joining for dinner, you can ask the questions there.
All right. Thank you for your attention.
Thank you, Professor Pasare, for that wonderful presentation. May I request worthy chair, Professor Satya Das, to felicitate our worthy speaker, Professor Chandrasekhar Pasare. Please, sir. Sir, may I kindly request Professor Mohammad Ashraf Ganai, Director and University of SKIMS, to kindly come to the dais, please. Thank you, sir. Thank you, sir. Please take the chair. We now move to next talk by Dr. Ajay Dikshit. Our speaker for the evening is Dr. Ajay Dikshit. He is an assistant professor at the Cincinnati Children's Hospital Medical Center. His work focuses on the immune mechanism in pancreatitis, leading to uncontrolled inflammation and cancer development and development of next generation therapeutics. He'll speak on decoding mechanisms of immune suppression in pancreatic cancer.
This is your own laptop?
Yeah.
Laser does not work on this board. Okay.
Good evening, and thank you all for taking so late. First, I would like to thank the organizer for giving me opportunity to present today, and also thank you all for sticking up so late. And in interest of time, I'm going to keep it simple and short. So my name is Ajay Dikshit. I'm an assistant professor in Division of Gastroenterology, Hepatology, and Nutrition at Cincinnati Children's Hospital, and my lab works on pancreatic diseases, especially exocrine pancreatic diseases. And before I jump into data, I wanted to walk you through the anatomy of pancreas. So pancreas is both exocrine and endocrine organ, and it's consists of mainly exocrine acinar cells, which synthesize and secrete digestive enzyme, which help digest the food. It also secretes insulin, which regulates the blood sugar levels. And two major diseases of exocrine pancreas is pancreatitis and pancreatic cancer. And this is the current understanding how the pancreatic cancer develops. So you have acinar cells, by any means, by any stress, it transforms into ADM, which is called acinar-to-ductal metaplasia. And this acinar-to-ductal metaplasia acquires KRAS mutation. And so, and this initiates the tumorigenic process. So bad news, like half of the populations have KRAS mutation, not one, thousands of them. Good news is like not all KRAS harboring panels develop into pancreatic cancer. And by any means, it acquires further mutations such as CDK or P53 or SMAD4, and that's when the KRAS transform into fully grown tumor. And what makes pancreatic cancer so deadly? Like, it's one of the most lethal cancer known to mankind. And the survival rate is just around 12 to 13%. And as I mentioned, this is mainly driven by KRAS mutation, P53, and SMAD4. And what makes this cancer is that it's soon going to be the second most leading cause of cancer-related death by 2030. And the two main features of the pancreatic cancer is like highly immunosuppressive desmoplastic tumor microenvironment and its metastatic potential. And this desmoplastic tumor microenvironment has all the immunosuppressive cells which you can think of. And this is highly metastatic, and metastasis can start as early as Pannian stage. So most of the patients, they die because of the metastasis, which actually starts in lung, liver. Liver is the most common site of pancreatic cancer metastasis. And our lab is interested into the immune suppression part, like how the immune suppression is established and how immune suppression actually inhibit the anti-tumor immunity. And we are interested in macrophages, and macrophages are the most abundant population in pancreatic cancer, and they are essentially one involved in each and every steps, from initiation, drug resistance, metastasis, and migration, and especially they're involved in immunosuppression. And patient who has like tons of macrophages, they die early. And to get a better understanding of this macrophage population, we did the single cell sequencing from human samples, human pancreatic cancer. And what we found is that these macrophages are tremendously heterogeneous. Every patient has their own unique set of macrophages, so that makes things pretty challenging. And we were trying to find something which is common. So what we learned from this data is like the top panel shows is the ontology. Like macrophages can be tissue resident or they can derive from bone marrow. And what we learned that most of these macrophages comes from bone marrow, and there are also some tissue resident macrophages, but more or less these are all derived from bone marrow. And we are looking at the immunomodulatory. So except CXCR4, some patients has SPP1, some patients have IL-1 beta, some patients has IL-10. But the one thing which was common among all patients was CXCR4. And interestingly, all these macrophages, they showed TNF-alpha activation, which is quite surprising to us because most of the macrophages in pancreatic cancer are M2 type. But for some reason, they're expressing very high TNF-alpha pathway. And before I move to experimental model, I wanted to walk you through the data model, pancreatic cancer model, which we are using in this study. So we are using KPC model, which has KRAS mutation, P53 mutation, PDX1. This is spontaneous model. This mice develop spontaneous tumor. They show all the stages which present in human pancreatic tumor, and they develop spontaneous metastasis in liver, lung, diaphragm. And like any other curious scientist, we deleted CCR2 genes into KPC model. And what we found is like the amount of circulating monocyte drastically reduced in blood as well as in the tumor. And TNF-alpha, like total TNF-alpha, also reduced in tumor microenvironment. And we also studied what happens, like we followed this survival, and what we found is like CCR2 KPC mice, like CCR2 knockout KP mice, they survive more than just KPC mouse. And their tumors they develop pretty slowly. And you can see here that in knockout mice, at 10 weeks, they have most of their pancreas like stuck in the normal phase, and they only develop very few pancreatic lesions. Whereas the KPC mice, they develop a lot of PDA lesions and high-grade pannins. Since I mentioned that metastasis is one of the challenging part of pancreatic cancer, so we looked at what happens when you delete this TNF-alpha producing macrophages. And what we found is like all these metastatic lesions, they didn't grow as big as they compared to KPC in actual KPC mice. And there was significant reduction in liver metastasis. There was significant reduction in lung and diaphragm metastasis. So what this suggested is like TNF-alpha producing macrophages, they initiate and promote tumor formation. And as I mentioned earlier in my talk, that immunosuppression is another challenging thing in pancreatic cancer. So we were interested, like, can we use this TNF-alpha producing macrophages? Can we block this and get some immune response in pancreatic cancer? And before that, one thing we also noted that once you delete this TNF-alpha producing macrophage, there was no change in fibrosis or pre-metastatic niche formation, or no change in the fibrillar collagen in the pancreatic tumor. Yeah, to answer that question whether inhibiting this TNF-alpha producing macrophages can improve immune therapy. So we use again this model, and we wanted to do some preclinical trial. So our trial designs goes by, like, we let the tumor grow for 4 to 8 centimeter, and once they reach 4 to 8 centimeter, we divide these mice into 3 different groups. One group received just gemcitabine, another group received gemcitabine plus PD-1 and CTLA, and the third group received gemcitabine, PD-1 and CTLA-4 and plus the CCR2 inhibitor. And what we found is, like, once you use this combination, there was significant reduction in TNF-alpha in tumor microenvironment. And the triple therapy group survived longer than just gemcitabine or just gemcitabine and ICB group. And when we looked at their tumor microenvironment, what we found is, like, there was significant change into all this immunosuppressive population. We found a significant reduction in M2 macrophages, which are anti-tumor. We also found significant reduction in MDSCs, which are like SKICC positive. We also found reduction in neutrophils, which are another immunosuppressive population, and we saw increased migration of T cells into tumor microenvironment. And we also saw increased cell death. So overall, this suggests that once you inhibit this TNF-alpha producing macrophages, you get better immune therapy response, and you also have less metastasis. We also looked at the metastatic in this model, like what happens. And just to remind you, like, when we started treating these mice, they already had established metastasis. So this actually recapitulates whatever we see in the human patients. And when you look at this data, what we found is, like, the combination, the triple combination significantly reduced liver and lung metastasis. And there was an increased cytotoxic T cells in metastatic lesion, and there was increased cell death. We also saw increased dendritic, cross-antigen dendritic cell in the tumor microenvironment. And at this stage, we were curious, like, how it's happening, like, what's the mechanism of this response? So we did some TCGA database analysis on what we found is, like, since we have the high dendritic cell, we wanted to correlate this with any, like, in the protein level. So what we found is, like, IL-33 is actually highly overexpressed in triple therapy treated group, and IL-33 positively correlate with dendritic cells. And then our working model hypothesis was, like, IL-30 perhaps increase the dendritic cell migration, and that's the reason we see a reduced tumor and reduced metastatic lesions and increased immune response. But the story gets a little bit complicated here. IL-33 can be anti-tumor and can also be a pro-tumor molecule. So to see what's the mechanism in our model, we implanted tumors with recombinant IL-33, and we monitored the tumor growth. And what we found is, like, in our model, IL-33 reduces tumor growth. It increases the migration of CD8 cells into the tumor. And since I showed you that this is all happening because of the TNF-producing macrophages, so we thought that, okay, TNF-alpha, does it directly, we asked the question whether it directly affects IL-33 level. So in an in vitro experiment, we isolated cancer cells from this KPC mouse, and we treated them with TNF-alpha, and we measured the IL-33 level. And what we found is, like, a TNF-alpha indeed reduces the IL-33 level at mRNA level as well as the protein level. So overall, what we found is, like, the TNF-alpha producing macrophages actually inhibit IL-33 levels, and it reduces the dendritic cell functionality in the tumor microenvironment. And as the tumor heterogeneity is another important factor, we wanted to see, like, which cells are recruiting this TNF-alpha producing macrophages. To remind you, these are CCR2 positive macrophages. So these are recruited by CCL2. chemokines. So again, we generated primary tumor cell lines, metastatic tumor cell lines, and fibroblast cell lines from these mice. And what we found is like there's a tremendous heterogeneity in ability of these cells to recruit macrophages. And primary tumors secrete less CCL2 compared to the counter metastatic part. And when you look at the CAF, again, CAF are like heterogeneous population, there's MyCAF and ICAF. What we found is like ICAF secrete more CCL2 compared to MyCAF. So overall, what we found is like CCL2 is highly secreted by ICAF, followed by MyCAF, followed by metastatic cells, followed by primary tumor cells. And overall, our finding was that that CCL2, which is secreted by this ICAF, MyCAF, or tumor cells, they recruit this monocytes, which are highly TNF producing monocytes. They differentiate into macrophages. They produce TNF alpha into tumor microenvironment. And these cells actually, they reduce IL-33 in their cytosolic inside. And when these cells die, they die silent death, as IL-33 is alarming. When you block this TNF-alpha producing macrophages, whenever these cells die, they secrete a lot of IL-33 in the tumor microenvironment, and that attracts more dendritic cells. That makes tumor cells more antigenic, and you get better immune response. And no work is complete without, like it takes a village to do great science, and all thanks to all my lab members and all my collaborators who one way or other contributed this work. And I'm happy to take any questions.
Thank you, Dr. Dixit, for your nice talk. And I thank all the audience for their patience. There will be no discussion, no question now, because we are very late. And I take this opportunity, I and my co-chair take this opportunity to thank the organizers for good science and good hospitality. If there was any lapses on the part of organizers, you can forget it. Remember the good science, क्योंकि you forget it because बड़े-बड़े events में छोटी-छोटी बातें हुआ करती हैं.
Yes.
Thank you very much.
Thank you.
Thank you, sir. Thank you, Dr. Dixit. May I kindly request you to felicitate our speaker, Dr. Ajay Dixit, sir. We thank you for your patience. You have been sitting there for 1.5 hours. Thank you so much for that. Please. Sir, please. May I request you, sir, to kindly come to the dais? I now request Professor Mohammad Ashraf Ganai, Director and EOSG SKIMS, to felicitate our worthy chair, Professor Satya Das. Sir. Chairperson. I now request Professor Mohammad Ashraf Ganai to felicitate Dr. Riaz Ahmed Mir, our worthy co-chair. Thank you so much, sir. Thank you so much.
Mansoor, please join us. No, please. Sir, we know our team. Core committee. This is core committee. We know our team. Core committee.
I have this till 9 o'clock today, so don't worry. I told them nobody is coming here. Dr. Kamal, please.
I don't know what I'm doing here.
You are here.
Thank you so much, everyone. Please. I request all the delegates to kindly be seated. Ladies and gentlemen, we now arrive at one of the most cherished and important moments of Immunocon 2026, the Women in Science Presentations and Awards. The Indian Immunology Society has long been committed to recognizing and nurturing the extraordinary contributions of women scientists to our field. This session celebrates 4 outstanding awardees, one from each zone of our country, whose work represents the very best of Indian immunological science. To chair and preside over this special session, may I respectfully invite President of Indian Immunology Society, Professor Amit Avasthi, and Secretary of Indian Immunology Society, Dr. Kaustav Naik. Sir, please take your chairs. Yeah. Take it. Go in. No, okay, I got the thing. Our first awardee represents the North Zone. It's my proud privilege to introduce Professor Garima Khare, Professor in the Department of Biochemistry at the University of Delhi. Professor Khare's research is focused on understanding the metabolic and immunological mechanisms underlying tuberculosis pathogenesis, with particular attention to vaccine development. Professor Khare will speak on understanding the role of amino sugar pathway in the pathogenesis of Mycobacterium tuberculosis, a step forward towards TB vaccine development. Professor Khare, congratulations, and the podium is yours. Ladies and gentlemen, Women in Scientist Award.
Thank you. I just like to take a few minutes to tell about the genesis of this award. So this award we started in, we decided to felicitate women in immunology and by and large in science, because they contribute tremendously by doing other things, but they are kind of need to be encouraged and felicitated. So when we took over in Bangalore, Immunocon, and our EC decided to have this new award for the women in science. So first time we had this award in the Prayagraj Immunocon last year, and this year we have 3 recipients who will be presenting, and then after the presentation we will be presenting the memento and the citation. The other important thing regarding this award, when they apply, the independent committee actually sees and evaluate their application, and this is done in a very discreet fashion. So there is no bias against, for anyone or against anyone. So this is done in a manner the way we do for Senior Scientist Oration Award or the Mid-Career Scientist Award. So this is, I wanted to tell before Garima starts, Thank you. So, Garima, now stage is yours.
Thank you. So I would like to thank the organizers for giving me the opportunity to share the work that we do in our lab, and thanks to IIS for this award. So I am at University of Delhi South Campus. So I would like to begin with what we do and why we do. Of course, this audience does not require an introduction to TB and how deadly the disease is. I would just like to mention that TB is responsible for more than 25% of the total incidence rate globally, and is also responsible for around 30% of the total mortality globally. And that, of course, highlights the importance of the discovery of new intervention strategies, including the novel inhibitors and TB vaccines. My lab at University of Delhi South Campus is involved in various aspects of TB research, primarily understanding host pathogen interactions, how MTB persists and goes into dormancy, and how we can exploit that to prevent reactivation. We are also doing discovery for a lot of new antimicrobacterial inhibitors and TB vaccine development. For today's talk, I'm going to talk about intersection of two of these areas, how we started with a host pathogen interaction question and how it led to us with a vaccine candidate. So I'm going to talk about an enzyme which is basically N-acetylglucosamine 6-phosphate deacetylase, which is responsible for the synthesis of peptidoglycan precursors, or basically synthesis of amino sugars. And why we were interested in amino sugars was primarily because of its role in the mycobacterial cell wall. Mycobacterial cell wall is considered one of the most important virulence factors of the bacterium, and thus we started working on this enzyme. This enzyme primarily does a reaction which is the deacetylation of N-acetylglucosamine 6-phosphate to glucosamine 6-phosphate, which is an essential gatekeeper, which is an essential amino sugar and acts as a gatekeeper between the glycolysis and the cell wall biosynthesis precursors. So we were particularly interested in this molecule, and there were other organisms also wherein the importance of this particular enzyme was shown. So we started making a knockout of this gene in M. tuberculosis. So the NAGA gene, which encodes for this enzyme, was disrupted by homologous recombination, wherein we inserted a hygromycin-resistant cassette into this gene, and this is just showing the characterization of the mutant. The first thing that we noticed was that the colony morphology of the NAGA mutant was extremely altered as against the rough and serrated classical M. tuberculosis colonies. The mutant appeared very different, so we went on to do some more cell surface and cell structure architecture analysis. So we did TEM analysis, and we found that it can be seen here that while the parental mycobacterium and the complemented strain showed a very uniform and intact cell wall, the mutant had very diffuse and irregular cell wall. This makes sense because we were expecting that the peptidoglycan layer would have shown some alterations. And we also did scanning electron microscopy, and the analysis showed that the mutant colonies were slightly elongated as compared to the wild type and the complemented strain, suggesting that the mutant has definitely changes in the cell wall architecture and is also slightly growth defective. Then, since this mutation interferes with the cell wall architecture, we wanted to see what is the growth pattern of this mutant when we subject it to cell wall stressors. So we chose lysozyme for the obvious reason because it targets peptidoglycan and the cell wall, and the mutant displayed a lot of growth defect when it was grown in lysozyme, and similarly in hypoxic stress also it was growth defective. And we also interestingly found that it had impaired capability to form biofilm, which is also considered as one of the virulence factors. So all these in vitro experiments showed that it was a very growth defective mutant, especially in the host-related stresses. And since the cell wall was found to be compromised, we also wanted to see whether this mutation confers antibiotic susceptibility to the pathogen, and we chose a lot of antibiotics, and we found that we are just showing some control antibiotics and some of the relevant ones here. So we can see here that the mutant was found to be more susceptible to vancomycin and pyrazinamide, but not to isoniazid and ethambutol, which makes relevant sense because vancomycin and pyrazinamide both are related to cell wall alterations, and again suggesting that this is actually a very growth defective and a cell wall defective mutant. And since, like I mentioned in the first slide, that this makes a molecule called N-acetyl glucosamine, which is a essential gatekeeper between glycolysis and cell wall precursors. So when the bacterium is grown on mixed carbon sources, for example, in glucose and in glycerol, what happens that there is an appropriate catabolite division of labor, that one of the carbon sugars can be used for the synthesis of amino sugars and the other can be used for making growth and for other cellular metabolism. So we hypothesize that, let's say when we have mutated NAGA and we grow the organism only in a single carbon source, then what will happen that the glycolysis intermediates will also be pulled towards the peptidoglycan or the cell wall biosynthesis, and the effect of NAGA should be much more pronounced, and that's what exactly we saw, that when we grew the mutant and both the parental and the complemented strain in only glucose containing media without glycerol, we can see here that the knockout was unable to grow as compared to all other strains, again suggesting that the importance of NAGA and this pathway in the sugar metabolism and the glycolysis cell wall intermediates. And recently there was another study by a UK-based group which also showed, this was a study in M. smegmatis, in which they showed a pivotal role of this enzyme in salvaging peptidoglycan in mycobacteria, again emphasizing on this role. So we then based on all these in vitro studies, we then moved on to ex vivo and in vivo studies in animal models. So before going to animal models, we checked in macrophages, activated human macrophages, where we observed that there was a growth defect that was observed in the intracellular survival of the pathogen as well when this gene was mutated. However, it was not related to the phagosome-lysosome fusion capability of the pathogen. And then we wanted to see that what is the role of this enzyme in the in vivo virulence of the pathogen. So we chose the guinea pig model of experimental TB because of the relevance of this model in TB. So and then we infected the animals with the wild type mutant and the complemented strain, and we euthanized the animals at 5 weeks post-infection, at 10 weeks post-infection, and we observed that at 5 weeks post-infection there was indeed a growth defect of the mutant in vivo also, and this was involved in virulence, and this is the gross pathology of the lungs, liver and spleen, and it can be seen that the animals which were infected with the mutant strain showed minimal gross pathological damage as compared to the wild type strain, and this effect was more pronounced at 10 weeks post-infection, wherein we can see that there was a very significant 1.6 log CFU reduction, or to the tune of 2 log CFU reduction. And again, the gross pathology was much, much better as compared to the wild type strain, suggesting a significant attenuation of this strain in vivo. And most importantly, we also did time to death survival assay, wherein we saw that the mutant infected animals showed 100% survival rate, and none of the animals succumbed to death as compared to the wild type and the complemented strain. So based on all these studies, we also wanted to understand a little bit insight into the host pathway. So we did RNA sequencing analysis of the infected lung tissues of wild-type mutant and the NAGI complement. And we did host transcriptomics and made various analysis groups, and we saw that the mutant versus uninfected comparison showed very, very minimal alterations in the host transcriptome. And then we did a detailed analysis. This is again showing the volcano plots. Again, we can see here that the mutant versus the uninfected has very, very specific changes. And we did some pathway analysis looking deeper into the key pathways which were differentially expressed. I would like to mention here that we can see that there were some pathways like LPS-mediated signaling, type 1 interferon pathway, antigen processing presentation. They were altered in the animals which were able to take care of the infection with the mutant while when we compare it to the wild-type infected animals. This is again showing the alterations of the genes between a wild-type infection versus the knockout infection. And this is just showing a proposed model. Herein, I would like to emphasize that there is if we compare these two profiles, the mutant attenuation profile and the wild-type virulence profile, there is a distinct type 1 interferon upregulation that is seen in the virulence profile, whereas the downregulation is seen in the knockout attenuation profile. Similarly, a protective Th1 type immunity was there. Based on all these studies, there are of course a lot of things that we have found out, but going quickly to the inference of this, what we wanted to see that since it was a very significantly attenuated strain, we wanted to check its protective efficacy as a vaccine candidate. So we first quickly checked what is the persistence of this strain, and when we give this intradermally, there is persistent still 3 weeks, which is much higher than the BCG, and subsequently by 8 weeks, it is cleared from the system. And then we did the MTB challenge experiment using after vaccination with these strains, and it can be seen that this strain is providing a very, very significant protective efficacy in the animals which were later challenged with MTB. And while we are doing detailed immunogenicity analysis and some mechanistic studies for this mutant as a vaccine candidate, what we are now currently doing is that as per the regulatory requirements and as per the Geneva Consensus for making MTB-based live attenuated vaccines, it is required that the MTB-based vaccines should have 2 independent mutations at 2 different loci, and the final vaccine candidate should be an unmarked candidate. Unmarking means that it should not have any antibiotic resistance. So what we did was that we took this background strain, which was the NAGI knockout strain, and we have unmarked the hygromycin cassette out of this, generating an unmarked strain of this mutant. And here you can see that the strain does not grow on hygromycin. There are 2 colonies that we got as unmarked, while it grows on 7H11. So, and we have also done some more PCR-based analysis to confirm that the hygromycin has been removed. And further, what we are doing is that in this unmarked strain, we have selected a couple of 3-4 genes which we think might also be responsible in virulence to different varying degrees, because it should not happen that the second deletion should lead to a very, very highly attenuated phenotype which will not lead to enough immune response in the host. So we have selected a battery of genes, and we are making some double deletion mutants which will later be unmarked to produce the final vaccine ready candidates, and we'll be doing their extensive preclinical studies for protective efficacy and immunogenicity. Just to summarize quickly, this was the knockout generation, and we did some in vitro studies, ex vivo studies, and in vivo studies to and time to death experiments to quickly see the attenuation. And after seeing a significant attenuation and the role of this gene in the virulence, we had also looked at mechanistic insights of the profile of attenuation by the host. Just winding up, finished. And then we used it as a vaccine candidate, and it is indeed showing a good vaccine efficacy, and we are trying to develop final unmarked double deletion mutants which will be ready for further clinical development. With this, I would like to acknowledge my team at UDSC. This work has been primarily done by Neha, Dr. Prachi, and Ilaf is continuing the work further. And I would like to mention that this is the UDSC, BSL-3 and ABSL-3 National Facility for TB Research, which is a service facility for any TB investigator across the country. They can avail this for doing MTB-related pathogenic work or animal studies, animal challenge work or anything. And we are on and off also doing some training programs to increase the awareness of how to work in a BSL-3 in a containment lab to just get rid of the fear that people have of working with pathogens. So this is just like our outreach program that we do. Thank you so much.
Thank you, Dr. Garima. In the interest of time, we can have just one question. Mid-career में होता है sir.
Maybe they can discuss.
Yeah,
Anybody has questions?
Discussion
Can be there. Yeah. Thank you. Thank you so much. Thank you. Thank you.
Okay. Our West Zone awardee is Dr. Praniti Sar, Assistant Professor at the Nirma University. Dr. Sar's research explores how genetic and epigenetic signatures shape immune responses and influence disease outcomes. She will speak on host MHC2 genetic and epigenetic signatures associated with clinical disease outcomes. Dr. Sar, congratulations, and the podium is yours.
Thank you so much for the nice introduction. A very good evening to one and all present here. I'm Dr. Pranati, currently working as an assistant professor at Silver Oak University. First of all, I would like to thank Indian Immunology Society for giving me this recognition. And secondly, I would like to thank the organizer of Immunocon 2026 for giving this platform to present my research work. So basically, the title of my study is Host MSC2 Genetic and Epigenetic Signatures Associated with Clinical Disease Outcomes. So for this, we have chosen malaria patient sample as the study model. So as the WHO Malaria Report 2025 shows that the malaria incidence, the rate is going down from 28.6 in the year of 2000 to 13.8 in the year of 2024. But still, we don't have the effective malaria vaccine. And it shows a similarity in both the cases, that is Plasmodium falciparum as well as in the Plasmodium vivax. But the limitations are that the vaccines which are developed is not giving the effective immune response, and the death rate of the in case of children's is 72%, basically under the age of 5 years. And the limitations basically due to the anti-malarial drug resistance in parasites. So that is very difficult to eradicate completely malaria from the world. And the second is limitation for the development of an effective blood transmission stages malaria vaccines. And whatever malaria vaccines are developed are for the particular malaria parasite, that is Plasmodium falciparum. But currently there is no vaccines have been developed for Plasmodium vivax, ovale and Plasmodium malariae. And the most important point is the incomplete understanding of the mechanisms of acquired immunity modulated by the parasite. So basically today we'll discuss more about how the malaria immunity is regulated by the MSC2 molecule. So as it shows, the signaling pathway and immune cells involved in anti-malarial immunity is basically a combination of innate immunity as well as the acquired immunity, and the MSC molecules are present the antigen, malaria antigen to CD8 T cells and CD4 T cells, and if there is any defect in antigen presentation by the MSC molecules, the immune response cannot be developed efficiently. And we are basically focusing on the MSC2 molecules because the immunity to blood stage malaria is MSC2 dependent, and it is restricted to CD4 T cell activations. And basically RBCs do not express the MSC1 molecules. That is why it is mainly focusing on the MSC2 molecules. So recently the paper has been published and emphasized that how the MSC2 molecule is very important, although the vaccine is developed. Due to lack of this co-stimulatory molecule like CD86 and MSC2 molecules expression, the enhancing vaccine efficacy is in doubt. So before that, we should know what is MSC2 molecules. It is well known. It is present on the chromosome number 6. And we basically focus on the polymorphism present on the antigen binding site. But here I have worked on the regulatory variants, how the MSC2 gene expression is regulated at the promoter level. So MSC2 gene regulation is carried out by a small promoter, which is 150 base pair from the transcription start site, and having the transcription binding sites which is very specific, having SXY boxes, and the important transcription factor like NFY, CREP and RFX binds to a particular sequence, and that allows the C2A, which is the most important transcription factors, to sit and initiates the MSC2 gene expression. MSC2 gene expression later on undergoes the exogenous antigen processing pathway to present the antigen on the surface of the antigen presenting cells like the dendritic cells, B cells and macrophages. So our hypothesis of this study, if there is any defect in MSC2 gene regulation, whether that reveals the immunity to malaria or not. So why we think like that? Because when a plasmodium infect the individuals, they try to suppress the host immune response so that they can facilitate its persistence inside the host cells. So the immune suppression is the most important mechanism behind the malaria immunity, how it is down regulated in the malaria infections. So this is our study plan, where we have collected the blood samples from malaria patients as well as the healthy individuals. Then we isolated the genomic DNA, then RNA, and the plasma samples. From the genomic DNA, we checked the genotyping, then parasitic load, as well as the if we find any variants, then we cloned in PGL3 basic vector, which is promoter-less vector, to check the promoter activity. Then we check the RNA expression level for the MSC2 DRB expression and other genes which are involved in downstream immune response. Then we carried out the flow cytometry to check the expression of the HLA DRB on the surface. Then cytokine assays, then we correlate all the results. So first is the genetic variants. Yes, we got the genetic variants among the individuals which are malaria patients having minus 87 position, minus 70 and minus 57 position. Why it is minus? Because we are taking the promoter region, the regulatory region. So after getting these variants, we made the 6 different haplotypes from that populations. So this polymorphisms or whatever variants we got is in Y box and cat box and the tata box, which are very important region present on the surface of the promoter, which regulates the MSC2 gene expression. So based on this polymorphism, there are different haplotypes we found, around 6 different haplotypes. And from these 6 different haplotypes, the most prevalent haplotypes are the GT80, and which is present in both malaria as well as in the healthy individuals. And when we clone that promoter fragment into the promoter-less vector and we carried out the reporter activity, and we found the GT80 haplotype promoter reporter activity is showing the highest luciferase activity in comparison to the GTAC, which is less prevalent in the populations. So then we carried out and checked the promoter activity, what we are observing, whether it is showing the RNA-able as well as the surface expression. And we found the flow cytometry result that the expression is in the same line what we have observed in the promoter reporter activity. So the GT80 haplotype is showing the higher MSC2 gene expression, and GTAC haplotype is expression level is very low. Accordingly, the parasitic load is also correlated with the expression level of the MSC2 DRB. So where the DRB expression is high, the parasitic load is less, and where the expression is low, the parasitic load is high. So then we want to find out why these things is happening, why the polymorphism is there and that regulates the expression level, then why this causes the expression of the MSC2 is depending on the polymorphism. For that, what we checked, the polymorphism is mainly in the promoter region, so it might be affecting the transcription factor binding. So what we did, transcription factor binding prediction assays we did by using various softwares, that is Jasper, and we found certain important transcription factors are lost due to this polymorphism, and some transcription factors are recruited due to this variants. Then what we found, the important transcription factor, that is NFY, is binding to the GT80 haplotype, which is highly prevalent in the population, is binding at 3 different sites, and the binding affinities are also higher in comparison to GTAC. So it shows that the variation in the promoter region could not allow the transcription factor to bind, so that is why the HLA-DRB gene expression is going down. So this is at that stage we didn't stop. we want to find out this is not the only solution that the MSC2 gene expression is going down due to the polymorphism. There must be some parasite role that down regulates the MSC2 gene expression, because we are not getting the exact correlation of the polymorphism with the parasitic load. So that is why then we checked whether the parasitic load is affecting the MSC2 DRB gene expression or not. So for that, what we did, we checked the DRB gene expression as well as the C2TA with high and low parasitemia, and what we found, in case of higher parasitic load, MSC2 DRB gene expression is going down, and lower parasitemia, the MSC2 DRB gene expression is not that affected by the low parasitic load. So that gives you the idea that parasite has some certain role that MSC2 gene expression is affected by the parasitic factors. So then we checked, so it might be some epigenetic changes happening. So then we checked the chromosome 6, we checked all the chromosomes, whether they are affected by the parasitic mediated methylation or not, and we found chromosome 6, where the MSC2 gene is located, is having higher methylation rate. So that gives you the idea that might be some epigenetic modification is going on, and that is due to the parasitic load, and that causes the down regulation of MSC2 DRB gene expression. So this is what we want to summarize, that the genetic variants in the proximal promoter region alters the MSC2 expression, and MSC2 expression is not only regulated by the nucleotide variation, it's also regulated by the availability of the C2TA, which is the most important transcription factor that regulates the MSC2 gene expression. So as we have shown that parasitic load downregulates the C2TA expression, which is the most important transcription factor. If the transcription factor is not available, then how can it will initiate the transcription? So then lower MSC2 DRB expression leads to poor parasite clearance. That is obviously known that if the expression is low, then it cannot properly present the antigen to the surface of the antigen presenting cells, and hence the immune response cannot be developed. So there is less clearance of the parasite. So MSC2 gene regulation determines the immunity to the malaria. What we have hypothesized is proved here, and this is overall study what we have discussed today, that genetic regulation of MSC2 is a key predictor of malaria clinical outcomes. 72% of malaria deaths occur in children under 5. So this is the most important point we need to think about it, and all the limitations of the malaria vaccine, why it is not developed yet. So what we found, the haplotypes performance depending on the MSC2 gene expression, and we checked the chromosome 6 DNA methylation also. Then later on we found the parasite has some important role in down regulation of the MSC2 gene expression. So this is the future plan, what I want to do. So to identify the parasitic effector proteins which exactly cause the DNA methylations, and then we want to find out the inhibitors against that parasitic effector proteins. Then after introducing the inhibitors, we can reprogram the epigenetic modifications, and if we could successfully get the epigenetic reprogramming, then we can think about the drug repurposing of the small molecular inhibitor, which can be used in other diseases like cancer, where the immunosuppression is observed, as well as in the viral infections and chronic infections. So I would like to acknowledge all the blood donors, patients from the hospitals. We have taken the consent for our study, DSD Sir for providing us this funding, and Institute of Science Nirman University as a host, where I have done this project, and Professor Dalai was my mentor, and the students which are involved in this project. So I would like to collaborate if anyone is interested in this study. So basically malaria immunogenomics, epigenetics and chromatin regulation, therapeutic interventions, And this is all about the International Immunology Day we celebrate regularly in each and every year. For that, we would like to thank Immunology Society for providing us the funding to celebrate this Immunology Day. Thank you so much.
Thank you, Dr. Pranati.
Thank you, Dr. Pranati. Finally, we honor our South Zone awardee, Dr. Kartika Rajavi, who is a scientist E1 at the Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Her research focuses on studying host pathogen interactions and the mechanisms of immune evasion. Dr. Rajavi will speak on pathogen-associated neutrophils shape a long-living immune silent milieu. Dr. Rajavi, congratulations, and we warmly welcome you to the podium.
So good evening, everyone. So first of all, I would like to thank the organizers for organizing this. It's in a beautiful place, as well as thank you for the IIS for thinking about such an award or a recognition for women scientists in immunology. Thanks a lot. This will be a great boost for all of us to do science with more power. So today I'll be talking about a project that we had been working from 2021 since I joined the laboratory here in RGCB. So we have actually literally identified a pathogen-associated neutrophil population that is long-living in stages, and it is providing an immune silent milieu. So if you look into the pathogen, what we work, it's a bit new here, so I will just introduce it a little bit. So it's called Chlamydia trachomatis. It's a major cause of sexually transmitted infections. As you can see, there are nearly 129 million infections recognized or seen every year, new infections coming up every year. And nearly 70% of these infections remain asymptomatic. That means 1 in 20 women are positive for chlamydia. So not only does it infect the genital tract, but also it can infect any mucosal surface that you provide. So if you look into the bacteria, they are obligate intracellular. It's a little bit like virus. So they just grow only inside the cell. So they cannot replicate outside. So making mutants is really a pain. So as you see here, they just grow inside the cells, like it's called an inclusion. So they basically enter the cell as one specific type called EB. And once they get inside, they convert into EB-like structure, and then they start replicating. And once they replicate, after 48 hours, they can lyse the cell, or they can extrude out. Two major mechanisms by which they can go outside the cell. So this is a TEM image that clearly shows how the EBs and RBs looking like. And RBs, as you can see here, they are the replicating, the blue ones. They are undergoing binary fission. So not only that, this pathogen is actually able to infect the genital tract, it can also infect lungs, causing pneumonia. Then it's called chlamydia pneumonia. So there are several serovars. The most interesting fact is that we had been working so long with these cell lines, particularly the HeLa cell lines, the cancer cell lines in the laboratory to infect this pathogen. And so far, 18 years, I have not seen even one cell line that cannot be infected with this bacteria. So the infection looks like this. So this is a live cell imaging where you can see the bacteria really going in and infecting the cells. These are two different kinds of bacteria we have put, GFP and RFP positive bacteria. If you can concentrate on this particular cell, you can see two inclusions are coming together. There's a lot of biochemistry going on. They fuse together. They become huge. At the same time, they also let the infected cell divide. They don't interfere into anything. Finally, so you can see that happening. And now what will happen happen is they will lyse the cell, they'll come out and they'll start infecting new sets of cells. So is this really what is happening in our body is a major question, right? So what we did, we had actually grown fallopian tube organoids and then started infecting them. In red, what you see is bacteria, and we had to really go for a MOI of 10. That means a really huge amount to infect even. So normally MOI 1 gives fantastic infection, but in this case, we had to go for a very high infection rate. And if you can closely watch, not all cells gets infected. So there is some specific cells which is chosen by the bacteria, we do not yet know what, and they extrude, they do not lyse the cells at all. So this is what could be actually naturally happening in our uterus. So these organoids can be maintained for really huge number of days without affecting its stemness, basically. Okay, so this is what we see, and very recently we also found that this bacteria can also harbor your intestine. And not only that, if you look, if you try to grow these intestinal cells on a plate, we can see that they do not really infect the apical site, rather only go to the basolateral space. It's again telling you that they are very specific in their type of cells that they infect and where they infect. So it's not like what we saw in the laboratory that they infect any and whichever cell that we give them. So, okay, now let me come to the story. So, we were working actually with neutrophils for quite some time. And neutrophils, because the major reason why we work is that they are basically asymptomatic. So 70% of the infection remains asymptomatic. This is a major reason why this infection stays as a silent epidemic inside the society or in the population. So we started working with immune cells, that is the neutrophils. We isolated them, and in our first paper what we show is that when they infect, the bacteria is basically secreting a protease called CPAF, and this protease is able to cut or cleave away the surface receptors on the surface of these neutrophils, for example FPR2, and render them mostly paralyzed. Okay, so they do not react. So the neutrophils that seize the pathogen do not react to anything. So they remain completely naive. They do not react to PMA. They do not react to LPS, nothing. So basically they invite other pathogens to come and infect the space. So now our major question was, does this paralyzed neutrophils die or do they persist? So what we did, we isolated neutrophils, we infected them, and in red whatever you see is infected, in blue is uninfected cells. So it's a very simple experiment. So we did the FACS analysis. You can see that in the control, soon after like 12 hours the neutrophils are in your tube, they'll start dying. This is a normal procedure happening. So but we are infecting them. We can clearly see that there is a long lifespan for the neutrophils. They do not die. They are stable in the tube. I mean, this is just an in vitro experiment that we see. So now we know that there's an increased lifespan. So now the very basic question came. So since neutrophils are killers, are these bacteria viable, or is this just, what we see is just the first observation? So what we did, we just took the neutrophils. In blue, what you see is the bacteria, and in red, you see the neutrophils. You can see that a very small population is blue, interestingly. So we'll catch up with that later. So what we did, we took the neutrophils, infected them with chlamydia, what you see in green, and then we lysed after some time, and then we looked for the progeny to see if it is infectious. So what we see is definitely there is a big reduction, to be honest, very big reduction. That means the neutrophils are kind of killing some of them. And 12 hours post-infection, you can see in green is the bacterium. So we can see that the reduction is much worse at 12 hours. But later, see, 24 hours and 36 hours, you can see a resurgence. That means the bacteria can survive very well in these neutrophils. So now came the important question, what percentage of the human neutrophils can harbor the bacterium? So when we looked, this was a very interesting finding for the whole story, that we used MOI of 10, we could not see more than 20% of the cells getting infected. So now we increased to an MOI of 100. That means one cell, 100 bacteria. Still, not more than 20% of the cells will get infected. So now we call them bystander, the infected, and the bystander population. So we also did some immunostaining to confirm this finding. So now we know that there is a restricted population of subpopulation of neutrophils that can harbor the bacterium. The rest is not really getting infected. So now we wanted to see if this bystander and the infected population, can they both block cell death? So yes, the answer is yes. We sorted them out using FACS. the GFP positive and the negative population, and we looked for the cell death, and we could see that they can both block. So now came the very, very simple question. So maybe there is some secreted factors, that is basically preventing the bystanders from getting infected, right? A very direct question. So we just infected them. We took the supernatant, we put it in fresh cells, and we just checked if there's this affecting the cell death. And interestingly, in red, what you see is this supernatant from the infected cells. They can pretty well block and save the cells from cell death. So now, what is the identity of the cells? This was a major question. So we went for single cell sequencing. We actually did infected the cells and took them from 3 different patients or 3 different donors. And what you see here is very very new actually in the field. So in the uninfected population, normally neutrophils exist as 2 or maximum 3 population. That is depending upon the aging of the neutrophils. And once you get infected with chlamydia, you can see here. And well, after 6 hours or 24 hours after they are sitting inside the tube, when they are uninfected, definitely depending upon the stage of the cell death, you can see different populations coming up. But what is interesting here is the cyan positive population that is only seen in the infected population. You can clearly see that this population is like 8% 6 hours, 24 hours, it will come to 37%. So this is a new population that is set up in the neutrophils, which are basically supporting the growth. So this is what is shown here, the 7 different populations that are being produced during infection. And if you look here, so from where are this N7 population coming, right? So they should be from one of this population, must be derouting to produce this N7 population. So what we saw is this N3 and N4 population gets driven to N7 population. That's what we know at the moment. So now, is the bacteria specifically targeting is the next important question. So we went into the transcriptome, and we can see the transcriptome of N1 to N7. N7 was mostly interesting for us because this is the infected population. And we looked into all the genes that are present. And interestingly, one of the most highly upregulated gene was PI3. And then I thought, okay, PI3 is something which I know for ages. It's PI3 kinase, and it's highly upregulated upon infection. But it isn't. So we annotated it. It is called elafin. PI3 is otherwise called elafin. Elafin is actually a neutrophil elastase inhibitor. It's a very strong inhibitor of elastase. So if the elastase is blocked, the cell death is blocked, and this is exactly what we see. So could be. So then we just looked into the single cell profile, and you can see that the elafin positive cells are really enriched upon infection. And you can also see that the infected population is only showing the elafin. At the same time, the N7 population is showing the elafin expression. And we also looked for those cells which are elafin positive, were absolutely chlamydia positive also. Okay, so now came the very important question: is it really indispensable for the infection? So now came the wet lab experiments, where we basically infected the neutrophils with chlamydia and different time points, and we could see the mRNA level of elafin really high. At the same time, the protein level is also very high. So we also see that elafin is basically a secreted protein. So you can see in red that it is being secreted. So once elafin is secreted, neutrophil elastase goes down. That's exactly what you say here. The neutrophil elastase activity goes down. So we try to give recombinant elafin to just the neutrophils to see if it can protect from cell death, and they do protect it. And now we quenched the elafin using antibodies, and the cells will die. So basically elafin is blocked, the bacterial clearance will take place. So how critical is elafin? So you can't do any experiment with human neutrophils because you cannot do any genetics with it. So we had to adopt HL60 cells, which can be converted into neutrophil-like cells. So you can see even in HL60 nicely upon infection, elafin is upregulated. We used lentivirus to knock down the elafin, as you see here, and then infect them. And knocking down of elafin itself can deplete the bacteria. So elafin is absolutely important for the bacteria to live there. So we went to the mice models. The mouse do not have elafin. So we had to introduce elafin using lentiviral model and infect them. And we could see once the mouse is given elafin, there is more and substantial increase in the bacterial population, telling that elafin is somehow supporting the bacterial growth. So how is elafin regulated? So elafin is already known to be regulated in the literature using via the CREB and CBP pathway. And this is exactly what we see upon infection. The CREB pathway and the CBP pathway is highly upregulated. So now we simply introduced inhibitors to block both these pathways, and that's how you see it here. The elafin level goes down, and there is more cell death. And it's exactly the same with the second inhibitor also, telling you that this is really important, this pathway is really important for the regulation of elastin during infection. So now came how is CREB regulated. Everybody knows it's a cyclic AMP regulated protein, isn't it? So what we saw is a big surprise again that chlamydia do not activate cyclic AMP at all. So this was a big stop for us. We didn't know what to do next. So how to go forward? So chlamydia does not, then how does CREB activation now take place? Because this is also completely new in the field. So we went into the literature to really figure out. From our earlier publication, we knew about this paralysis effect, right? There is nothing happening. There's no receptor recycling happening inside the infected cells. This could happen only when beta-arrestin, for example, gets deubiquitinated or upregulated. So we look to the level of beta-arrestin and tada, the beta-arrestin is highly upregulated and highly deubiquitinated also. So when something is deubiquitinated upon chlamydia infection, we clearly know what happens. So there is a protein that is responsible for this deubiquitination process that is called DUPS or CLADUPS or chlamydial deubiquitinase 1 and deubiquitinase 2. So this is a kind of not so well studied, but people know that they have some very important role in immune evasion because the mutants are viable. Oh, I need few more minutes. Yes, I will. So what we did is we basically overexpressed CLADUP, and we could see that the protein, the beta-arrestin gets upregulated. At the same time, it gets deubiquitinated. And if you knock down or the catalytic domain, you can see that the beta-arrestin level is going down, indicating that this is a mutant, which clearly shows that there is a beta-arrestin is really responsible for this whole thing that we see. So now this is kind of the final slide for me. So here we have actually used all the different strains. So this is the wild type bacteria. You can see that beta-arrestin is up, elastin is up. This is a mutant, and we have used the recombinant bacteria as well as the recomplemented strain. So in all these cases, you can rescue the effect. Actually, you can rescue the neutrophils only by incubating the recombinant elastin or using recombinant CLADUP. So what we clearly show is that the bacteria can go in and can activate this whole pathway and increase, leading to the secretion of elastase and can block the cell death, not only of the infected cells, but also the whole population. So two more slides. Because this is a completely new data from the laboratory. So we wanted to publish this, but we didn't want to see this major question: how conserved is this neutrophil heterogeneity? Is this only for chlamydia or for other pathogens also? Then God's grace that there was a nice paper which came out in 2024, which actually did the single cell sequencing of MTB in whole blood cells. Okay, so we analyzed the sample and we could see what comes up is really PI3. PI3 is highly upregulated even in the case of MTB. So we simply did one simple experiment to just see. We could not do this at a very high MOI. MOI of 5 or 10 will it's devastating. It will completely kill the neutrophils. So at a very low MOI of 1 or 0.5, we can see that they can block the cell death, as you see here. And at the same time, again, only very small population gets infected. And you can see PI3 is highly upregulated and elastin level goes up upon infection. And cyclic AMP, everybody knows it activates cyclic AMP. At the same time, the whole pathway is activated by this pathogen also. So this is a very conserved pathway that this bacteria, which do not really explicitly have the LPS, uses to evade the immune system. So if you use any of the inhibitors, you can clearly see that the pathogen can be, can be killed, or there is a better clearance. So the relevance is that we have now investigated and found that the bacteria, that the human neutrophils exist, at least 7 different subsets are there, and they exist into a long living phenotype. And there's a new mechanism that we found, that is CLADUP is deubiquitinating beta-arrestin to regulate elastin. And obviously, if this elastin-mediated elastase inhibition could be a targetable mechanism for immune evasion, which can be a druggable target, basically. So thanks, first of all, to IAS and Immunocon for giving the opportunity here, and of course to the kind collaborators. So this was a very big teamwork from a lot of people actually in Denmark, in China, in US, and so on, and also from a lot of effort from RGCB. And this whole work was actually the wet lab work was done by my PhD student, a very brilliant one, Paridhi Agarwal, who will soon keep her thesis. So also to the other PhD students and of course to the most important funding agencies who funded during this time, and also the fellowships that I received during this time. Thanks a lot.
Dr. Rajavi.
So my name is Rajiv. It's not Rajavi.
Sorry. May I request Dr. Pranati and Dr. Garima to be on the stage, and all the EC members present here, please join us.
That's why we went to mycobacteria.
Congratulations to all the awardees. We now, after I think 5 minutes, we are going to convene the general body meeting of the Indian Immunology Society. Non-members are welcome to step outside and avail of the refreshments, if any. Please.
Folder है treasurer और वो report बोल के folder है क्या? Folder treasurer and report है कोई? ये रहा secretary and treasurer report.
खुद ही convene कर लेंगे.
हाँ, yes.
हाँ, हम लोग कर लें.
हाँ, yes. Yes.
मैंने पेन ड्राइव सेव कर दिया. हाँ, सेक्रेटरी जाएगा. मिनिमाइज कर दो उसको, पहले सेक्रेटरी करेगा.
आप कर लो.
नहीं, पहले सेक्रेटरी. ठीक है, पहले सेक्रेटरी. ये कौन नहीं पहले?
नहीं, दूसरा है.
इसको मिनिमाइज कर दो.
नहीं, और उसके बाद GBM. पहले रिपोर्ट सबमिट करेंगे, फिर GBM.
नहीं, GBM तो स्टार्ट है.
नहीं, एजेंडा बाद में.
GBM एजेंडा बाद में आएगा. पहले आप सेक्रेटरी रिपोर्ट.
हाँ, वो सेक्रेटरी रिपोर्ट खोल दीजिए.
पहले ही चलाएंगे इसमें. वर्ड ही चलाएंगे. मैं PPT खुला हुआ है ना, वो बाद में उसके बाद में 5 मिनट का प्रेजेंटेशन कर ले. चल जाएगा.
अच्छा, ये ऐसे चलेगा.
ऐसे ठीक है, वैसे चला रहा है. हाँ ठीक है, कुछ ठीक है. ऐसे दिखेगा बड़ा बड़ा.
अच्छा, इसमें उसमें भी 2 दिखेगा.
हाँ, सिंगल दिखने दो, पढ़ लेंगे.
हाँ, सिंगल दिख. काला क्यों है?
नहीं, अभी मैंने उसमें नहीं डाला. ऐसे रीड कर दो सिर्फ पीपीटी में आराम से अच्छे से
किस में पेस्ट करने पे?
नहीं आपने पीपीटी में पेस्ट कर दिया था ना, पहले कर लिया था।
इसको क्रॉस कर दीजिए पीपीटी चले
नहीं नहीं अभी क्या स्क्रीन शेयर नहीं हो रहा है?
स्क्रीन पे
हाँ स्क्रीन पे डाल दीजिए।
अच्छा वहीं से डलेगा?
वहीं से डलेगा। यहाँ बैठेगा कौन?
हाँ अमित कहाँ गया?
तो पता नहीं, अनाउंस करो ना। हाँ कुछ चला गया मुझे। मैं तो बैठू जाके।
हाँ So we can start the proceedings of the general body meeting. Yep, Dr. Amit, Dr. Deepak. And I request all of you if you can come forward. Yeah.